Hi, I'm Dusty Donaldson, founder of the Dusty Joy Foundation. We have a network of educational patient groups. Terri Conneran has been a member of our Charlotte group since that group started about 5 years ago. On December 5th, 2019, I had a discussion with some pharmaceutical representatives who asked if I knew an advocate with the KRAS biomarker. Even though KRAS is the largest biomarker by a long shot, there was no KRAS community. The next day, who walks through my office door but Terri? She had just learned that she has the KRAS biomarker. The rest, as they say, is history. Terri is a trailblazer. She created a community that brings together patients, caregivers, researchers, and industry partners. She works day and night to make KRAS Kickers more meaningful, more engaging, and more impactful. The KRAS community extends beyond lung cancer and beyond the U.S. KRAS Kickers brings hope to people around the world with all types of KRAS cancers. Continue sharing hope, all of you KRAS Kickers, because as you know, hope shared is hope multiplied.
Good morning, everybody. Hi, I'm Terri Conneran, and I'm with KRAS Kickers, and I am beyond excited that you guys are all here. Okay, we have, over the course of the past year, we have all faced cancer, we have faced COVID, there's monkeypox, and we have, and now we have a hurricane. And in spite of all those things, and despite all those things, y'all are here. And not as many of us are here in this room as we were originally planning for or hoping for. And those folks we really miss. We really miss you guys online, but that's okay. We've got rooms and we've got surprises for y'all later on. But at the same time, the people that are here, I gotta tell you, I don't know how excited you are, but for me, this just fills my heart. It fills my heart in this room to see each and every single one of you, and hoping that by being here and connecting with each other, we really do know, and we can really share that hope with each other. And above and beyond that, work together to learn about, to know KRAS, right? Encourage your research, get the resources, and build alliances for more survivorship. And as we like to say, cancer's KRAS. But I like that you guys are here. And over the course of the past year, we've had the opportunity to see a number of different doctors and do a lot of different online learnings. Finally, I was able to get out and about. COVID had, shall we say, clipped my wings along with everybody else's. And so now it's an opportunity for us to get engaged, us, the organization. When I say us, I mean us. I do not mean me. I'm the one that's able to run around because it is an honor and a privilege, the fact that I'm able to be here and be able to do this. But for right now, that's what I can do. And everybody's got their role, and you guys are all doing what you need to do. And truly, thank you guys for being here, because it's just really heartwarming and spirit-filling to really do that. So as we've got, and we're going into today, we have a number of just different amazing speakers. I don't know if you had the opportunity to take a look at the program or anything. I'm going to go over it like super high level because, like I said, more people are online than they are here, but that's okay. We love them too. And next year, hopefully we can see them in person, and we'll figure out a way to do that. Starting up today is going to be Channing Durr, Professor Durr. I'm going real high level to give you an idea of what the lay of the land is. Professor Durr is the person that found historically the existence of this KRAS thing, that it is a problem. And it's a privilege that I'm going to be able to include, that he is agreed to attend and be here. Later on, a little bit later on today, let's see, I'm going to check this out because so I don't mess anybody up. Dr. Skoulidis is going to be here from MD Anderson. And he's gonna be talking about some of the new emerging things, and he's a real treat to listen to. Later on, we do have Dr. Kristen Marrone from Hopkins. So as you can see, we're not slackers. We've got the top tier docs, got the one that started it, and we have the ones that are learning about it and moving things forward, moving the needle, as it were. Then later on, we have Dr. Daniel on from Mayo Clinic, which is going to be talking a little bit more specifically around colorectal cancer. And we've got a treat as we learn about how drugs come to market. And Amgen, I can't turn my page. Sumita, Bhatta, and pardon me when I butcher names, but I should have made better notes. This afternoon, we've got better understanding around the clinical trials process and what that means. And we're going to be treated with not only a doctor, but a panelist from several people in this room, including Brenda. And we have Eric Madsen who's going to be joining us online. And then we'll be ending the day with integrative oncology and therapies and what that means, what that looks like. So how we can truly live with this crazy disease that we have, illness. And then I'll be closing the whole thing out. So you guys are going to be stuck seeing a little bit of me today, but I hope you don't mind, because I certainly don't mind seeing your faces. And being up here, I get to see everybody's face. So, all right, with that, if we've got our speakers online, just give me a thumbs up. Yeah, okay. With that, I'm going to go ahead and officially introduce one of the most humble men that you're ever going to know. And one of the people that truly we would not have been able to be where we are today unless he had done the research and was the one brave enough to kind of step up and recognize it. He's also in Carolina. He's over at Chapel Hill, University of North Carolina. And I'm looking desperately, just hang on, hold on, we'll get it. Oh my gosh, I can read all that. Dr. Channing Durr is Sarah Keenan's Distinguished Professor at the University of North Carolina in Chapel Hill. That's a big deal. He received his PhD from University of California in Irvine. He's done postdocs at very impressive places such as Harvard and Dana-Farber. And you'll notice in the booklet, it says, since his initial discovery of the RAS oncogene. Put power into that. And here he's going to go a little bit over a little high level, see what's going on and how we got to where we are today. And it is with that, rather than reading all the specifics about how incredibly amazing and distinguished and powerful he is, it is with great pleasure that I can introduce to you my friend and the amazing Dr. Channing Durr. I mean, welcome. Thank you for joining us today. It's so good to see you. And I had the pleasure of seeing you earlier this week, but thank you for joining us online today.
Well, thank you very much, Terri, for that wonderful introduction. And I'm truly honored, and it's a great pleasure for me to be involved in this organization. And I hope that I can continue to contribute to the cause that KRAS Kickers is perpetuating, because I think it's a wonderful and very timely effort. So as Terri indicated, I've been involved in RAS research for many years now. And Terri already gave you a brief introduction, but I'd also like to just sort of give you a glimpse into what we do. And so, yes, I was involved in initial discovery of KRAS in cancer way back in 1982. And so our research is focused on trying to understand how RAS drives cancer in order to utilize that information to develop effective anti-KRAS strategies. So our research centers on the cancers at the highest frequency of KRAS mutations, pancreatic, lung, and colorectal cancers. And the ultimate goal of our research is to transition our findings from the bench to the bedside, to initiate clinical trials, to try out new ideas to block KRAS for cancer treatment. And in addition to what we do in the lab, an equally important part of our mission is community outreach, patient advocacy. We feel that this involvement with the community is critical for what we do, because at the end of the day, why we're doing this is to try to help the cancer patients. Okay, so this is just my disclosures. I've been fortunate to be involved in a number of the companies that are now embarked on the development of KRAS inhibitors. Okay, so this is a outline of what I want to cover today. And so the first question I want to address is, what is KRAS? And so KRAS is actually one of 3 RAS genes. But it is perhaps the most significant one in cancer because of the 3 RAS genes. They're all mutated in cancer, but KRAS is the king of RAS genes. It's the most commonly mutated of the 3 RAS genes in cancer. And so the additional basis for KRAS importance relative to the others is that when one looks at the frequency of the 3 different RAS genes here, by far, KRAS is the most commonly mutated. It's mutated overall in about 80% of all cancers. And the other important thing is, if you look, the panel on the right is a list of the major causes of cancer deaths in the US. Numbers 1, 2, and 3 are lung, colon, and pancreatic cancer. These cancers have the highest frequency of KRAS mutations, and KRAS dominates the landscape of the genetic mutations in these cancers. Okay, so just a brief glimpse into what KRAS does. Cancers have not 1 mutation or 2 mutations. They have many mutations. And this panel here on the left just indicates that different cancers have different mutations. But the key thing that you need to know is, of all the genetic mutations in cancer, KRAS is the king of mutations, as indicated in this cloud image here on the bottom right. And so because of the significant frequency of KRAS in cancers, in particular, are major causes of cancer deaths, many in the cancer field believe that this is one of the key holy grails of cancer drug discovery. So just a little bit of science here for you. I don't want to get into this too much, but because G12C is now a mutation that's talked about, I just want to emphasize that G12C is but one of multiple mutations in RAS, and that there are multiple hotspots in the protein, shown here on the right, where these mutations occur. So there are a number of mutations, and this becomes important as we talk about targeting KRAS in cancer treatment. Okay, so this is KRAS made simple. KRAS, what KRAS does is actually, it works as a simple on-off switch. When it's on, cells grow. When it's off, cells stop growing. And normal cells maintain some control over their growth. In contrast, in cancer cells, KRAS is mutated. And basically, the net consequence of mutations in KRAS is that this on-off switch is stuck on the on position and resulting in cancer cells, which now have uncontrolled growth. So moving on to the second topic, drugging KRAS. So KRAS, we've known about KRAS now for nearly 4 decades, discovered in 1982. So where do we now stand in developing KRAS drugs? So I'd like to call the journey that we have taken as researchers the long and winding road. And those of you may recognize that I stole this from the Beatles' 20th and last #1 hit. And when Paul McCartney, who wrote this song, was asked, what's the significance of the long and winding road? He said, it's all about the unattainable, the door you never quite reach, the road that you never get to the end of. Well, for many of us in the RAS field who have been desperately searching for the elusive anti-RAS drug, it seems as if we can't seem to quite get to the finish line. So just a bit of information about the process of drug discovery. Drug discovery is indeed a long and winding road. There are many steps in the process, from the initial identification of a mutation in cancer And then you go through many steps in developing drugs, and then you test them in cell culture models and in animal models. You go through clinical trials, and if the results are promising, hopefully the FDA will approve the treatment for cancer. So this is a long and winding road because on the average, it takes at least 10 years for a new medicine to complete the journey from the initial discovery to the marketplace. And so why does it take so long? Well, there are many steps. But the other thing I want to emphasize is that the odds of success are very, very low. For every 10 to 15,000 ideas that we have in the research bench, only one will actually succeed and traverse through all the important steps in trying to determine whether this is an effective approach for cancer treatment. So 1 in 10 to 15,000, these are terrible odds. Now, they're certainly better than your odds of winning the lottery or being hit by a meteor. However, you're more likely to be hit by a car or by lightning or winning on the slot machine jackpot than making a successful drug. So the point is to emphasize that this is a tough business. Okay, so long and winding road. I've been on this road since the very beginning. And it's clear that we have at many times underestimated the complexities of RAS. We certainly have made mistakes. We've had misconceptions and we've had missteps. So in this long and winding road, because of the failures along the way, RAS developed a reputation as an undruggable target. And so one of the reasons people consider it undruggable is when one looks at the protein, it simply is not attractive for drug development. My colleague at UNC, Adrian Cox, when she was interviewed on National Public Radio to talk about efforts to try to develop KRAS inhibitors, she said RAS is a greasy ball, meaning it's something that's really hard to get a hold of. Okay, so because of some of the challenges that we have faced to develop anti-RAS drugs, sometimes in the field, we have considered this potentially an impossible mission. Okay, so we have certainly tried many different strategies, and this slide here just summarizes some of the key strategies that have been taken through the decades to make KRAS drugs. What I'd like to emphasize in this presentation is the number 2, the direct inhibitors of RAS. And so the reason I bring this one up is because this is where the breakthrough began. So seminal studies by Keyvan Shokat and colleagues back in 2013 made a remarkable and amazing discovery, and he identified the first small molecule that can bind to RAS and block RAS function. So this set off tremendous excitement in the pharmaceutical industry, and many companies then embarked on pursuing the advanced development based on the Shokat observations to develop KRAS G12C inhibitors. And so in 2021, a milestone in KRAS drug discovery was achieved. The very first KRAS inhibitor was finally approved for the treatment of cancer. Now, I should emphasize that the very first KRAS inhibitor does not target all KRAS mutations. It targets only one mutation called G12C. And additionally, so far, it's only approved for the treatment of lung cancer patients with this mutation. There are additional KRAS G12C inhibitors that are now being evaluated in the clinic, and hopefully soon a second one will be approved and perhaps more to come. So let me bring you up to today and where we imagine the future to be. So with this milestone discovery of the very first KRAS drug, are we done now? Have we finally climbed the Mount Everest of cancer research? While this is certainly an important milestone, we have many questions that we still have to answer. First of all, not every lung cancer patient with this mutation responds. Only about 35 to 40% of patients respond. So the question is, why do the other some 60% of lung cancer patients with this mutation not respond? Can we figure this out and then determine and then make them respond to the drug? I tell you, it's only been approved for lung cancer. Can we extend the success of these drugs to cancers beyond lung cancer, such as colorectal cancer, pancreatic cancer, and so forth? The other thing is, I emphasize that the current drug is only approved to target only one KRAS mutation. There are many KRAS mutations, as I mentioned at the beginning, and there are also mutations in the other RAS genes as well. Are we going to be able to develop drugs to target other RAS mutations? And the last issue, of course, is one that we face all the time in oncology drug development. Will resistance be a limitation? So, and with regard to that last question, indeed, despite the dramatic results that are being seen with the KRAS G12C inhibitor, shortly thereafter, reports that the patients who responded initially relapse and they develop resistance to the treatment. So on the average, patients respond for about 6 months until the cancer comes back. This is not a surprise because essentially all oncology drugs are limited by treatment-associated drug resistance. And so at this point, the solution to this is drug combinations. And this is the classic approach in cancer chemotherapy, to develop cocktails of drugs to make the combination much more effective. So the ongoing clinical trials now are not just evaluating KRAS G12C inhibitors, but these inhibitors with combinations of many other drugs. So these are ongoing studies, and fingers crossed that some of these combinations will help advance and make these type of treatments much more long-term, much more durable, and hopefully resist the concept of drug resistance. Okay, so what about other RAS mutations? These are exciting times. There are many companies that have now embarked on a development of even more KRAS inhibitors, and we now have some early stage preclinical findings that say that, yes, we can develop inhibitors against other RAS mutations. So the clinical trials to test these other KRAS inhibitors are now beginning. And so these are certainly exciting times. And with a bit of luck, hopefully we will soon have KRAS inhibitors, not just to target KRAS G12C, but other RAS mutations as well. So let me just end by saying that KRAS has certainly been a major challenge. I've done research on KRAS now for 4 decades. I borrowed this quote from Winston Churchill, and I call KRAS a riddle wrapped in a mystery inside an enigma. And so while we have learned a lot about KRAS, and we have now succeeded in developing at least one KRAS inhibitor, we are still in the early days of KRAS drug discovery. So while these are exciting times, I want to caution that we still have a long ways to go. And so we anticipate that we will continue to learn and that there will be more surprises to come. So I think we're just seeing the tip of the iceberg of what KRAS is. And as we begin to understand further what KRAS does, we will hopefully be able to harness this information and develop even more effective KRAS therapies. So I'll stop there, and I thank you very much for your attention.
That was wonderful. Thank you so much, Channing. I really appreciate your being able to kind of like highlight some of the different things and how we kind of got where we are here today. Do you have a minute to answer a couple of questions and kind of give us a little bit of guidance and your expert tutelage, as it were?
Of course.
Okay, well, that's great. Now, I'm not sure how promising it makes it sound when you're like, oh, I have a better chance of winning at a slot machine than I do of getting my drug approved. So, but I also know from real life experience, you can't win if you don't play. So it looks from your slide like there's an awful lot of people in the field. Can you kind of like just go over what your thoughts are on that?
Yes, so you're completely correct. Despite the fact that when we, as researchers, have looked at RAS, and it has been a daunting and challenging, and if not scary, and very humbling situation. But the reason many of us go into research is because we like the challenge. We want to do something that has never been done before. And because of that, we have stuck with it, and we have done research over the last 4 decades to try to do something which many people considered impossible. And so with the successes in recent years, the number of researchers that have now continued to come into the field is just remarkable. In my 40 years of research on RAS, I've never seen the level of enthusiasm and excitement in terms of trying to push the needle further to develop even more KRAS inhibitors. So yes, it is a tough challenge, but us researchers are excited at the odds, despite the low odds, and continue to make significant efforts in these studies.
Oh, okay. Well, that's a good answer. And so we need more to keep playing, and the competition in this field is a good thing. So even though it makes it a little bit more complicated and hard for us to understand at a patient level, because let's be real, none of us knew about KRAS until we in fact got diagnosed with something having to do with it, which is a cancer of some sort. We need them to keep going. Okay, I can see we've got a couple questions in the chat, so I'm going to read the question to you, and so we can kind of then I'll let you see if you've got any information around it. And it says, good morning from Canada. Oh, okay. Thank you for putting all this together. Let's Kick Cancer's KRAS. Don't worry, we're not going to let you get out of here without saying that too. So just like a heads up. So like you can start warming up a little bit, okay, with this cold, damp weather that we're all facing. Okay. And see, that's kind of not really your thing about the clinical trial and study specifically targeting any of the particular specific mutations. If you know anything that you'd like to kind of like share around that, that'd be fantastic.
So most of the activity right now is against the KRAS G12C mutation, which is the most common mutation in lung cancer. Unfortunately, for other cancers, this mutation is relatively rare. In particular, pancreatic cancer, where virtually every pancreatic cancer has a KRAS mutation. Unfortunately, the G12C mutation represents but 2% of KRAS mutations in pancreatic cancer. But there's hope. And a new KRAS inhibitor entered clinical evaluation back in June. And this KRAS inhibitor is developed by Revolution Medicines. And this KRAS inhibitor can potentially target every KRAS mutation. And so the trials have just begun. People are really, really excited to see what's going to come of these, because if this drug works, then we could potentially treat every patient with a KRAS mutation, not just those with G12C mutations, but any KRAS mutation, and potentially even an NRAS mutation or a HRAS mutation. So that is certainly one of the most exciting ones out there. There are several companies that have drugs that target G12D. G12D is the most common KRAS mutation. It's the most common one in virtually every cancer, from pancreatic to colorectal cancers. And so that is another one that is generating considerable excitement, and it's expected that some of these will begin entering clinical trials towards the end of this year.
That is exciting. And the reality of it is that what we've seen, okay, and by we, I'm almost including myself in an inappropriate group, because as a patient, I'm just looking for what's going to help me, what's going to help the community. But I know that you're able to kind of like stand back and take a look and see what's helping all of us. community-wise, right? You're looking at the whole big group of the ocean that we're able to face. And so I'm excited. I mean, I'm not like just a little excited. I'm really excited that we had the breakthrough. We had the initial start, and we're able to start looking at how we can start attacking those pathways using combination therapies. And I think that that really is just truly where it's going to be going. Let me kind of glance around the room. Does anybody in the room have a particular question? Like, I'll just walk up to you with a microphone, because like, I'm not afraid to just put you all on the spot. Does anybody have a question though? Yes, come on, don't be shy. I promise y'all, they're going to get a little bit more enthused as the day wears on. We get them a little bit more caffeinated, and a little bit more warmed up. We do have a question, and this is an excellent question. It says, can you please explain the difference between HRAS, NRAS, and KRAS? And what is it just go do your professor thing and tell us the difference, please.
So KRAS, HRAS, and NRAS, they're all genes that they share a lot of relationships, and that's why they share a similar name. They're all mutated in cancers, but they're mutated in different cancers. So I mentioned that KRAS is most commonly mutated in cancers such as pancreatic, lung, and colorectal cancer. NRAS is not common in these cancers, but we see NRAS mutations more commonly in melanomas, acute myelogenous leukemias. HRAS is found in yet different types of cancers. HRAS mutations are found, for example, in head and neck squamous cell carcinoma, bladder cancers, and several other cancers. And so the different RAS mutations all do the same thing. They're all cancer drivers. They all cause cancer, but they seem to occur in different types of cancers. And we don't know yet why that is the case. We suspect is that they each do something slightly different.
Is it ever possible for like one of those NRASs, HRASs, or anything to turn into a KRAS?
So that's an interesting question. Some of the patients that have been treated with the KRAS G12C drug have acquired resistance mechanisms that involve mutations in NRAS. So yes, there certainly is crosstalk between the different RAS genes.
Okay, and does that only happen as a result of being on a medication?
I'm sorry, could you repeat that again?
Does that only happen, or does it get that acquired resistance or change the mutation only as a result of being on a medication, or does it happen spontaneously?
That's an excellent question, and the answer is probably both. Some mutations are pre-existing, and some mutations are likely due to the treatment itself. The thing you have to understand is that cancers are genetically very unstable. They're like genetic time bombs, and they accumulate mutations at a rapid rate. And this allows them to escape treatment with different drugs. And so the resistance mechanisms are both present and pre-existing, but also caused by the treatment itself.
Okay, and one last question. All right, well, actually one last question, and then we're going to get your request. Okay, so this KRAS is something that happens in our body naturally, and then it starts out like everybody's got a KRAS thing, and you keep referring to the genetics. Is the genetics part of this mean that it's can be passed on to my kids?
Yeah, so that's an important question that I think people need to understand. So KRAS is actually a good gene, but when it gets mutated, it becomes a bad gene. And so we all actually need KRAS to develop as normal humans. It's when it becomes mutated. So cancer basically is a process where good genes become bad genes.
Okay, well, I think that was an excellent way of answering. Oh, you have a question? Hang on. We're gonna put you on microphone.
And how does the cancer get mutated? Like, what are some of the factors that make it grow or spread?
Okay, so KRAS becomes mutated. So majority of people do not have the KRAS mutation when they're born. It usually occurs during your life. And the mutations occur due to environmental carcinogens. So for example, the KRAS G12C mutation, I mentioned that it is the most common one in lung cancer. It is a cigarette smoking associated mutation. So different carcinogens will cause different KRAS mutations in different cancers. Now, the RAS mutation alone is not enough to cause cancer. You need additional mutations in other genes before cancer arises. And that in general is why cancer is a disease of old age. You need to accumulate multiple mutations. The general guess is that you need 5 to 10 mutated genes to have a full-blown cancer. So KRAS mutations alone are not enough, and you need to accumulate additional mutations in other genes. And so while cancers require the mutations in many genes, the good news is that if you can correct just one of them, for example, the KRAS mutation, that alone is enough to collapse the cancer.
Okay, well, we could stay here all day asking you a whole bunch of questions because you're just so great at answering the questions, and we really appreciate that. But we're not going to let you get out of here. I don't know if the other folks that are here remember what we did last year, but we asked our speakers to participate in a quick little video snippet where they got to say "Kick Cancer's KRAS," and we got to do it together in a room. And it's not that we really want to cheer. It's just really that we want to feel like you know how important this is to us and for all of us. And so if the room will cooperate with me in doing that, and you would do it as well, I kind of like do a whole count off thing. Okay. And so we like 1, 2, 3, and then kick cancer's KRAS. Y'all with me on that? Okay. All right. All right. Here we go, y'all. 1, 2, 3. Kick cancer's KRAS. All right, you guys. Thank you so much. Thank you for joining us this morning, Channing. And I hope to see you again. Stay warm and stay safe. Okay. All right. I think we are just so I make sure that we have speakers coming up next. We're good. Okay. I just want to make sure, and I want to keep everybody on track. That was excellent. And I just cannot begin to explain how miraculous it is that it takes somebody to find this teeny tiny thing. I mean, last year we learned it it's like over a quarter of a million KRAS mutations in order to get just to the tip of one hair. That's how this starts. And so for Channing Durr and his lab and his team to be able to have identified this 40 years ago. Okay. And it was 40 years ago. Using that technology, that's one reason why we're coming better, farther, faster. And even though, like myself, I'm not a G12C, it also gives me a lot more confidence in knowing that more things are coming. We're learning what we need to do, and we're able to start targeting like these things in a panel sort of a way in order to be able to make a difference. All right. And with that, I think we're even going to stay pretty much on time, which is pretty miraculous, all things considered. I'm kind of proud of myself for that, but the day is early. So we have from MD Anderson. It is really kind of a super treat for us to be able to get this amazing speaker. All right, Dr. Fernandos, Skoulidis, whose name I'm totally butchering, and I'm so sorry. I didn't write down a better role of it, and he's smiling at me, and I appreciate your kindness. And he received his medical degree from Aristotle University in Greece in 2000, and his PhD. Well, that's an amazing place to be, and a PhD from the University of Cambridge in UK, and subsequently a C. Oh my goodness, I'm going to have to make better notes next time. Dr. Skoulidis' laboratory is focused on unraveling the molecular and clinical heterogeneity of KRAS mutant non-small cell lung cancer. And heterogeneity just means differences, y'all. Don't get freaked out about it. And developing novel therapeutic strategies to target our common enemy, KRAS. So with a great privilege and such a pleasure to welcome you this morning. Thank you for joining us.
Morning, and thank you for having me. Thank you, Terri, and thank you, KRAS Kickers. Let me share my screen with you. Hold on. Share screen. All right. Can you all see it? Yes, you can. All right, so it's an absolute pleasure to be presenting to this group, to this very active group. It's doing a fantastic job. The title of my talk is going to be Alive and Kicking, Current and Emerging Strategies to Tackle KRAS Mutant and Non-Small Cell Lung Cancer. I am an associate professor here at MD Anderson. I'm a physician scientist, which means I spend part of my time treating patients with non-small cell lung cancer, many of them with KRAS mutant non-small cell lung cancer, but I also spend most of my time running a lab trying to understand how best we can treat this disease. So these are my disclosures. So as you know, KRAS mutations represent the most prevalent oncogenic driver event in non-squamous non-small cell lung cancer. In this pie chart, KRAS mutant non-small cell lung cancer always represents the biggest part of the pie, the biggest piece of the pie. Approximately 30% of all non-squamous non-small cell lung cancers will harbor a KRAS mutation. And within non-small cell lung cancer, KRAS mutant non-small cell lung cancer, by far the most common KRAS allele is KRAS G12C. It's approximately 41% of the total, followed by G12V and G12D and G12E. So KRAS G12C mutations overall are found in approximately 1 in 7 to 1 in 8 non-squamous non-small cell lung cancer. It's a very big part of the overall patient population. One important point that I would like to make from the beginning, and this will become even more relevant as the talk advances, is that KRAS tumors are very different from each other. They are, as Terri mentioned, heterogeneous. That means that one patient's KRAS mutant tumor behaves very differently than another patient's KRAS mutant tumor. And there are two major types of determinants, two major factors that determine why one patient's KRAS mutant tumor is different from another patient's KRAS mutant tumor. One of them is the different KRAS alleles. And this is, for example, KRAS G12C, KRAS G12V, KRAS G12D. This is particularly relevant nowadays where we have allele-specific inhibitors that will only work for one specific KRAS mutant allele, but not for the other. But also, and perhaps even more importantly in trying to decide what makes KRAS mutant tumors different from one another, is mutations not in KRAS itself, but what we call co-mutations. So these are mutations in genes that happen together with KRAS. Dr. Der just told you that KRAS itself is likely not enough to transform a cell to make it cancerous. It requires additional mutations. So it's these additional mutations that happen that to a large extent determine the heterogeneity of KRAS mutant and non-small cell lung cancer. And several years ago, when I first joined MD Anderson in 2015, that was the key question that I wanted to answer. What makes one patient's KRAS mutant tumor different than another? So we found that there are three major co-mutations, three major genes that can classify KRAS mutant tumors into three groups that we now call KP. These are tumors that have got mutations in KRAS, but also in a second gene, P53. KL, these are tumors that have got mutations in KRAS and in STK11 or LKB1, so a different tumor suppressor gene, and KC. These are tumors that have got mutations in KRAS and inactivation of the CDKN2A and CDKN2B tumor suppressor gene. And this is important because approximately, especially mutations in STK11, as well as a second gene that happens frequently with STK11 called KEAP1. This is important because approximately 1 in 3 KRAS mutant tumors will have a mutation in either STK11 or KEAP1. So how does this affect the biology of the disease and how does it affect response to treatment? What I'll say from the beginning is that it has a profound impact on the response of KRAS mutant non-small cell lung cancer to various standard of care therapies. So today, before the discovery of KRAS inhibitors, how would we treat a patient with a KRAS mutation with metastatic lung cancer? We would use immunotherapy, the combination of immunotherapy with chemotherapy, chemoimmunotherapy, or platinum doublet chemotherapy. And if they progressed on that, we would use second-line chemotherapy with docetaxel. Where here we are showing that clinical outcomes of KRAS mutant tumors, non-small cell lung cancer, with immunotherapy depend to a large extent on the commutational status. So tumors that have got the KL tumors, the ones that have got mutations in STK11, have got worse clinical outcomes with immunotherapy, whereas immunotherapy works very well for patients that have got a mutation in KRAS and a concurrent mutation in P53. So this is an important consideration as we assess the impact of different therapies, both standard of care therapies, but also the new KRAS inhibitors on clinical outcomes. So with that, I'm going to move to the KRAS inhibitors. And of course, this represents, and KRAS was considered undruggable for almost 40 years due to a variety of reasons, including the shape of the protein, as well as its affinity for GTP and GDP. This all changed. The watershed moment was in 2013, when Dr. Shokat's group identified compounds that were able to bind to covalently, so irreversibly, to the cysteine at position 12 in KRAS G12C, and occupy an induced pocket, a pocket that was previously unappreciated, a groove that was previously unappreciated. And by doing that, it locked KRAS into an inactive conformation, the GDP bound form of KRAS, that then shuts KRAS signaling down and triggers proliferative arrest, as well as the tumor cell death, apoptosis. And since then, we know that multiple rounds of medicinal chemistry have followed, and we now have clinical grade inhibitors such as sotorasib, adagrasib, and various others that we're going to talk about. So with that in mind, these are the pivotal studies for the two drugs, for the two frontrunners. Sotorasib, of course, a drug that I had the privilege to participate in its clinical development. This drug got FDA approval for the treatment of patients with advanced KRAS G12C mutated non-small cell lung cancer that have failed at least one prior line of systemic therapy, with an objective response rate of about 37%, median overall survival 12.5 months, median progression-free survival of 6.8 months. More recently, we saw data for adagrasib, another mutant selective inhibitor that shows very similar efficacy to sotorasib. These were data published in the New England Journal of Medicine by Passiyani and co-authors in July of this year. And on the basis of these results, adagrasib was granted FDA breakthrough therapy designation in June 2021. More recently, in just a couple of days ago, we also had another major milestone in the treatment of KRAS G12C mutant non-small cell lung cancer, because we had the results of the first randomized phase 3 clinical trial of sotorasib versus docetaxel. So this is not a single arm study. This is a randomized study where 345 patients are split 1-to-1 to either receive sotorasib at the recommended phase 2 dose of 960 milligrams once a day, or docetaxel 75 milligrams per meter square, IV once every 3 weeks. And the conclusion from the study, this was a positive study. Sotorasib resulted in a 34% reduction in the risk of disease progression, the tumor growing, or death compared with docetaxel. So this was a positive study, and this is the first positive randomized phase 3 clinical trial for any KRAS G12C inhibitor. This is also a big moment. Responses to sotorasib in CodeBreak 200, the phase 3 trial, were 28% compared to 13.2% with docetaxel. So it more than doubled the likelihood of achieving tumor shrinkage. And responses to sotorasib occurred more rapidly. The median time to response was 1.5 months, 1.4 months, and were more durable. They lasted longer, and they were also deeper. In other words, the tumor shrinkage was on average higher with sotorasib than it was with docetaxel. Let's look a little bit about the safety of these drugs, which I know is something that quite rightly concerns patients significantly. So both sotorasib and adagrasib are by and large safe drugs. Sotorasib, this is the safety of sotorasib. The majority of treatment-related adverse events were low grade, and they were managed with standard supportive measures or dose modification, interruption, or dose reduction. Only 7% discontinuation of drug due to treatment-related adverse events. For adagrasib, a slightly higher rate of GI adverse effects, but again, the rate of discontinuation of the drug approximately 7%. Another important thing that is the intracranial activity or the potential intracranial activity of these drugs. And these are retrospective data from CodeBreak 100. These were patients that had previously treated brain metastases, and we were looking at whether sotorasib was able to maintain those responses or induce radiological responses. But these were patients previously treated. And in this retrospective analysis, approximately 13% with evaluable brain metastases had evidence of an intracranial response. More recently, we saw data for adagrasib, both in previously treated brain metastases, where approximately 1 in 3 had an intracranial objective response, but also more importantly, and this is the lower part of the curve, in patients whose brain metastases were not previously treated. So in this study, among patients with previously untreated brain metastases, adagrasib yielded 32% intracranial response with a median intracranial progression-free survival of 4.2 months, which is certainly encouraging. I'm going to spend some time talking a little bit about other KRAS inhibitors, which are entering or advancing in clinical development. So this is JDQ443, developed by Novartis. So we saw results earlier this year at the American Association of Cancer Research meeting in spring from the Contrast-01 clinical trial of JDQ443 monotherapy. And again, some encouraging news there. The safety profile of the drug appears pretty good, and the efficacy of the drug also looks highly encouraging, with an objective response rate of 45%. This includes both confirmed and unconfirmed responses, and perhaps an even higher objective response rate among patients with KRAS G12C mutated non-small cell lung cancer treated at the recommended phase 2 dose of 200 milligrams twice a day. Obviously, we need to see more data and more advanced data, but this is certainly promising. And we also saw at this year's World Conference on Lung Cancer in the summer in Vienna, data from GDC 6036. This is from Genentech Roche. And again, the conclusion is similar, that this is the safety profile of the drug certainly is compatible with what has been reported for the other KRAS G12C inhibitors. And again, the efficacy is very promising, with an unconfirmed objective response rate of 53%, confirmed objective response rate of 46%, and what appears to be durable responses, although of course the follow-up is relatively limited. And also another KRAS G12C inhibitor that was, this was presented again at the World Conference on Lung Cancer by Dr. Shun Lu. So this is the safety and efficacy of D1553, a phase 1 clinical trial. Again, the safety data is reasonable, perhaps a little more ALT, AST elevation, but the objective response rate 37%, disease control rate 90.3%, median duration of response 5.4 months. We need more data. This is a relatively immature cohort, so we'll see whether this evolves as time passes, but this certainly is in keeping with the clinical efficacy of other inhibitors. One important point I mentioned at the beginning, that co-occurring alterations, co-mutations, impact clinical responses to standard of care therapies. So this is important because they may also impact clinical responses to G12C inhibitors. So this is early data from both the CodeBreak 100 study as well as the Crystal-1 study for adagrasib, where you can see that perhaps the likelihood of tumor shrinkage with G12C inhibitors to some extent depends on commutations. You can see, for example, that there's a lower objective response rate in patients with KEAP1 commutations, which is one of the key commutations that occurs with KRAS, perhaps suggesting that we need different approaches or more enhanced approaches, combination approaches in this genetic subset. So with that in mind, I am moving to the next chapter, which is we now know that KRAS inhibitors, which were not previously possible, are a reality. KRAS is druggable, and the objective response rate is in the region of perhaps 1 in 3 patients, 30 to 40% across different clinical trials. This is very encouraging. This is great, but of course it's lower than other targeted therapies. So we are kind of thinking that combinations of G12C inhibitors with other drugs might represent the optimal therapeutic approach for the future to enhance both the likelihood of achieving a response, but also the durability of response. And you can see here in this great review from Cancer Discovery, both upstream components as well as downstream components of the KRAS signaling pathway, and suggesting different potential points of rational intervention with additional drugs in order to increase the efficacy of mutant selective inhibitors. And so we can either target upstream components of the pathway, such as the SHP2 phosphatase or the SOS1 guanine nucleotide exchange factor. These factors affect the balance between GDP-bound KRAS and GTP-bound KRAS, therefore potentially affecting the amount of drug, the amount of KRAS that is available to bind to mutant selective inhibitors such as sotorasib or adagrasib, but also downstream components of the pathway, both in the MAP kinase pathway, for example, RAF, MEK, or ERK, as well as in the PI3K AKT pathway. And on the basis of this conceptual framework, all the different KRAS G12C inhibitors are currently being combined with different agents targeting different nodes in this pathway, such as EGFR inhibitors, SHP2 inhibitors, SOS1 inhibitors, as well as more conventional treatments such as chemotherapy. I'm going to present very briefly some data on most recent data with some of the most promising combinations that this is the first data that is emerging. So this is data presented at World Lung for sotorasib and the SHP2 inhibitor RMC4630. So the combination, and this is from Codebreak 101 and subprotocol C. The early readout from this trial is that the combination is tolerable. There are some side effects that one needs to be aware of. About approximately 30% of patients have got edema, fluid retention, but by and large, the combination is tolerable. And in patients that had not previously been treated with a KRAS G12C inhibitor, KRAS G12C inhibitor naive, the early efficacy signal is positive. The objective response rate was 50% among KRAS G12C inhibitor naive patients. And in the two highest doses of the SHP2 inhibitor that were studied, 3 out of 4 patients achieved an objective response. So this is, of course, early data, limited number of patients, but nonetheless positive data. And a study is underway to further define the efficacy and safety of this combination in patients with metastatic KRAS G12C mutated non-small cell lung cancer that were treatment naive. Other combinations had been tried before. So this is the combination with sotorasib plus afatinib, the pan-HER inhibitor. So this what was completed previously. This was an active combination, but it also led to significant overlapping toxicity, mainly GI toxicity, potentially limiting the efficacy of the combination. So this is something that we always need to take into consideration when we try to combine KRAS inhibitors with other drugs. What are the overlapping toxicities, and what is the optimal window what's the optimal therapeutic window? And a similar conclusion from when sotorasib was combined with trametinib. It was doable, but the objective response rate of this combination was a little bit lower, 20%. One important point, KRAS G12C inhibitors are being combined, and this is one of the most exciting and one interesting areas for future work, with immune checkpoint inhibitors. Why is this important? This is important for a number of reasons, because at least in mouse models, the efficacy, the ability of a KRAS G12C inhibitor to induce a long-lasting remission, in other words, not just initial shrinkage, but really a cure, if you want, in the mouse, is only achievable in mice that have got an intact immune response. If the same experiment is performed in mice that are immune deficient, that do not have an intact immune response, you see an initial shrinkage of the tumor with many of the G12C inhibitors, but then eventually the tumor outgrows. However, when the same experiment is performed in mice with an intact immune system, a fraction of the mice can get cured. So this suggests that our own immune system, the anti-tumor immune response, the adaptive branch, both the innate and the adaptive branch of our immune system, contributes significantly to the efficacy of KRAS G12C inhibitors. So with this in mind, and we also know that when we treat mouse tumors with a G12C inhibitor, we inflame the microenvironment. Suddenly we see an influx of effector cells, of immune cells, as you can see here in C. which is being stained for CD8 T lymphocytes. And you can see that in response to AMG 510, we inflame the microenvironment. So these immune cells are going in and are playing a role in the clearance of the tumor. So with that in mind, it makes sense to try to combine immune checkpoint inhibitors that with KRAS G12C inhibitors. And you can see in B that by doing so, you're achieving synergistic efficacy and long-lasting cures, long-lasting responses in these mice, which are not achieved when you treat with each individual agent alone, sotorasib or anti-PD-1 alone. But where you combine them, you can see that these tumors do not grow. So with that in mind, several trials for different KRAS G12C inhibitors have assessed the efficacy and safety of combinations of G12C inhibitors with inhibitors of both PD-1 and PD-L1. From previous experience with other targeted therapies, whenever a targeted therapy, especially drugs targeting the MAP kinase pathway, are combined with immune checkpoint inhibitors, one of the concerns is the safety. So as a way to overcome safety concerns and treatment-related adverse events, one strategy that has been employed in the past is to try not only different doses and different types of immune checkpoint inhibitors, but also different schedules of administration. In other words, concurrent administration, where you're giving the G12C inhibitor, the targeted therapy, together with the immunotherapy, or sequential or a leading kind of schedule, where you're starting with a G12C inhibitor and then you're incorporating the immune checkpoint inhibitor further down the line. So both of these approaches were tried in the multi-arm CodeBreak 101 clinical study, as well as arms from the CodeBreak 100 clinical study. The conclusion from these studies is, as you can see in C, the objective response rate across all different schedules of administration and dose levels was 29%. Encouragingly, the median duration of response was almost 18 months. So that's higher than what is observed with sotorasib on its own. However, on the other hand, the drawback was that for several of these combinations, especially the concurrent schedules, when immune checkpoint inhibitor is given at the same time with a G12C inhibitor, we saw significant toxicity and in many cases prohibitive toxicity. And the main type of toxicity that is observed is hepatotoxicity, elevations in liver tests, which in many cases were grade 3 in this clinical trial. However, in the sequential cohorts and at lower doses of sotorasib, the efficacy was improved. So right now there is a cohort that's assessing sequential administration, so lead-in administration of sotorasib at a dose of 240 milligrams with pembrolizumab, and with the pembrolizumab starting 21 days after the start of sotorasib. That seemed to be a safer combination or schedule in this arm, and we'll know more. So this is going to be explored further. What about adagrasib in the immunotherapy? So this has been tested in the first-line treatment setting in the Crystal-7 clinical trial. And at least from the preliminary data that we have seen, the safety appears reasonable. It appears to be a tolerable combination, and preliminary anti-tumor activity also appears encouraging. So here, across all patients, the objective response rate was above 50%, was in the region of 55%, 11 out of 20 patients in this cohort. Obviously, the median follow-up, the time that we were followed patients in this trial was limited, so we need to see more long-term data, but this is certainly an area that, this is certainly something that will be looked at quite carefully. What other kind of approaches are being followed for incorporation of G12C inhibitors into the first-line treatment space? Well, there's two different approaches. So one is this standard approach with adagrasib on its own or in combination with pembrolizumab. However, the alternative approach is to use sotorasib not in combination with immune checkpoint inhibitor, but with platinum doublet chemotherapy, but limit the patient population to patients that are less likely to derive benefit from immunotherapy, that may require immunotherapy less. So these are patients in this study, for example, with lack of PD-L1 expression, PD-L1 0. And Amgen have announced that they are planning a phase 3 randomized control trial studying precisely this. So the combination of sotorasib with platinum doublet chemotherapy versus the standard of care approach, chemoimmunotherapy, in previously untreated patients with a KRAS G12C mutated non-small cell lung cancer and PD-L1 less than 1%. The other approach is to also target these commutational subgroups, for example, the subgroup with STK11 mutations that we know derives less benefit from the addition of immunotherapy to chemotherapy. And there are 2 clinical trials assessing this in the first-line space, the CodeBreak 201 clinical study, and the cohort within Crystal-1 is also assessing the efficacy of adagrasib monotherapy in patients with STK11 mutant non-small cell lung cancer. What about moving KRAS inhibitors to earlier disease stage? This is an area that I'm very interested in. So there are currently 3 clinical trials and perhaps more in development that are assessing different combinations of KRAS inhibitors in surgically resectable disease. The first one is a study that I'm leading here at MD Anderson. So this is a phase 2 study of neoadjuvant sotorasib in combination with platinum doublet chemotherapy, either cisplatin or carboplatin and pemetrexed. This is for patients with stage 2A to 3B non-squamous non-small cell lung cancer with a KRAS G12C mutation. And in this study, patients are going to be treated with sotorasib and 4 cycles of chemotherapy. After that, they are going to go to surgery. And we're going to test 2 different dose levels of sotorasib, but we're going to start with a full dose. And the second study assesses just sotorasib, not sotorasib with chemo, but sotorasib alone. This is being led by Dr. Borkhei at Fox Chase Cancer Center. This is in patients with stage 1B to 3A single station N2 disease. And here sotorasib is being given 960 milligrams once a day for 28 days prior to surgery. And the third study is being led by Dr. Marrone, who I know is speaking later. And this is the NeoCan study. This is a phase 2 trial of neoadjuvant adagrasib with or without nivolumab in patients with resectable stage 1B to 3A KRAS G12C mutated non-small cell lung cancer. In cohort A, patients get adagrasib at the full dose, 600 milligrams twice a day, just as single agent. And in cohort B, they get the lower dose of adagrasib, 400 milligrams twice a day, in combination with nivolumab for 6 weeks. So 3 doses of nivolumab given every 2 weeks, and then they proceed to surgery. So I'm going to spend the last few slides talking about emerging novel approaches. So I've told you about what is actively going on right now, but we're going to spend some time talking about novel approaches. And there are many, so I'm not going to do justice to all of them, but I'm going to highlight 3 different approaches that I think are interesting. So both sotorasib, adagrasib, as well as the other G12C inhibitors that I mentioned up to now are off-state inhibitors. So in other words, they bind to the inactive form of KRAS G12C, which is KRAS G12C bound to GDP. However, there are conceptual advantages to targeting the on state of KRAS. We may be able to overcome certain mechanisms of adaptive resistance that are observed with off-state inhibitors if we were able to target the on state of KRAS. In other words, KRAS bound to GTP. So there are now several approaches to develop RAS on inhibitors, so inhibitors of the active state. And one that is advanced, that's kind of entering clinical development, is the tri-complex inhibitors. This is a strategy by Revolution Medicine. So this is based on novel chemistry. So these are inhibitors that bind to an abundant protein within the cell. It's a nucleophile, it's an immunophilin called cyclophilin A. And then the binary complex between the inhibitor and cyclophilin then binds to GTP bound RAS and creates a tri-complex, a ternary complex, if you want, that impairs the downstream signaling. So kind of shuts off signaling downstream of KRAS. And the advantage of this approach, and if you want the appeal of this approach, is that you can develop tri-complex inhibitors to different KRAS mutant alleles, not just KRAS G12C or KRAS G12D, but a variety of KRAS different alleles. And you can see that there are inhibitors in development for G12D, G12V, G13D, Q61, G12R, and G13C, and other. But also, there are inhibitors that are called RAS multi-inhibitors. So these are drugs that are able to simultaneously target multiple forms of mutant RAS and not only KRAS, but also NRAS, other RAS isoforms, as well as wild type RAS, potentially affecting and reducing the ability, reducing adaptive responses that promote resistance to mutant selective inhibitors. And the phase 1/1b multi-center study of RMC-6236, this is the RAS multi inhibitor in patients with advanced small tumors harboring specific mutations of KRAS, is active and enrolling. One interesting approach is also the development of tools to not inhibit the KRAS, but degrade it and actually break it down, extinguish KRAS. And there are several approaches in development. Some of them are called PROTACs, proteolysis targeting chimeras. So these are drugs that are bifunctional drugs. So they bind on one hand to, if you want, a warhead such as adagrasib that is able to go and bind to KRAS G12C, but on the other hand, they bind to a protein within the cell, such as the VHL E3 ubiquitin ligase, and these are proteins that are able to target other proteins for degradation using the internal natural degrading protein degradation system that exists within the cells, and it's mediated by the 26S proteasome. So this is a very promising and emerging strategy. We will see, no doubt, we'll see much more of this in the future. And then finally, I'm going to do justice to data presented by my colleague, Dr. Negrao, yesterday in New York. So adoptive T-cell therapy approaches, and there are several different approaches, but I'm going to highlight the TCR T-cell therapy approach here because for the first time, the first patient that was treated with this mutation had an objective response. So this is a highly promising approach, a completely different approach using T-cell therapy to target KRAS mutant tumors. And no doubt we'll hear much more about this in the future. So I want to be mindful of your time. I'm going to mention the key conclusions and next steps. So what are the next steps? So we need to define optimal and tailored to the individual therapeutic combinations to enhance the efficacy of KRAS G12C inhibitors. We need to identify biomarkers of response to both monotherapy and distinct combinations of G12C inhibitors with other targeted therapies. We need to develop strategies to maximize the immune sensitizing potential of KRAS G12C inhibitors. What are the optimal immunotherapeutic approaches? We know that immune checkpoint inhibitors such as pembrolizumab is one approach, but can we target other components of the tumor immune microenvironment and synergize with KRAS G12C inhibitors? We need to see data on the first-line metastatic studies in combination with chemotherapy and/or immunotherapy, and we need to see data for KRAS inhibitor combinations in earlier disease settings. We need to understand mechanisms of de novo and acquired resistance, and of course, we now have novel mutant selective inhibitors, including inhibitors of the active KRAS state, as well as RAS multi-inhibitors, drugs that are able to target multiple RAS isoforms. And finally, we need to see new approaches to tackle KRAS using degraders to extinguish the protein rather than just inhibit it, as well as novel immunotherapy approaches, including both adoptive cell therapy as well as vaccines. I didn't elaborate on vaccines because I know that Dr. Marrone will be talking about this in the future. So with that, I'm going to thank you and happy to take any questions. Sorry, I've taken a little bit more time.
Well, thank you so much for that amazing fire hose of information. I don't. I have probably about 1,000 questions that I could ask you, but I also need to stay on track on time. And so I'm gonna just ask. Well, first off, I'm gonna mention to everybody that's watching that Dr. Negrao's information that was presented yesterday, we're going to have him come back to have a conversation with us in November. We've already got that scheduled. And if I could just get you to answer one question that's kind of back a little ways. You mentioned that you do studies on immunocompromised mice and I wrote it, hang on a second, I wrote it down. Deficient and also competent mice. Do you often see a difference in the way they respond to different treatments?
Yes, absolutely. This is a very important question, and this is the whole base, the whole conceptual basis for for combining KRAS inhibitors with immune modulatory drugs. So in immune deficient mice, we see in response to a G12C inhibitor, you will see initial shrinkage in many models, but that shrinkage does not last. So eventually the tumor finds a way to overcome the KRAS inhibitor and grows out. We only see long-lasting responses and potentially cures, what you would consider a cure in the mouse, in the context of an intact immune system. This suggests that our immune systems get turned on in response to a G12C inhibitor, and that the immune system becomes to a certain point responsible or contributing to the clearance of these residual KRAS mutant cells that may not be, that may not die immediately in response to the KRAS G12C inhibitor. So the efficacy of the G12C inhibitors is always higher in the context of an intact immune system. And this is why we want to combine G12C inhibitors or other KRAS inhibitors with drugs that either relieve immune checkpoints, such as the immune checkpoint inhibitors, or other drugs that modulate the immune microenvironment, for example, drugs that target immunosuppressive myeloid cells in the tumor microenvironment, to further unleash the power of our own immune system against the residual KRAS G12C mutant cells that have not died as a result of the KRAS inhibitor on its own.
Okay, that leads me to the next question. Does it have an implication? I said one question, I lied. Leads me to the next question as, does it have anything to do with the co-mutations that we have? You mentioned ATM, STK11, KEAP1, those sorts of things. Are you seeing big differences and big swings, or is that something that's being concerned and being evaluated?
This is a fantastic question, Terri, and this is something that I'm working on in the lab quite intensely. It's actually one of the main streams in the lab. So yes, in answer to your question, the commutational status of the tumor impacts very much both the baseline immune profile, what it looks like before the G12C inhibitor, but also the immune modulatory effects of the KRAS G12C inhibitor. For example, we know that in STK11 mutant tumors, these are rich in myeloid suppressor cells. So these got few lymphocytes, which are the good cells that fight the tumor, and we've got a lot of myeloid cells that have got a suppressive effect. And it's actually KRAS to a large extent, KRAS signaling, that promotes this accumulation of suppressive cells. KRAS itself can be immunosuppressive. So when we treat these KRAS LKB1 or KRAS STK11 mutant tumors with a G12C inhibitor, we see a dramatic reduction in the number of suppressive myeloid cells, which no doubt contributes to the efficacy of these treatments. But this is a very important point, because different combinations of immunotherapies or immune modulatory drugs might be more applicable to different commutational subgroups of KRAS G12C. So this is something that is no doubt will be important.
Okay, I think that's an excellent answer. And it seems like it just really pushes us as patients to have to learn how to advocate to make sure that we know not just like the one marker that we have, not just that we're KRAS G12C or D or whatever, that we have co-mutations and what those might be, so that we can best identify things that will work best for us. So, okay, and I could talk to you all day, but we're gonna have you come back another time.
Yeah,
and
happy
community.
to.
So yeah, so that was my way of kind of like volunteering you that you'll be back. So we appreciate you doing
I would love to.
Okay, but at the same time, we're not going to let you slide away. Okay, we want to, we're engaging all our speakers to make sure that, you kind of know our war cry, and that you do get a sense of who we are and what we do. And so we're kind of doing a count off with just the group that's here in the room, which is a little bit more limited as a fact of the hurricane. But we'd like to do another Kick Cancers KRAS live with you, engaging in along with the folks that are here. So if you're willing, I'm going to do like a count off, 1, 2, 3, and then we'll do a Kick Cancers KRAS all together. So if okay, everybody in 1, 2, 3, Kick Cancers KRAS. Thank you so much. We appreciate you.
Thank you.
Okay, with that, well, actually highlighted in Dr. Skoulidis' slides, you'll see that we have next up Dr. Kristen Marrone. who, I mean, with I couldn't even do a better introduction. She's already doing a little bit of research around the very things that we're talking about. And she specifically asked that we be a little bit more engaging. And so we're going to be able to kind of like just pelt her with questions, and we're going to keep you busy on that end of the screen. But as a brief introduction, and I'm going to just hit the highlights. Dr. Kristen Marrone is the Assistant Professor of Oncology in the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins and Associate Program Director at the Hematology Medical Oncology Fellowship Program. She is the current clinical investigator with a research focus on lung cancer immunotherapy clinical trials. And she is the PI, principal investigator, for a lot of these other ones. And so I'm going to stay up on stage, and then we're going to be chasing y'all around because we want to hear questions. So I'd love with that to welcome Dr. Kristen Marrone. Thank you. Take it away.
Thank you so much, Terri, and thank you all for having me. Hopefully you can hear me okay. I'm excited to speak with you all today about how I have worked in my junior facalitydom here at Hopkins to build a clinical trial portfolio specific for our patients that have KRAS-driven lung cancer. And I'm going to highlight 2 of our new clinical trials, recognizing that Dr. Skoulidis, who is a tour de force and the absolute international expert in KRAS-driven lung cancer, kind of stole my thunder for one of them. So please ask questions. Like Terri said, this is supposed to be very informal and really designed to kind of talk through my strategies of how I think about designing better trials. So these are my disclosures. Just as by way of introduction, Terri mentioned this. So I completed my fellowship here at Johns Hopkins in 2017. And here we do a research-based training program. So I had sort of formalized training in how to develop clinical trials and think about different ways to approach new questions for patients and how to bring new drugs into the clinic to create new trial treatments. So in clinic, sort of early on, I recognized that patients whose tumors were being driven by the KRAS mutations, we really were seeing great outcomes for some patients with our immune-based therapies, but we wanted to increase that. So we wanted to think about how best to help our patients live longer and better lives with our immune-based therapies that have been approved in lung cancer since I finished fellowship all the way back in 2017. And so developing clinical trials is definitely a team-based sport. So without my mentors and the lab-based scientists that I work with, as well as the pharmaceutical companies, these trials wouldn't be possible. So I recognize Dr. Skoulidis talked about a lot of people today, and I hope you hear us when we say there is like a whole army working hard to develop new options. So the way that I kind of approach clinical trials, or how we think about developing novel clinical trials where I work, is we sort of think about a need, right? Who of our patients really need better, faster treatment options? How best to answer the question? So the specific question that I had was, why are our immune-based therapies not working as well in KRAS, and how can we get them to work even better? We then think about how to develop a treatment approach that will be better than what we already have. So not taking away from what's already FDA approved, but how to make those things better. I think the other thing is, anytime you try something new, scientists are kind of pretty interested in mechanisms. So how do we use our colleagues in the lab to study why or why not these treatments are working? It does pragmatically take a lot of funding to set these studies up. So we think about that up front to make sure that we can actually do the science that we are wanting to do and help the patients effectively. We then need our whole entire research team to operate the study. For those of you that have been on a clinical trial, your clinical trial nurse, they're brilliant. They are subspecialists in their field, and we would not be where we are without them. My study nurse for the vaccine study I'm about to talk about keeps me honest. She knows how much and when we give the vaccine, and we'll talk about that in a minute. And then, of course, we need to treat you all. So we need to see you in clinic, make sure that things are going well, check in often to make sure you're feeling okay on the studies, and then also understand how best to share the results in the context of what we know or don't know. So the trials are constantly ongoing. We're constantly analyzing new data, but we want to get it out there for when, if we see signals that are encouraging, or alternatively, if we see concerning safety signals, we want to make sure we're sharing those in real time with the patients and the teams kind of nationally or internationally that are running the studies. So just as a reminder, everyone, I think you've all been hearing a lot about these different phases, but phases of a clinical trial sort of try to answer different questions, right? So for phase 1 clinical trials, really those are maybe first in human drugs or first time combining medications maybe we've used before. So they really are primarily to evaluate safety and determine how best to give that treatment to each patient. We want to identify and grade the side effects that it causes, so we know exactly how best to counsel patients when we use these drugs in clinic. Phase 2 is saying, hey, I think we've seen some data of this drug before or this combination before. Let's start analyzing how effective it is. How well does it shrink the cancer? How long does it control the cancer for? Phase 3 studies, those are used to confirm the effectiveness. So we say, hey, this looks really encouraging. Let's compare this new treatment to what is already considered standard of care in this setting and make sure that it really works better, because we want things better, right? We want to move the field forward and move and improve treatments. So, and then phase 4s are sometimes done in the context of trying to provide additional information about the risks and benefits of that specific treatment. So in that context, when we think about KRAS-driven lung cancer, I'm sure you've seen this all day long from everybody, but it does help us sort of think about how common KRAS mutations are and how best we need to think about the different KRAS mutations that we see in our lung cancer patients' tumors. When we think about 30% of lung cancers and how many patients we see in clinic, this is definitely something that we are seeing more of. And also when we think about the different mutations and how Dr. Skoulidis just went through how the different codons affect options, that's also really important to know. I also heard towards the end of his talk, I think you all were talking about the co-mutation patterns. So that's just a reminder that we want to make sure that all of our oncologists are doing next generation sequencing testing. So making sure that we are looking at the mutational patterns. Co-mutations such as KEAP1, ATM, STK11, those mutations are in a lot of the commercial panels, but are not in all of them. So it's important for us to make sure we're communicating well if we have or do not have that information from your tumor with you, so that you're aware and can make best treatment decisions for yourself. So why is that important, right? So we know that when we look at the most commonly used regimen in adenocarcinoma, which is, we call it Keynote 189, which is chemotherapy and Keytruda or pembrolizumab put together. What we saw was that certainly for patients whose tumors had higher PD-L1 expression, which is the most common biomarker we use in lung cancer, and then for those whose tumors had a higher tumor mutational burden, so more mutations within their cancer, that seemed to correlate best with outcome. We also saw, for when they looked at specifically KRAS mutated lung cancer tumors, they did see also that those numbers went up. So you can see that in the right there. But then the question really is okay, so if we're saying that those two markers, the higher they are, the better this treatment works, and those numbers are usually higher in KRAS-driven lung cancer, why is not everybody with KRAS-driven lung cancer getting optimal benefit from this treatment option? So we sort of think to ourselves, what else do we need to know to select the right treatment for each patient? So this also was seen, so they did the same analysis saying, hey, what does within the Keynote 042 regimen, which is just Keytruda or pembrolizumab alone, did it all look the same? And the answer was yes, it did look the same. TMB was higher, PD-L1 was higher. So in our minds, this means right now that immune checkpoint inhibition based first-line treatment approaches are the best treatment for our patients with KRAS mutated lung cancer. But we recognize there's a lot we don't know about how to make it better and how we maybe should be able to tell upfront what the individual benefit is for each patient so that we can best counsel you. So we can say, is there something about the tumor mutational burden? Is there something about the co-mutational patterns? We definitely think that those things play a role, but exactly how and for how long, Dr. Skoulidis, who runs his lab, is evaluating this, and several others are nationally and internationally as well. So recognizing that, I run our KRAS clinical trial portfolio here at Hopkins.
Can I ask you a question?
Oh,
Okay,
yes.
I'm sorry. All right. Is it okay if I interrupt you while we're going?
Please. Yes, I'm sorry. Terry, there we go. I lost your video, but now it's back.
Oh, okay. All right. Yeah. Well, I'm not even sure what I can see, what you can't see. Okay. So yeah, I moved off the podium so I can listen to you a little bit better and see in the screen. Okay. So TMB, I keep hearing this term thrown around, but it's not like PDL1 and it's not like a KRAS thing. Could you kind of just explore a little bit what that means?
Absolutely. So TMB is tumor mutational burden. So it counts how many mutations in the tumor per megabase is seen. So the reason that is being evaluated or is often discussed is that we see that a lot of cancers, there are certain cancers where that number is very high, and that when that number is very high, it is associated with improved outcome with immune checkpoint inhibition. What we think that is because though each mutation in a gene presents what's called a neoantigen, which is a new antigen. So something that looks more different and more foreign to the immune system, which sort of increases the chance that your T cells are rolling around and can recognize that those look different and can help attack and eliminate them. So for example, go ahead.
Oh, I was going to say, so how is that exactly different than the PDL1? The PDL1 represents something that's covering the two tumors, so your body can't find it. But
Great
the
question.
TMB,
Yes.
is that a good example, a good explanation? But the TMB is more a matter of, it sounds much more mathematical and not specific.
Yeah, no, that's a great question. So they are a little bit different. So PDL1 is a protein, I think of it like a hair sticking up off the cancer cells. And so it almost works like a inhibitory hat on top of the cancer cell. So it protects the cancer cell from being seen by your immune system. So the higher the percent of so when you get a PDL1 score, so if your oncologist says, oh, the tumor PDL1 score is considered high because it's 90%. What the pathologist did was they actually looked under the microscope, they counted 100 cells and they said, how many of those cells stained positive for the PDL1 protein? And that's where the number comes from. So the more, and to be clear and a little semantic, there's actually two PDL1s that can be reported. So in lung cancer, the PDL1 that's reported is called the TPS, which is the tumor proportion score. There is another PDL1 test called CPS, which is the combined proportion score. That's actually used in different cancers, specifically esophagus and others. So I'm saying that because the numbers can sometimes look a little different. And now that they're both FDA approved, when you look at your surgical pathology report, actually both of those might be on there. So for lung cancer, you want to specifically look for the TPS score, because that is what is used to help determine, at least in our clinic, thinking about if we're using first-line Keytruda by itself or Keytruda with chemotherapy together. So the way that the PDL1 and the TMB are different, just logistically, is to say that when you look at the FDA approvals for immunotherapy in lung cancer, PDL1 is what is used. Okay, TMB is more of an exploratory endpoint because it has sort of been shown in different trials to matter a little bit more, and there's been some statistical differences. So we think about TMB as a potential biomarker of immune checkpoint efficacy, but it's not necessarily tied to a specific regimen the way PDL1 can be.
Okay, and is it different you mentioned specifically in lung, is it different in colorectal, pancreatic, and other cancers as well?
That's a good question. It is not used in the same way. PDL1 is not attached to FDA approvals in colon and pancreas cancer. The relevance to tie back the question about TMB. So we know that pembrolizumab is approved in mismatch repair or deficient GI cancers. So that is tied to the TMB. So that's where the TMB is very high. Now, what the molecular background of that is, when you are deficient in the proteins that can repair changes in genes, you then cause new tumor mutations. So your tumor mutational burden will be very high. So that is something like Lynch syndrome, or that testing is standardly done in GI cancers.
Okay, thanks. I'll let you get back to your slide. Sorry.
Yeah, no, that's great. Thank you. So the way that we are sort of approaching right now our clinical trial portfolio, we're thinking about early stage lung cancer and metastatic lung cancer. So we do know that unfortunately right now, we are diagnosing most of our patients with lung cancer in the metastatic setting. So that is certainly our the thrust or the main priority of our portfolio right now, but we are recognizing we are catching lung cancers earlier. And so thinking about how to improve cure rates for patients diagnosed with non-metastatic disease is also a focus. So sort of breaking out how we are approaching this, I would say to Dr. Skoulidis's point, because the current small molecule inhibitor approval is only in G12C, and there are a lot of G12C small molecule inhibitors out there in trials, we have it broken down into G12C and non-G12C. So we do that just because we recognize numerically how many patients within each group we're going to see. So we want to have the most options for the most patients. So I'm going to talk with you today. I don't know if you're going to, hopefully you can see my cursor. We're going to mostly talk about NeoCan, but then we're going to mainly talk about this vaccine study, which is for the first line metastatic treatment setting. And it can include patients whose tumors have G12C mutations, but it will also include non-G12C mutations. These other studies, Crystal 7 and Codebreak 101, I have one slide about those, but I saw Dr. Skoulidis talked about it. So we'll go through those if there's questions or if there's
Real
time.
quick, I just want to make sure. So this vaccine is not like we think of vaccine for, like, say, smallpox or something. This is a vaccine for somebody who already has an existing cancer.
I'm so sorry. Can you say that
Okay.
again?
Okay, so this vaccine that we're talking about
Yeah.
here, this is not for, like, to prevent cancer. This is a vaccine for
Correct.
somebody who has cancer, right?
Correct. Yes. For our lung cancer, that's correct. Okay. So, so talking about the vaccine study, so this is the very long-winded name, but I did put the clinicaltrials.gov identifier here if you all wanted to sort of look at that. So it is a KRAS peptide vaccine that was created here at Johns Hopkins, combined with nivolumab and ipilimumab, nivolumab being a PD-1 monoclonal antibody, ipilimumab being a CTLA-4 monoclonal antibody. This regimen is FDA approved in metastatic lung cancer based off of a clinical trial called Checkmate 227. So this study is specifically going to be talking about if and how the vaccine can actually improve outcome for patients treated with a treatment that's already been approved. So again, kind of when I thought about this study, I said, you know, why am I trying to do this? What is the approach here? And again, this is the data showing that there really is a variability to the response with immunotherapy in KRAS mutated lung cancer. We think we are working, I mean, we hope we're working really hard to figure out exactly mechanistically why that is. But while we do that, we also want to make sure that we are adjusting and improving our treatment options to best use the information that's coming out in real time, and then also think fluidly about how we could study and identify maybe new or novel ways that these treatments affect the immune response in KRAS-driven disease. So here, again, you can just see that this is the association on the left of KRAS mutations with survival in patients treated with the Keynote 189 regimen. And really, unfortunately, you know, when you think about when they subdivided out the patients whose tumors had KRAS mutations, those patients did not have as much benefit as those without KRAS mutations. And then on the right is actually data showing that in the specific G12C setting where the differences may lie. So, and this is because, and I think Dr. Skoulidis discussed this already, so I won't belabor the point, KRAS mutations have an incredibly complex effect on our immune system. So when you have the mutation, you can see, or, you know, and this is any mutation in the KRAS gene, you can see that in some ways they increase or drive pro-inflammatory signaling. So we think about pro-inflammatory signals as things that will draw in immune cells. So which we have found in lung cancer to be an important metric of the ability of immune-based therapies to work. So we need to get the T cells into the tumor in order for them to have an effective cytotoxic effect and actually kill the cancer cells. We also recognize that KRAS has immune inhibitory effects. So there are ways that it signals and tells immune cells to not come to the tumor, or if they do get there, they actually turn them off. And so looking at that, you can see the different colors here are the different types of immune cells. So we see, things called Tregs, myeloid-derived suppressor cells, macrophages, and different T helper cells. They're all affected by these KRAS mutations. And then again, talking about what Dr. Skoulidis discussed, the co-mutational patterns also have further complex effects on these immune cell populations in the tumor microenvironment. Tumor microenvironment, again, being sort of what cells are in and around the tumor cells that we can hopefully positively affect to increase the chances of the cancer cells dying. So when we think about peptide vaccines, so Dr. Jaffe on the right there is the Deputy Cancer Center Director here at Hopkins. She has a lab in pancreas immunology, and she is the driving force behind the preclinical science that developed the vaccine that we're talking about. So when we think about vaccines in lung cancer, there has been really encouraging early phase studies looking at vaccine improving outcome. In confirmatory trials, though, and this is in the pre-immunotherapy era, the responses and ultimately the survival were not significantly improved enough for those vaccines to be approved by themselves. So this vaccine, this peptide vaccine that Dr. Jaffe and Dr. Zaidi have developed is designed to overcome the mechanisms of immune resistance that we see in KRAS-driven lung cancer. So the reason being is that the way that you see the T cells change in the tumor cells related to the vaccine, you can see that there is an increased KRAS-specific T cell response. And then you can, so there's new T cells, but then you also see an increase or an amplification of pre-existing T cells. So not only are you driving a better T cell response with what you have, we're also creating hopefully a new one. KRAS is an attractive vaccine target. So what does that mean? So that just means that we're saying these KRAS mutations are specific to the tumor itself. So it will drive hopefully a more tumor specific T cell response than something where vaccines are often developed with peptides, which are proteins that could be found on other cell surfaces, which would make the T cell response less specific and would, in theory, potentially increase the risk of side effects. What we found, or what Dr. Jaffe and Dr. Zaidi have found, is that these vaccines require pairing with the immune checkpoint inhibitors, and that is because it helps increase the sustain the T cell response. So make sure that the T cells don't sort of get excited and then go away too quickly. So Dr. Zaidi here on the right is the junior physician scientist working with Dr. Jaffe and has really been the driving force behind setting up these studies in different patient populations in the GI cancer world, which we'll talk about in a minute. So this is Dr. Zaidi developed while she was a fellow using this vaccine in patients who had early stage pancreas cancer. The pancreas cancer was removed, and then using the vaccine with a few doses of immune checkpoint inhibitor afterwards to decrease the chance of relapse or recurrence. So what has been shown, or what she has seen in the first 3 patients that were treated with this regimen, is that there was an increase, all of these lines are increased T cell responses to the mutated peptides in the vaccine. So what that means is that you can see as the weeks go by after the vaccine, there was differing changes in these T cell populations. And so understanding and looking at both the patient's specific KRAS codon as well as the other codons within the vaccine is something that we're currently working on, and she's working on in the GI cancer setting. So how do we think this works, right? So the vaccine has anywhere from 4 to 6 synthetic long peptides targeted to the KRAS neoantigens. And the way that this, we obviously match the peptide to what given mutation is expressed in the patient's tumor. The vaccine is given as a subcutaneous injection, and it is given and then immediately followed by nivolumab and ipilimumab. The poly-ICLC is added to the vaccine as a way to support the immune response. So we hope that with all of these drugs given together, there is an increase in tumor death. So logistically, what does this study look like? So, every clinical trial is very different. And so there are sort of different ways we talk about the components of creating and maintaining a study schema. So for this study, the key eligibility criteria, so what patients does this study make sense for? It's patients who have not yet been treated for metastatic or advanced lung cancer. Specific KRAS mutations that are included in the vaccine are KRAS G12C, G12V, G12D, G13D, or G12R. It should be noted that unfortunately our patients with stage 3 lung cancer who get chemo and radiation followed by durvalumab, if they were to have recurrence where their cancer was to come back, as long as it's been at least 6 months since they started that treatment, this study would make sense for them. The reason we define that specifically is to say, make sure that the cancer cells appear to have at least preliminarily been responsive to immune checkpoint blockade, because otherwise you might be trying to assess using chemotherapy with your immunotherapy at time of progression or recurrence. Unfortunately, brain metastases are common in lung cancer, and so we recognize that we don't want that to be something that precludes people from participating in the study if they would like to. So if those brain metastases are asymptomatic or have been treated, that those patients are safe to go on this study. So we are hopeful to treat 15 patients with this regimen. So there's different phases, which makes it sound really complicated, but it's just based off of how many doses of the vaccine you get at each point. And so what basically happens is it's called the prime phase. So that is to say we need to give the vaccine frequently. So it's weekly. And then spaced out to once every 6 weeks with the nivolumab and ipilimumab. And then after a year, you continue the immunotherapy for 2 years, which is the standard of care with the Checkmate 227 regimen, which this is based off of. So the primary endpoint is safety and feasibility. So to say, is it safe to give this regimen and is it feasible? Are there any delays in giving the treatment because of something maybe intrinsic to the vaccine or the schedule? Our secondary endpoints are median progression-free survival, which is how long patients are on the study until they are having to stop the study treatment, either due to progression of disease or some other reason. We're also measuring the KRAS-specific T cell response with a test called ELISpot, which measures the amount and quality of the T cells that are specific to the KRAS mutation that the patient's tumor cells have. Our exploratory endpoints include T cell gene expression dynamics, T cell quality and clonality. We are measuring circulating tumor DNA. The reason we're doing that is to see if that could be a surrogate marker for response. We have a physician scientist at our institution named Elsa Anagnostou who has shown that this can actually predict response and resistance about 6 to 8 weeks ahead of imaging for our patients treated with immune checkpoint inhibitors. Is there any questions about this? Because this is kind of overwhelming to look at.
Yeah, I can jump up and have a question first off, and then I'll pull the room here. Okay, I can see you've got G12C, G12V, G12D, G13D, and G12R. Why were those chosen?
That's a good question. So those are actually chosen because they are the most frequently mutated codons in KRAS for lung cancer, pancreas cancer, and colon cancer, which is what this vaccine was designed to treat. We had G12A included as well. That codon, that lot of the peptide vaccine is not currently available, but it may be added back in the future.
Okay, and then just kind of a general question, because how important is it for a patient to report the ongoing adverse events that they've got during a clinical trial?
That's really important. Thanks for bringing that up. So the reason is multifold. Number one, I would say, especially with immune checkpoint inhibitors, we have seen clear data even for survival, that the sooner we identify and treat toxicity, the better patients do. And we recognize that the severity of the symptoms can be significantly diminished quickly if we intervene right away. So pragmatically, what I mean by that is, if you were to develop diarrhea concerning for colitis, we recognize in the early studies of nivolumab and ipilimumab and melanoma, if patients were delayed, so they were having the diarrhea for weeks to months and then treated, it was harder to make the colitis go away completely. And so people had consistent diarrhea, or patients actually were passing away due to how severe the diarrhea was. When the recognition of how immune-based toxicity was working with these checkpoint inhibitors was truly sort of better discussed and shown at national and international conferences, the rates of that dropped significantly. So especially on clinical trials, it's best to know, because also what it means is we're more likely to be able to resolve the side effect and potentially, if it's safe to do so, give you the treatment again moving forward. So it's good to know. And I think also from the perspective of the study, it's good for us to know that this treatment is safe, right, for everybody. That's the whole, we want to make sure that what we're doing is definitely causing much more benefit than harm. And so it's good for us to know that in real time.
So it doesn't mean just because we complain of a stomach ache that we're going to get booted off the clinical trial. We might actually be able to take a treatment for it, or
Absolutely, yes.
I mean, like, some sort of side effect management, whatever that looks like, right?
Yes, absolutely. Yes. And again, I think, identifying it sooner means we can try best supportive medicines, like you were just saying. So, I completely unrelated to KRAS, but I work on a different study that uses immune-based therapies. We see a lot more reflux for whatever reason. And so treating with reflux medicines is absolutely something to try first before we even start talking about other options.
Any questions in the room, holler. Okay. Yeah.
I just had a question for the study, the requirements. You said untreated, advanced, unresectable cancer. Did you say that you can't have Mets to be eligible?
Oh, it's absolutely fine if there is metastatic disease for this study.
I didn't hear that. Okay, thank you.
Sorry about that. Yep.
Does anybody else have a question? Because otherwise I've got, I'll keep you here all day. You have faith. Oh,
Thank you so much. Great presentation and great institution. I just wanted to, I understand just a little bit, just enough to be dangerous, about there are sometimes some correlation with the KRAS and the BRAF. And I wondered, do you do anything with the BRAF, or is there anyone at Johns Hopkins who is your counterpart with the BRAF, since they are somewhat aligned, I guess?
That's a great question. So this study does not include or exclude co-mutation. So for example, if someone's tumor had BRAF V600E and KRAS G12C, this study would be fine to be on. There are BRAF specific clinical trials at Johns Hopkins. I run one of them, so it's right now only for V600E. There are nationally definitely studies for non-V600E BRAF mutations. I am not aware of any studies that directly combine precision oncology approaches for co-mutations.
okay. All right. And I have one question. You were referring to on one of the slides, pro-inflammatory signaling. Now, when I hear inflammatory, I immediately think of things like arthritis and those sorts of things. Can you kind of like explain that a little bit?
Yeah, absolutely. So inflammatory is a very non-specific word. It's basically an immune response. So, how does your immune system activate and respond to something? So you're right, arthritis, and let me clarify arthritis. So arthritis, when I think of arthritis, in immune checkpoint world, that's like an inflammatory arthritis. So it's actually, you're right, it's very similar. It's increased and angry T cells that are going after inappropriately in your joints. They're going after something in your joints and building up there instead of being activated and fighting the cancer. In this scenario, the signaling patterns are actually being studied to see if they're the same, because scientifically, if they're different, then you could envision a world where you could actually increase the signaling that's specific for the tumor while leaving the arthritis alone. Or if they're the same, then you would try to think about how to minimize the signaling in the joints, for example, and focus just on the tumor.
So it's good. I don't, any other questions until we move on? And I don't want to keep you here all day, so I'll let you.
No, that's okay.
Yeah, I'll let you continue with your presentation. Thank you.
Great. And this lovely guy here in the corner, this is our fellow Sam Rosner, who has been working on this study with me and received a grant actually for his work with the study. All right. So these are the study objectives. So this is, I think I'm just putting these up here to say, when people talk to you about clinical trials, sometimes we're so excited about it, we don't actually stop and kind of go through with you why the study makes sense for you or what actually the purpose of the study is. So I would encourage all of you and feel empowered if you are discussing a clinical trial to talk about what the purpose of the trial is, right? Because again, going back to that clinical trial phase discussion, they sort of have different goals. So this study is brand new. It's pretty cutting edge. We're really looking to make sure that it's safe and feasible to give this. We are not treating enough patients to definitively answer the question of, is this treatment better than just giving the immunotherapy? We're saying, we think it might be. We would like to make sure it's safe to do this, so then we can get more information about how or why that is true. So these are screening requirements, and ma'am, I'm sorry that I didn't put this slide up first. So this is where we say it's metastatic lung cancer or it's unresectable lung cancer. So these are technically stage 3 or 4 lung cancer patients. For imaging, there needs to be a CT scan or PET scan. There needs to be a brain MRI. This is also another important point about clinical trials, which is to say, are there increased risks of being treated on a study? So for this study, the way that we have the study written to answer some of the science mechanism-based questions, there is a research biopsy that is required. So this would be an extra biopsy that you wouldn't necessarily otherwise have if you weren't doing the study. So this is something that you would go through on the consent form and talk through with your doctor to make sure that that risk is acceptable to you. The KRAS testing is, as you all probably have heard about, there's a lot of different ways to test for KRAS. This study is designed to use whatever molecular testing that has already been done. It does not need to be reconfirmed. There's baseline blood work, an EKG, and then a physical exam and history. The other important thing to point out about clinical trials is the logistics. So to say that this study is only open at Johns Hopkins. So unfortunately, what that means is right now we would only be able to treat you with this regimen if you came to Baltimore. There are other studies, and some of the ones that we'll go through if we have time, those are multi-institutional studies. So it's important when if you use clinicaltrials.gov or talk through with your oncologist, what the options are is to think through if that travel is feasible and available to you. And then there was one other thing I was going to say. Oh, there's extra visits. So the way that clinical trials work, again, depending on the phase and what the treatment is, there might be extra visits where you're seeing the doctor more frequently than you would otherwise. And that also can be a strain. So it's good to know that up front so that you can talk through that with both your oncologist and your family.
Okay, let me
So
interrupt you one more
Yeah,
time.
yeah.
Okay, all right. Are there going to be any instances where a co-mutation would be targeted before the KRAS mutation would be?
Good question. So as of right now, the answer to that is no. The long answer to that is I think Dr. Skoulidis is working very hard and is trying to identify if there are treatments that make more sense scientifically if your tumor has co-mutations. So to say, for example, if your tumor has STK11 co-mutations, does it make more scientific sense to start with a different regimen than perhaps a checkpoint inhibitor only regimen, which is what this is. So, or is he going to find ultimately that maybe using something like ipilimumab, which we don't always use in our lung cancer patients, if that is maybe better for patients with co-mutations because it somehow changes the immune response in a more effective way than just the PD-1 inhibitor. So I could see it going either way, but you are correct. I think in the not so distant future, the co-mutation status of the tumor is going to help select treatment regimens.
That also leads to the question, is this only lung cancer patients or pancreatic, colorectal, other patients can engage?
Yes, I'm so sorry. I hit the wrong button trying to respond to that question. So let me just jump ahead.
I'm sorry, I'm messing up your.
No, that's totally fine. So let me see if I can find it. So this vaccine is being used in colon and pancreas cancer. It is also a single institution study. So only at Johns Hopkins, Dr. Zaidi is running it. I put the clinicaltrials.gov information up for you. I'm sorry, my dog is really excited right now. It is for patients with resected colorectal and pancreas cancer and is open and enrolling.
Okay.
Does that answer the question?
Yeah, I think so. I think
Okay,
so, yeah.
okay. Talk about TP53 and RAS is the other question in the chat. I can say, when you think about co-mutation patterns, we've been focusing on ones that we think are linked to less response to immune checkpoint blockade. So STK11, KEAP1, ATM. The TP53 co-mutation with KRAS is actually a positive indicative biomarker for potential response to PD-1 blockade. So that would be considered in my clinic, if I saw a patient whose tumor had KRAS G12C and TP53 and a PDL1 of 90%, I would feel extra confident that giving single agent Keytruda is very likely to induce a great immune response and effective cancer control. As opposed to in my clinic, which is not necessarily, I would say, standard across the country, if I were to see a patient whose tumor had KRAS mutation, an STK11 co-mutation, and a PDL1 of any number, I would feel less confident about using Keytruda by itself because I think the data we have right now from Dr. Skoulidis and others suggests single agent PD-1 is not enough for those tumors.
Yeah, I hate to interrupt one more time, but I have asked way too many questions. And so like now I'm causing like us to go over and I can see that our lunch arrived and I don't want to take up your whole Saturday.
Oh, no.
So if you want to like wrap up and then
Yep.
we can set up a time for you to come in and like just continue to be patient answering these amazing questions that we're getting from these folks.
Yeah, no, that's absolutely greait. So I'll put up the NeoCan and then we'll be done. So just for this study, I would say we're trying to, in the lab, look at these T cell responses and figure out how best to increase them because we think that's going to increase the benefit of the treatments. For our study, we got approval in August and we actually treated our first patient on September 12th. And that person is doing really well. And then ultimately, we want to use the results of this study to look at the immune response and then hopefully, if it works, move the treatment forward. All right, and then NeoCan, I'm going to just say, this is the neoadjuvant study that we have. This is a multi-institutional study. So I'm going to be the one running it, but it's going to be open at about 12 places in the US and Canada. And it looks at early stage disease prior to surgery, giving either adagrasib, which is what this is called, by itself, or adagrasib with nivolumab prior to surgery. And so I can stop there, Terri. That's
okay. Well, I'd like to thank you so much for being so patient and tolerant with our amazing questions that we've been getting today. And thank you for taking your Saturday, but you're not getting out of here without doing what we've asked all the speakers to do along with the rest of the crew here. And just say alongside of us, kick cancer's KRAS. And so I'll like do a full countdown, kick cancer's, like 1, 2, 3, and then we'll all do it together. And then we're gonna go out there and grab us some lunch. So 1, 2, 3, kick cancer's KRAS. All right, thank you so much.
Thank you. Yes, and I have my socks on, so thank you and have a great
All right.
weekend.
All right, we'll keep on working it. We'll walk in the walk.
Yeah.
So we'll see you again. Thank you.
Bye-bye.
Bye. Okay, and I know that there's we're kind of like losing people online, probably because like we're just so interesting. We're having all the fun here, but I'm gonna try and do something a little bit different. And they did not know that I was gonna do this. We put together some of the names, the folks that registered to attend, and these lucky folks, because a lot of them were planning on being here in person, but were unable to attend in person. So they don't get the same swag that you get. Okay, they kind of get like a limited mini version of what you get. So they get to walk the walk, they get a pair of socks, but they don't actually get the other cool stuff. So I'm going to grab this real quick and and then we'll go to lunch. I promise y'all get to eat. So okay, and I don't know, there's like, see, there's lots of names in there and I'm not in here. So okay. Oops. Okay, all right, let me see. Pardon my fancy bowl here. Okay, what should we be giving away? How about a bag? Okay, KRAS Kickers bag. Oh, Lisa Haynes. Lisa, we're gonna be mailing you a bag so that you get a bag of swag, my friend. All right, thank you guys. And we're gonna be throwing some videos up online, and then we'll see you back here at like 1 o'clock. I don't even know. I lost my program. 1 o'clock, I think. So we'll see y'all in a little bit. Bye.
Hello, everyone. I'm Corina Patelli-Bell and president of Fairlife Lung Cancer Care, founded in memory of my beloved husband, Simon Bell, who lost his life to lung cancer in August of 2020. Fairlife works to raise public awareness of lung cancer causes, to spread the word about the importance of preventing care, to approach lung cancer holistically. Our vision is to give every patient a fair chance against lung cancer, equal access to information, innovative therapies, biomarker testing, and clinical trials. Simon was diagnosed with KRAS G12C. However, he did not have the chance to take part in a clinical trial in the USA or in Greece, which could have saved his life. Today, important steps are made to targeting mutation and bring about new treatment possibilities for lung cancer patients. As a patient advocate, Fairlife joins pharma partners to provide information about biomarkers testing. We now feel privileged to run together with Amgen Greece a campaign to build awareness for the importance of biomarkers and KRAS mutation among patients, families, and caregivers. We are on our way to kick cancer's KRAS. Along with so many people, we move forward with determination and hope. Hope shared is hope multiplied. Thank you.
My name's Anne-Marie Baird, and I'm My name's Anne-Marie Baird and I'm president of Lung Cancer Europe, also known as Lucha, which is a non-profit umbrella organization working at the European level to help improve outcomes for people impacted by lung cancer. So we are really looking forward to working with the KRAS Kickers, because we know in Europe there's a lot to be done within this space. And we also know that by working together, we can achieve better outcomes for all of the community, because hope shared is hope multiplied. And by working together, we can really kick cancer's KRAS.
Fernandos' laboratory is based here at the Molecular and Biotechnology Center of University of Turin. I am Marcella and I work here as a postdoc researcher.
And I'm Eva, a PhD student. In this video, we'll show you how we study oncogenic KRAS.
Our work always begins from the hypothesis, where we examine the recent progress in the scientific literature. Also, this is where we spend time to carefully design our experiments.
In this area, we analyze DNA and protein isolated from our KRAS-driven cancer model to explore the signaling pathways that happen at the molecular level.
This is the tissue culture room. Under these laminar flow hoods, we ensure sterility conditions for culturing our human cancer cell lines that are maintained in special incubators.
Our lab is equipped with cutting-edge technology, such as a drug printer for screening of potential new anti-cancer compounds, or an IncuSite system for live cell imaging.
Hello, my name is Miranda Goff, and I'm the Senior Manager of Support Services at GoTo Foundation for Lung Cancer. GoTo is a nonprofit organization dedicated to saving, extending, and improving the lives of those vulnerable, at risk, and diagnosed with lung cancer. We work to change the reality of living with lung cancer by ending stigma, increasing public and private research funding, and improving access to care. We also provide direct support to people with lung cancer and their loved ones through our helpline, our treatment and trial navigation services, and our peer-to-peer support network. Over the last year, KRAS Kickers has partnered with GoTo to make it easier for people to connect with one another through the Phone Buddy program. The Phone Buddy program is a peer-to-peer program in which our dedicated volunteers of patients and caregivers connect with others to offer their support based on their shared experiences. It's through partnerships like this that the KRAS Kickers helps to multiply hope and help to kick cancer's KRAS. Enjoy the conference.
Hey, we are the Rising Tide Foundation for Clinical Cancer Research.
Our grants support clinical trials that aim to reduce toxicity and improve the quality of life. It is close to our heart to hear the patient community. That's why we give a high priority to patient involvement in clinical research.
Reach out to us if you're a patient organization, clinician, or funder planning a patient-directed clinical trial. We also want to kick KRAS. Our team is here to help you. Let's connect.
We are very excited to partner with KRAS Kickers. We love how you are making a difference and wish you all a Kick KRAS event.
Yay!
Hi, my name is Dr. Stephanie Snow. I'm a medical oncologist in Halifax, Nova Scotia, and president of Lung Cancer Canada. We are the only national organization focused exclusively on lung cancer in Canada, serving as the leading resource for lung cancer education, patient support, research, and advocacy. With lung cancer continuing to be Canada's #1 most commonly diagnosed cancer, Lung Cancer Canada is here to support patients, caregivers, and families through the numerous programs and resources we offer. We work to spread awareness that anyone with lungs can get lung cancer and to educate the public to fight the stigma that unfortunately still surrounds lung cancer, because hope shared is hope multiplied. We advocate for access to medical therapies and public funding of lung cancer treatments and are thrilled that KRAS targeted drugs are now being added to that list to change outcomes for patients around the world. With KRAS being one of the most common mutations linked to all types of cancers, Lung Cancer Canada is here to support the KRAS Kickers in the fight against cancer. Let's kick cancer's KRAS.
Hi, my name is Pierella Peralta, and I'm a patient advocate at Inspire to Live. At Inspire to Live, we bring together patients, researchers, and clinicians to be the patient's voice in cancer. And we hope by doing that, we do better, because it is with hope shared that hope is multiplied. And in that way, we're going to kick cancer in the KRAS.
All right. Okay. Yeah, are we ready to play the video? Can we hear it in here too? All right, let's rock and roll. Chris.
Here's what motivates us at Amgen to kick cancer's KRAS.
Helping patients not just with KRAS G12C, but also working for new innovative therapies to help patients with all variants of KRAS.
The patients of tomorrow.
My mom's cancer journey.
Drugging the undruggable is what motivates us.
Because everyone deserves a fighting chance.
Patients and their families.
We work for inspiring advocates just like you.
Serving patients because every day counts for KRAS.
Because making a difference matters.
I'm motivated to kick cancer in the KRAS because I believe everybody deserves a chance to breathe freely and live a long, healthy life.
For my pets, right, Luna?
Amgen is proud to partner with KRAS Kickers as part of our collective and committed efforts to improve the lives of patients with KRAS-positive cancer. On behalf of Amgen, we thank you for the continued partnership and dedication to the cancer community.
Welcome back, everybody. Thank you, Amgen, for your contribution and participation in what it is that you're doing. They are, I can't remember what we're calling it, the top tier, amazing sponsor, and we would not be able to be here today if it wasn't for them. So that was a couple of the faces behind the scenes of the folks are working together to truly kick cancer's KRAS. And so thank you guys for that amazing video. So we got to see a little bit of the people and know that you really are working alongside of us for that. Okay, and once again, I am Terri Conneran, and I am a KRAS Kicker, and I am really pretty jacked up to be here today with y'all. And even though we've had a couple of little hiccups, like a hurricane, we have not really just let that go to the side, because we still have an amazing group of people that have been able to make it here from as far away as like New York and Georgia and North and South Carolina and just across the state and in Pennsylvania and what have you. So I'm happy to hear, and I'm not going to chat too much because you guys know that I do like to talk, but I want to be kicking off for our next upcoming speaker so that we can stay on target. Pardon the pun. And it is called Targeting KRAS from the druggable to the druggable, and colorectal cancer. Colorectal cancer does have a very high predominance of a KRAS mutations. And with us today, we have Dr. Daniel Ahn, and he's a GI medical oncologist and associate professor at the Hematology Medical Oncology at Mayo Clinic. He conducts clinical and translational research focused on developing anti-cancer agents for patients with GI cancers. He is an excellent collaborator, and we are really thrilled to have him joining the KRAS conference today. And welcome to you today, Daniel. Thank you.
Great. Thank you for the kind introduction. I'm sorry I couldn't be there in person, but I have clinic and I have an infant at home. So I'd rather be there. So thank you again for the opportunity to speak. So let me get my slides ready. Sure. So I actually decided to cover it a little more broadly. So actually, we're going to talk about kind of targeting KRAS across kind of GI malignancies, just to be a little bit more broader to, just because colorectal cancer is not pertinent to everybody here. So just to have a little bit more broader discussion. And so, when I think about KRAS, I kind of think about this. I remember when this article came out in 2001, so early during my med school training. And, this was the big thing that came out at that time. This Time article was talking about Gleevec with CML. And, as I continued to develop through my training and eventually through fellowship, it made me think about KRAS. And, as I quickly garnered an interest in GI malignancies, I remember reading many papers about, why isn't KRAS targetable? And, I read all these papers, and I was thinking back and saying, one day we'll eventually get there. And so I think this is just kind of a premonition of kind of where we are today, at least with KRAS. And so, in the past, we used to kind of really think about KRAS as being a prognostic biomarker, meaning we know that it's associated with bad outcomes. We know that this is the driver of many solid tumor malignancies. This is what causes cancers to be resistant to chemotherapy or treatment and causes the cancer to spread or metastasize. And then on the left, we have predictive biomarkers, meaning are there any potential markers that are highly suggestive of response to treatment? I think with KRAS, we're kind of in the middle right now. So we're going from being a bad actor to potentially a good actor. And, as we continue to develop some of these new agents, hopefully we move more from the center to the left. And so a lot of times we hear about these terms, what we call precision medicine. So in the past, we kind of think about precision medicine as doing the testing, but what is the intent of the testing? So in every patient, we know that there are specific biomarkers, and the one of interest today is about KRAS. And are any of these potential genetic alterations of these biomarkers actionable? Meaning, are there any potential drugs that can potentially act on it? And when we think about actionability, I think this term below is what kind of stood out to me. So what is the appropriate therapy that leads to a disproportionate benefit? And so do certain biomarkers provide a better benefit in terms of efficacy, in terms of response for tumor shrinkage, and compared to what we have in terms of standard therapies? So really, that's kind of the crux or the goal for a lot of these targeted therapies. And so in the past, this is the way we used to think about clinical trials. I think this is a really nice depiction of kind of where we were in the past. So in the past, we used to look at drugs specifically for specific tumor types. So, they're talking about on the top, paclitaxel, which is chemotherapy for stomach cancer. We have pemetrexed in lung cancer, and we have dacarbazine in melanoma. And the way that drugs used to get approved in the past would be they would go through a phase 1 trial, so looking at the safety and tolerability. Then they would go to phase 2 to look at the potential efficacy, and then eventually they would go to phase 3 to get approved. And then as clinical trials developed, we kind of fell into this second basket where we would look at specific biomarkers within a specific tumor type. So we would look at HER2 in stomach cancer or potentially breast cancer. We'd look at ALK, which is a biomarker in lung cancer, and then we would look at BRAF and melanoma. And then they would go through the phase 1, phase 2, depending on the type of study, and potentially getting approved or conditional approval then and going to phase 3. Now here we're really at the third part. So what are these agnostic biomarkers that are not disease specific? Meaning, are there potential biomarkers like KRAS that are shared across various tumor types, where if we can show a disproportionate benefit across various solid tumors, we can potentially get an earlier approval. So meaning really in the early phase 1, where we can get a conditional or accelerated approval based off the benefit. And I think that's kind of where we are right now with KRAS, and specifically we see that with KRAS G12C. And hopefully now we have the approval in lung cancer that will eventually get it in colon cancer and other solid tumors. But I think this is really the evolution or development of where we are with clinical trials. And so I think one area of interest that I've been working a lot with is looking at what we call polo-like kinase 1 or PLK1. And so this is a target that's relevant in terms of for cell cycle signaling. So we all kind of remember from our initial days in biology where we go through the cell cycle where initially you have DNA synthesis and then the cell grows and they eventually split and then the cell regrows again. And so PLK1, it plays a vital role in terms of the cell cycling process. And that also includes the cell cycling process in cancer cells. And we know that PLK1 targets a lot of these key points within the cell cycle, specifically between the G2 and M phase, as well as in the DNA synthesis phase. And so what they've shown in the lab is this drug onvansertib is a polo-like kinase inhibitor, and it kind of attacks KRAS from two different approaches. One, it makes the KRAS cells hypersensitive to PLK1 inhibition. And also it shows synergistic activity with chemotherapy. So 1 + 1 being 3 or 4. And so when you combine it with chemotherapy, what we've shown in the lab is that it tends to have a very synergistic effect in making chemotherapy more effective. And so this drug onvansertib is an oral inhibitor of PLK1, and it works against, again, inhibiting various parts of the cell cycle. And what we've shown in the lab is that it's shown to be synergistic with various chemotherapy agents, specifically those that are typically used in GI malignancies, so irinotecan and 5-FU, specifically in KRAS mutant cell lines, as well as KRAS mutant xenograft models. And so based off this activity, we were interested in pursuing this in the clinic. And so this was a phase 1b/2 trial that I led with Dr. Lenz at University of Southern California, where we looked specifically at combining FOLFIRI and bevacizumab, which is a typical first or second line chemotherapy combination that we would use in colon cancer with onvansertib. And so the chemotherapy was given typically how we would typically give chemotherapy in terms of the dosing and scheduling. And we would combine that with this oral agent called onvansertib, and it was specifically for patients that were found to have a KRAS mutation. The reason why we chose to combine it with this chemotherapy was based off what we saw in the lab, where we saw the synergism with chemotherapy, as well as that most patients tend to get FOLFOX in the first line, probably about 70% of patients. So we specifically looked at it in the second line setting. And the intent of our study in the initial part was to really look at the safety, but when he conducted to the phase 2 portion was to look at the response rate, meaning what is the chance that a patient will experience tumor shrinkage by our measurement criteria. And so as part of the study, in addition to looking at the potential efficacy of the drugs, we also did some correlative studies. So some studies to look at potential studies to help us assess whether or not the drugs were actually doing what we were hypothesizing they would do. So after a month, and at the time of treatment and clinical trial enrollment, at the time of cycle 1 and day 1, and then the time of cycle 2, day 1, so a month later, we collected blood to look at circulating tumor DNA. And we kind of looked at two things. One, we wanted to look at the changes in KRAS mutant allele frequency. So essentially the changes of percentage of the KRAS mutations, as well as looking at the changes of all mutations found in the blood. And so I know that there's many of you are very familiar with what ctDNA is, but for those that are of you that are not, what ctDNA or circulating tumor DNA is, it's more or less what we call liquid biopsy, and it gives us a snapshot of kind of what's going on with the cancer in the whole body as a whole. And so as cells within our body grow and die, they shut off DNA, and that also includes cancer cells. And so what ctDNA looks at is the proportion of cancer DNA compared to all the DNA in our body. And so at the time, in July of this year, we enrolled a total of 50 patients at that time. And what we saw was that the response rate, meaning the percentage of patients that experienced tumor shrinkage of about 30% or more, was 35%. And so historically, when patients get second-line chemotherapy in colon cancer, the likelihood of tumor shrinkage of 30% or more, meaning a partial response or better, is about 5 to 10%. So this was about anywhere from 3 to maybe 7 times higher than what we would typically historically see. And when you look at disease control rate, meaning the percentage of patients that had disease control where the tumors were not growing or spreading or potentially even shrinking or even disappearing, was about 94%. And then on the right here, we look at median progression-free survival. So what that means is the time that the cancer is being controlled. When we look at the tumor measurements across the clinical trial, what we saw was a median PFS of 9.2 months. And historically, we would expect to see about 4 to 5 months based off previous clinical trials. So despite this being a very small sample size, we have a very good hint of signal of the activity of what this drug can do with chemotherapy in patients with KRAS mutant colon cancer. And so as part of the correlative work, we look to see the likelihood of responses based off the KRAS mutations. And so while this is a very busy figure, what this is looking at is each individual patient that was enrolled in the study. And on the blue on the left are the patients that experienced either a complete response or partial response, meaning tumor shrinkage of 30% or more. And then on the left, on the middle, we have the yellow, which is stable disease. And on the right, we have patients who progressed. And we can see that irrespective of what type of KRAS mutation they had, whether they had G12D, which is the most predominant mutation in colon cancer, and we have G12V, 13D, et cetera, patients did experience a response of at least, did experience a clinical benefit of at least stable disease or even responses, irrespective of the KRAS mutation. And so as part of the correlative work, which we discussed earlier, is that we did collect some blood to look at the changes in the KRAS, what we call mutant allele frequency. And so through statistical considerations, we determined that the KRAS mutations needed to decrease more than 90% to be considered a meaningful change. And what we looked at was to see whether or not the changes in KRAS mutant allele frequency correlated with correlated with the radiographic response, meaning if your KRAS mutation decreases, is that associated with the likelihood that you'll see tumor shrinkage on a scan? And so what we saw was that there was a strong concordance between the two. So in patients that experienced a complete response or partial response, nearly 90% of these patients experienced a KRAS mutation decrease of at least 90%. Similarly, when you looked at the other side, we did see that if your KRAS mutation didn't decrease as much, you were unlikely to experience this shrinkage in tumors based off the scans. And so in addition to looking at the radiographic response, we are interested in also looking at the clinical response, meaning if your KRAS mutant allele frequency decreases, is that associated with you benefiting in terms of surviving the cancer, and specifically looking at preventing the cancer from growing or spreading? And so we saw that if patients did have a decrease in their KRAS mutation burden, they were likely to experience a bigger clinical benefit than those that didn't experience a decrease in their KRAS mutant allele frequency. So those two things did show a high concordance as well. And so based off these results, we are proceeding to a phase 2 study, a randomized phase 2 study, where we're looking at combining FOLFIRI bevacizumab versus FOLFIRI bevacizumab in combination with onvansertib. This is going to be a randomized phase 2 trial where I believe we're going to be enrolling close to 150 patients. And this will be open across the country, hopefully within the next several months. And so similarly, based off a lot of these activity that we saw in colon cancer, we know that KRAS mutations are very prevalent in pancreatic adenocarcinoma. So we are currently also conducting a phase 2 study in pancreas cancer, where patients that have likely prior to receive gemcitabine and nab-paclitaxel or gemcitabine, who didn't receive prior 5-FU or irinotecan or nanoliposomal irinotecan would be eligible to be enrolled in this study. So this study, again, is a second-line study for specifically in patients with KRAS mutant pancreatic ductal adenocarcinoma, where they would receive onvansertib in combination with 5-FU and Naliri. And this study is currently ongoing and enrolling across the country. And so I know that there's been a lot of interest, and I know that you've had a lot of great speakers, and we see a lot of information about KRAS G12C. And so this is a mutation that is very prevalent in GI malignancies, most predominantly in the right-sided colon cancers. Typically, we think about ampullary cancers or right-sided colon cancers, but we can also see them in a less common incidence in pancreatic or hepatobiliary malignancies. And so this year at ESMO last month, we did get updates from both studies, from Crystal 1 as well as from Codebreak. And so what Crystal 1 was, this was led across various institutions, including at the Mayo Clinic by my colleague Tony Saab, who presented some of the earlier results at ASCO GI this year. They presented the updated results of patients that were enrolled in the study that received MRTX845 or adagrasib as monotherapy and in combination with cetuximab, which is an anti-EGFR monoclonal antibody. And so, as we know, KRAS G12C is prevalent in about, again, 3 to 4% of patients with colon cancer. And we know that this is associated with, again, a poor prognosis and potentially can be a biomarker suggestive of response. And so adagrasib is an oral small molecule inhibitor that inhibits KRAS G12C and has a very long half-life and suggesting potential strong efficacy. And so based off some preclinical data where we know at least preclinical and clinical data where we know that KRAS G12C inhibitor monotherapy may not be as effective due to the feedback loop mechanism. So on the right here, we can look at the MAPK pathway, looking at from KRAS, RAF, MEK, and ERK. We know that oftentimes what happens is that cancers are able to escape this inhibition or the blockage of KRAS by either activating upstream or downstream on the pathway. And so the study looked at the combination of adagrasib with cetuximab, as well as looking at adagrasib by itself. And so here's the brief schema without going into too much detail. They looked at, again, the drug as by itself, where patients got the drug twice a day, or they got it in combination with cetuximab. And so when you look at the patient baseline demographics, this is just the demographics for all the patients who were enrolled in the study. I think the main thing to really take home without going into too much detail is the prior lines of systemic therapies. So, and on average, most patients had at least 3 lines of prior therapy. So this would be their 4th line of treatment. We know typically in this scenario, at least with colon cancer, the treatment option would be kind of limited to either regorafenib or TAS-102, which provide modest responses. For some patients, they can be particularly clinically benefit, but in general, the responses are pretty modest. But some patients had up to 9 lines of prior treatment before going to the study. So some of the individuals that were enrolled in the study did receive a lot of treatment. And so here's the waterfall plot curve. So what a waterfall plot curve is to look at each individual patient that was enrolled in the study. So this is looking at patients that got adagrasib by itself, so just the KRAS G12C inhibitor. And each individual bar represents every single patient that was enrolled in the study. And in terms of the different colors, they denote what the patient experienced in terms of response, meaning did they have a partial response, meaning tumor shrinkage of 30% or more? Did they have stable disease where their cancer was controlled? Or did they unfortunately experience progressive disease where their cancer broke through grew by more than a certain criteria, in this case, 20%? So what they saw was that in all the patients enrolled in the study that received just the drug itself, they saw that the response rates were about 20%. So when you compare that to something like Stivarga or regorafenib or TAS-102, which is Lonsurf, the chances that tumor shrinkage, that we would exceed tumor shrinkage is about 5%, if not less. So despite patients having a lot of treatment before enrolling on this study, there was a good amount of clinical activity where nearly 20% of patients experienced tumor shrinkage of 30% or more. And so this is what we call, this is kind of like a spider plot, or tells us kind of the duration of treatment. As we can see on the bottom on the x-axis is that the time of the duration of how long patients were on treatment. We can see that several patients reached nearly that 2-year mark, and several of these are my patients in the clinic, where they did experience a significant benefit from just the drug itself, and they continue to tolerate treatment well. Similarly, here we have what we call the Kaplan-Meier curves, meaning the duration of the clinical benefit that patient received on treatment. So we have on the left, we have progression-free survival, and then on the right, we have overall survival. So the median progression-free survival was 5.6 months, and the median overall survival was 19.8 months. And so what that means into context is when patients were enrolled on the study on the right, is that on average, patients lived on average about 20 months after being enrolled in the study. And so again, this is after going through many lines of therapy. This is just really taken into consideration from the time of study enrollment. In terms of toxicities, which I think is probably the most important thing, especially when we're trying to balance quality of life as potentially the goals with treatment, we can see that the treatment was relatively well tolerated with primary gastrointestinal side effects. So diarrhea, maybe some nausea, vomiting, and some fatigue, but otherwise the treatment was well tolerated with minimal grade 3 toxicities. And so what grade 3 toxicities are those that potentially may require hospitalization, and those are based off specific criteria that look at. So in general, most of the side effects were manageable that did not require any significant intervention. So similar to the initial previous curves that we looked at with adagrasib by itself, here we have the adagrasib plus cetuximab arm, where patients receive adagrasib in combination with cetuximab. And here we can see a very impressive response rate of 46%, meaning 46% of patients experienced tumor shrinkage of 30% or more, and the disease control rate was 100%, meaning every patient that was enrolled in the study had tumor stabilization or shrinkage. And we look at any tumor shrinkage because we have specific criteria that look at before we denote it as, again, a partial response. 93% of patients did experience some tumor shrinkage of some magnitude. So again, this looks like the combination strategy may be more effective than the drug by itself. Similarly, here we look at the duration of response. So it was higher in terms of the combination rather than the monotherapy. And so here again, we look at both progression-free survival and overall survival. So without going into too much in the numbers, again, median progression-free survival was about 7 months, and median overall survival was about 13.4 months. Many of you may ask, why was the median overall survival lower? I think some of it has to do with the sample size, and the data still is relatively immature. So we'll see how this number changes over a period of time. But this is kind of what the early results that were presented in the study. So where do we go from here? Lastly here to the side effects, So in terms of the toxicities, the cetuximab can be challenging. It does add some increased side effects with cetuximab. The main side effects tend to be mucocutaneous, so skin-related toxicities, mainly with like acne, like rashes on the face. And if it's severe, it can be more than just the face, including the whole body. But otherwise, the treatment didn't, the combination didn't really add much more than just the drug itself in terms of the side effects. So the combination was in general well tolerated and looks like a very effective strategy moving forward. And so where do we go from here? So based off these early results, there is a phase 3 trial that is ongoing. So the phase 3 trial is looking at the combination of adagrasib and cetuximab in the second line setting, where patients would get randomized to receive the combination or chemotherapy. So if a patient got FOLFOX first, then they would get randomized to potentially receive FOLFIRI or vice versa. This study is a phase 3 trial that is currently ongoing across the country. And actually, it's international, so across the world. And this study is open at many sites, including the Mayo Clinic. So there are, without being biased, the reason why I'm presenting really the Mirati data is this is a study that I have a lot of experience with. There's also CodeBreak 300 as a disclaimer, which is looking at the combination of sotorasib in combination with panitumumab. And that's also a phase 3 study that's ongoing, but I haven't had any personal experience with it just because we can't open every single study, even though we want to. So that's another option as well. So what are the future directions for KRAS? And so, we've kind of briefly touched about what we call these direct inhibitors of KRAS, so G12C. There are several compounds that are being under investigation through clinical trials. We saw adagrasib, we know that we have sotorasib with Amgen, we have GDC 6036 with Genentech, as well as the Jacobi Bio compound. But shortly coming down the pipe, we will see studies involving G12D. We know that Mirati has their compound, which should be opening shortly in phase 1. And then eventually, I think things that we want to understand is how can we overcome treatment resistance? So we know eventually tumors can change and they can become resistant to these direct inhibitors of G12C or G12D potentially. And so how do we overcome these mechanisms of resistance? So not only do we have primary resistance, meaning treat patients that are not benefiting from these treatments because of either other mutations within other pathways, or what we call mutational heterogeneity, meaning that the mutation within each tumor themselves may not be expressed the same, or they may not show the same benefit. Or patients may eventually develop what we call secondary resistance, meaning treatment initially works and then eventually runs its course. They've presented some of the data and it can be related to new mutations that develop. So eventually there may be some more irreversible G12C, D, or C inhibitors that can potentially overcome these mechanisms of resistance. And then what about targeted regulation? So we know that the MAPK pathway, or in this case, the MAPK pathway is the pathway that KRAS sits on. We know that there are several oncogenes that are related to the activation or downstream activation of this pathway. One is through SOS, which provides the complex which KRAS G12C inhibitors work on, or SHP2 inhibitors, which leads to the downstream activation of MEK or ERK, which is downstream of RAS. So the pathway that leads to RAS activation, eventually causing tumor cells to grow. These are potential targets that are of area of interest. as well as inhibitors downstream of KRAS. So we can see that downstream of KRAS, it's kind of like going down the freeway. So you have RAS, RAF, MEK, and ERK before it goes intratumorally. We know that there are a lot of interest in looking at agents that target some of these downstream inhibitors, many of clinical trials that we've led, including MEK inhibitors or ERK inhibitors, and potential inhibitors upstream. So are there ways to block KRAS from above? So those are kind of the areas of interest that we look at when we look at direct KRAS inhibition. And then lastly, we know immunotherapy is always an area of interest, and this remains relevant also in targeting KRAS. So one potential strategy here is on so the development of cancer vaccines. So there are various different types of vaccines that are under development, peptide vaccines, messenger RNA vaccines, and then dendritic cell vaccines. And what these vaccines do is potentially promote anti-tumor activity through the stimulation of the immune system against KRAS immune cells. Then we have B, we have what we call adoptive cell therapy. And what that is that T cells or immune cells and NK cells are removed from the body, and they're genetically engineered through viral vectors. And then they're reintroduced in the body. And these T cell receptor therapies potentially can help the body better recognize these KRAS mutant cells, to where when the cells start to grow or potentially act up, they can really inhibit their activity or potentially kill these cells. And then lastly, we have what we call TIL therapy. And so what TIL therapy is when tumors are initially removed, they isolate or remove these tumor infiltrating lymphocytes, so the immune cells that are active against the cancer cells, and they find the cells that are most active and those that are most effective, and they help generate more and more of these cells, and they reintroduce these TILs back into the patient's body. So these are potential 3 immunogenic strategies that continue to garner interest, and that are also being conducted right now through clinical trials across the country. So in summary, while KRAS remains a biomarker associated with a poor prognosis, we know that recent advances have demonstrated to be potentially a druggable mutation. So we are making progress. Future strategies look at combination strategies with chemotherapy, with immunotherapy, or other targeted therapies. And lastly, thank you for all you guys do. You guys are the reason why we continue to do all the research, and really, this is a great opportunity for me to come speak to you guys, so thank you very much.
Well, thank you so much for that. We do have a couple questions if you have a sec.
Yes, so should I just read the questions and go ahead and answer them?
I'm gonna go ahead and ask you the questions and put you on the spot. So, okay, so, okay, I can't say, all of a sudden I can't spit it out. It's a nerve. Okay, let's try that one.
There we go.
How come I can say it in my head and then it comes out of my mouth like just mush? Okay, can you speak to putting that with prior exposure to, okay, can you just read the question, because I cannot seem to get these words spit out.
Not a problem. So the first question, it says, my dad has already had irinotecan. Why is this an exclusion criteria? Is there any way he can still access bevacizumab through compassionate care? So I think the question is, so firstly, the reason why this was an exclusion criteria was to look at patients that didn't have prior irinotecan, because we know that cancer cells can eventually develop secondary resistance. So what the intent of the clinical trial was to really look at patients that only had one type of chemotherapy. And the drug also showed that, at least in the lab, is that it works together with irinotecan. So specifically, they were trying to look at it in the second line setting. They are in development for looking at a first line trial, so for people who've never had treatment, but this was kind of the direction that the company wanted to go in. In terms of, they did have a compassionate use program, but this is a very small company, and so unfortunately, that program is kind of on hold because they want to make sure that there's enough drug available for patients that are currently on the study, but it could potentially reopen in the future. So I know that they know that they want to help a lot of patients, as many as possible, but for at least for now, that the compassionate or the expanded access program is kind of on hold.
Okay, that was great. I think I can spit out the next one. Okay, so as far as the MRTX 1133, is the MRTX 1133 phase 1 going to include other drugs with that, or is it just going to be a monotherapy trial?
It'll probably be both. So I know that Mirati is still under development with the drug. and I know that the plan is hopefully to open in the next several months, but when it will actually open, I couldn't tell you, but it's going to be nationwide. It'll be multi-center. So I'm sure there will be multiple centers that should be close by to many of you patients here.
Okay, and then as far as G12S patients with colorectal cancer.
So for the onvansertib, yes. For either one of the Mirati or the Amgen studies, no, because that's specifically for G12C. But if this is in regards to the onvansertib compound, yes, they do accept all RAS or NRAS mutations.
Okay, then the last question about crossover. Oh, not the last question, but as far as crossover.
Yeah, so it said, have you seen any KRAS G12C patients go from onvansertib trial to KRAS G12C target trial? Yes. So I've had several patients that came in to get chemotherapy specifically in the second line setting that went on the onvansertib trial. And then once they either progressed or became intolerant for the treatment, we did enroll them to receive a G12C inhibitor, and there still looks like there's clinical benefit. So I think there are a couple things or a couple nuances to that. One, I think is really important about testing. So I would recommend retesting if a patient goes on to any study to make sure that they still have KRAS as the prevalent clone, just make sure that that target is still relevant. But yes, you can go to multiple studies as long as you meet the eligibility criteria. So that is not an issue.
Okay, great. And I'm just going to keep reading down so G12D treatments across Europe. Any thoughts?
Yes. I don't know I don't know the answer yet about when when the Mirati compounds, MRTX 1133, when it specifically opens, but I believe it will be an international phase 1. And then eventually, as the study continues to grow, it should eventually be open in Europe as well. So that's the goal. And
actually, somebody posted the question I was thinking about, targeting EGFR and in RAS. I know it leads up down it's the top of the pathway to RAS. How does that play into treatments when it comes to colorectal cancers?
Yeah, that's a great question. So in the past, probably in the early 2000s, our thought process was that RAS in patients that had RAS activations, anti-EGFR therapies like cetuximab or panitumumab would be more effective. But as we began to understand about how these mutations work, we know that these drugs actually have a detrimental effect, or may not work or even have a detrimental effect. So if you do have a KRAS mutation, whether it's, in this case, Q61, or you have G12D or G12C, we know that these drugs aren't effective. And so we wouldn't recommend those treatments.
Okay, great. Okay, well, I appreciate your conversation with us today. And nobody gets out of here quite completely without really engaging with our audience. And how we're doing that is kind of our war cry. And, we're going to be doing a countdown to include you in it. And so if you will cooperate with us in the room, and we'll do like a countdown, like a 1, 2, 3, and all together say kick cancer's KRAS. Are you game?
Let's do it.
Okay. All right. 1, 2, 3. Kick cancer's KRAS. All right, you guys. Thank you so much for joining us today. I appreciate it. And we hope to have you back again real soon.
Anytime. Thank you so much.
Okay. Bye now. Okay. Well, we're close to on time. So, okay, we've got upcoming next, and hopefully I'll be able to get the marbles out of my mouth here momentarily. Okay. And I need to be practicing names ahead of time and making better notes. Okay. Dr. Sumita Bhatta, I hope I didn't butcher it too badly. Did I? How'd I do?
Perfect.
Not bad. Okay. All right. It sounds like totally like I think I've got it. And then I get up here and stuff comes out. Okay. So Dr. Sumita Bhatta is a board certified oncologist and the vice president of global medical affairs and the head of oncology at Amgen. She's had over 9 years of experience in the biopharma industry and held a number of research and developmental roles. And so today we're just pleased as punch to have you join us. And I see, what did we call your topic? Understanding how a new drug treatments are brought to market, which we're really interested in knowing about and having better understanding. So I hope that we have a few minutes at the end to answer questions. So welcome.
Thank you, Terry. I'll go ahead and share my screen. And you'll tell me if I did it right in just a second.
I think, well, we need the presenter mode.
Okay, here we come.
There you go.
Yeah, okay. Thank you so much, Terri. Thanks to everybody in the room and online. This is a great pleasure of mine to be able to speak to you all. I wish it could have been in person. I just want to echo what Dr. Ahn had just finished with, which is the patients and the patient caregivers are really my why. It's the why I go to work and why I work in drug development. And so this is really a huge highlight for me to have this opportunity. So thank you very, very much. So today, we will be talking about understanding how new drug treatments are brought to market. And we'll go through this overview. So first we'll speak about overview of the drug development process. And this will really be the bulk of the presentation. We'll talk about how then regulatory authorities look to analysis of data from clinical trials and making their decisions at a very high level. We'll look at what we should be thinking about when we compare clinical trials. What are some considerations that should come to mind? And then finally, we'll end with enrolling in a clinical trial. What are some of the benefits? What are some of the risks? And how can you go about doing that if you should so be interested? Cancer drug development is a multi-step process. You can see that there are multiple parts of drug development that lead ultimately to a patient being able to receive a drug. The first step is discovery and development. First, we have to find compounds that could be potentially beneficial. The next step is preclinical research. So before these patients even before these drugs even enter a patient, there is a ton of research that is done to make sure that it's appropriate to move on to the next step. The third step is clinical research. It's what a lot of you may have already been involved in or may consider being involved in the future. And that's really investigating whether a new drug can benefit you in a safe manner. Step 4 is the regulatory review process. Here we've highlighted the FDA, but as you know, there are multiple health authorities across the world that use similar type of processes in determining whether or not drugs really should be available for us. And step 5 is really the FDA approval and post-marketing process. So sometimes not all questions are answered in the clinical trial that leads to approval. And this opportunity, step 5, is a process by which questions can be answered in greater detail. So this is an interesting figure because it really highlights the multiple steps that are required in each of these different segments to get a drug to market. It also highlights the number of potential drugs that get whittled down before they actually make it to a phase 3 trial and subsequently to an approval. You can see from the funnel that there's multiple drugs that enter the drug discovery process, multiple potential candidates. And through these steps of drug optimization, which is really ensuring that the structure of the molecule is appropriate, preclinical testing in both cell lines and animals, and then clinical trials, fewer and fewer drugs actually make it to the finish line. That's important to highlight because you can see that the median time to develop a cancer drug is about 7 years, and the median cost to develop a cancer drug is around $650 million. And that is very much due to the fact that all of these processes with so many candidates have to take place before you get to the winning asset. So we'll talk a little bit about step 1 and 2 of the drug development process. That is drug discovery and preclinical research. Drug discovery can happen in multiple ways. Sometimes it's accidental. Sometimes drug discovery comes from things that we already have in nature, like plants, funguses, things that are, generally already available. For example, a cancer drug that we all know very well, paclitaxel, came from a tree. And there are other drugs that have come from things like sea sponges. And so this accidental discovery happens. Amgen takes what we call a biology first approach, whereby we try to understand the biology of a drug, of a disease, and then we try to identify a drug that can target that aberrant or mistaken pathway. So for example, we try to analyze the genetic makeup of a cancer and compare that to what happens in healthy cells. And once we can figure out what the difference is, we can try to develop a drug against that. That is by and large how targeted therapies are developed. That is also an approach that's taken for immunotherapies, which are drugs that harness the body's own immune system to fight off different types of cancers. Once this is done and drug discovery has occurred and the actual compound has been optimized, these agents can go into preclinical research. And preclinical research encompasses a host of different types of activities. The drugs first undergo laboratory testing, generally in human cell lines, to determine whether or not there's any impact. And if there is, then drugs can go into animal testing. This is generally called the in vivo approach, to assess their potential side effects, but also to determine a safe dose for first in human studies. A term that's often used here is MABEL, which stands for minimal anticipated biological level. And this is what we can get from these animal tests. And this minimal anticipated biological level is often used, not always, often used to determine the starting dose for phase 1 trials for which humans are now going to be enrolled. The next step is the clinical research process. Before this can happen, an investigational new drug application is submitted to health authorities. That's called an IND for short. And the IND has within it the data that was just generated from the preclinical studies and more information about the drug compound, as well as the clinical plan that we think we're going to need to demonstrate the activity of this drug to benefit patients. That regulatory authorities will look at this data and determine whether that's an appropriate plan. And if it is, they'll allow us sponsors, which sponsors, by the way, can be pharmaceutical companies or it can be academic centers, to initiate clinical trials. So the first phase of clinical trials is phase 1. We generally consider this early phase clinical trial testing. And here the primary goals are to establish safety and the dose of the drug. Oftentimes, we're also looking to understand how the drug works in the human body. We use some terms such as pharmacokinetics and pharmacodynamics to describe this. Pharmacokinetics can be things like how well is the drug absorbed? How is it metabolized? How is it eliminated? Which organ in the body is responsible for elimination? Meanwhile, pharmacodynamics can be such elements of, is the drug acting upon a certain marker in the body that we would expect it to, maybe decrease of a protein level or something similar that could just let us know whether the drug is achieving its target. The length of time for this phase of clinical development is variable. It can either range from several months to several years. It can be less than 100 patients to more than several hundred patients, depending on how the study is designed. Generally, the next step in clinical trials is phase 2 drug development. And again, the goal here is to continue to now test that safety that we think we are confident about in phase 1, but also now look for signals of efficacy. Phase 2 drug development can be done as single-arm trials, where patients are only getting the drug of interest, or this can be randomized, where patients are getting the drug of interest against what they might already be receiving that's already been approved. Again, there's a lot of variability in how this gets done, and this can take months to years. We say 3 years here, but again, this is a very variable process depending on the type of drug that's developed and the multiple different pathways that this phase 2 drug development process can take. Now we're sort of moving into the later phases of development. Phase 3, for example, is when we're really seeking to confirm now the effectiveness of a drug against what is generally considered standard of care in a larger group of people. And this can take up to 4 years, sometimes even more, depending on the complexity of the trial. The goal here is now to really confirm whether this candidate drug can improve outcomes over what's already available to patients. And finally, and this can lead to approval of the drug. generally speaking, that phase 4 part of drug development is there for monitoring after approval and to answer some of those unanswered questions that we didn't necessarily get through phase 1 to phase 3 of development. Now, I just highlighted what's considered a traditional path to market, but if a drug is spectacular and has a tolerable side effect profile that makes it promising, there are some approaches that the FDA can give us to make drugs available more quickly. So on the top left, we'll start with fast track designation. So this is an opportunity to speed up drug development and review of data to allow faster approval of drugs to treat serious conditions for an unmet medical need. So an unmet medical need, what does that exactly mean? An unmet medical need is where potentially there's no treatment available for a particular disease, or where there's a potential to improve upon what's already available, whether it be with greater efficacy or a better safety profile. What is the benefit of fast track designation? Well, it offers the opportunity to interact with our regulatory authorities with more agility. We have more opportunity for written communication, more meetings. Potentially, we have a rolling approval process, which would mean that as data is generated, it gets submitted for ongoing review rather than everything being compiled and submitted all together later on. And again, the goal here is to speed up the drug development process. Typically, the sponsor asks the FDA for this designation, and the FDA can then decide whether or not it's appropriate to grant this or not. The next type of approach is called breakthrough therapy designation. And this is generally given when drugs have demonstrated already that there's substantial improvement or the potential for substantial improvement over existing therapies. This affords many of the same opportunities that fast track designation gives, such as more agility in communication. This is generally either requested by the sponsor or even at some times requested by the regulatory authorities, given what they've seen with the data to date. On the upper right-hand side of the slide, you can see that there's something called an accelerated approval. This is a program that allows for quicker approval of drugs for areas of high unmet need or serious conditions based on generally what's called a surrogate endpoint. So what's a surrogate endpoint? A surrogate endpoint is a measure that often predicts overall benefit or ultimate benefit to a patient. Generally, it's not the true marker of, for example, survival, but it can be, for example, response rate that correlates with survival in the end. If a drug is approved via the accelerated approval pathway, generally another trial will be needed to confirm the results with the endpoint of interest. And if, for example, that confirmatory study doesn't confirm what the accelerated approval pathway has shown, a drug can actually be pulled from the market. The final approach that I want to discuss today is priority review. This is an opportunity that the FDA can provide whereby they commit to taking action on a drug application within 6 months instead of the standard 10-month review timeline. This is, again, done when there's promising activity, when there's really high unmet need, where there's a desire to make drugs available to patients quicker than normal. And generally, this is something that the sponsor can request, and then the FDA can look at and decide whether it's appropriate and give that designation. So we've talked about generally what the lengths are for development. Here is an example of, an accelerated drug development timeline from initial clinical trial to FDA approval and after. And what the reason we want to show you this is, again, to highlight that when a drug has compelling properties, there can be an opportunity to accelerate its development. And I think this is the goal for all of us, as long as it's done in a responsible way to allow patients to receive therapies that they may benefit from as soon as possible. Again, it has to be done in a responsible way. So here you'll see phase 1 development for this hypothetical drug began in August of 2018. The drug received many of the opportunities afforded by the FDA, breakthrough therapy designation, priority review, and ultimately accelerated approval within a 3-year time period. You can see then that the line below that demonstrates the phase 3 timeline. This is the confirmatory study that's required to then make sure that the data that was seen in that accelerated approval study is true and is confirmed by the endpoint of interest here. And there are also other questions that hadn't been answered. And so the drug, it has an ongoing phase 4 study as well. Again, the important thing to highlight here is that there are opportunities for drug development acceleration. It has to be done in a very thoughtful way in partnership with regulatory authorities. And the key goal is to ensure that it's responsibly developed to ensure that patients are not being exposed to an unsafe therapy, just for the sake of speed. Now, how is data analyzed? The FDA has a team of physicians, statisticians, chemists, pharmacologists, a variety of other scientists that review the data that's been submitted. And generally, this can be data from several clinical trials that get submitted through a marketing application. The FDA, with all of its team, can opine on the value of that product for patients. They may need additional data. They may ask for an advisory committee where they call upon experts for their recommendation. They may also request additional studies or more data to make sure that they feel comfortable with the decision that they're making. Primarily, the decision is based on the benefit versus the risks. And this is probably true for many of the things that we do in life, but this is, of utmost importance here for the FDA. And then they render a marketing approval decision based on all the available data that they have in partnership with the company or the institution submitting the data. Now, you'll come across tons of data from, you'll go to ASCO, you'll go to ASH, you'll go to, many of these congresses where data presentations are being made one after the other. And how do you interpret all of this? It becomes very complicated. And so, generally speaking, cancer drugs are evaluated for approval in this phase 3 setting when they're typically compared with what's already available to patients. And this is called standard of care in a clinical trial. The reason that this is done is comparison with standard of care allows assessing the new drug to see if it's superior, equal, or inferior to the existing drug that's being used to treat the disease. And this is true not only for efficacy, but also for safety. When you see trials from different drugs, there is a tendency to want to compare those results to one another. But it's actually quite important that we take caution when doing things, when trying to compare results from separately conducted trials, because these trials can be very different in the types of patients that were enrolled, the interventions that were made, the severity of the disease, what patients may have gotten prior to enrollment on the studies, et cetera. So there's a multitude of factors that make it very challenging to compare one study to the other. And why is this important? It's just very easy to misinterpret data without fully understanding the differences in those trials. So what are some possible benefits and risks of joining a clinical trial? You'll see them here on the screen. Some possible benefits of joining a cancer clinical trial is that you may ultimately advance cancer research and help someone else with the same disease that you have that might be afflicted with this disease later down the line. You might also get benefit from the drug. You could get a treatment that's not available outside the trial that you ultimately do benefit from. You'll likely be able to see your cancer team more often because these trials often require more visits for some of the monitoring that we discussed. And so that you can, be checked more frequently for some of the side effects you might be feeling. And some of the expenses that you may face may be paid for as part of your enrollment in a clinical trial. However, there are also risks of joining a cancer clinical trial. Especially in early stages, we really don't have all the answers for side effects. And oftentimes this evaluation is being done in patients for the very first time. Also, the treatment may simply not work. It may not be efficacious, especially in the earlier lines of clinical trials. While I just said one of the benefits could be more frequent visits to the hospital, that could also not be a benefit for folks who live further away from the treatment facility or have more barriers to getting care. So more doctor visits may not necessarily be a good thing, as it includes more time and travel. If you're part of a randomized study and you get randomized to the standard of care arm, you may not have a choice then to get the active therapy. And while I said some costs may be covered, this is not always the case. Insurers may not cover all costs of the clinical trials. And so this can be a financial element that needs consideration. Now, if you're interested in a clinical trial, or a loved one is interested in a clinical trial, or a friend or someone else that you work with is, it's important to know that generally these clinical trials are carefully supervised, monitored, and documented because this is research that is conducted in humans. They help determine if the study drugs and treatments are safe and if they produce desired results. And there are several ways in which patients can enroll. That information is listed below. Your physician can also tell you about what may be available at their center. And with that, that concludes the main portion of the presentation. I'd be happy to take questions, but before I do, I just once again want to reiterate how important it is for us to have these types of engagements, how valuable your feedback and questions are, and that you truly are the reason why many of us go to work every day, especially me. So that's it.
Well, thank you. That was wonderful. It really kind of helped simplify what the process is in order to get to a clinical trial, what a drug has to do in order to get to us as becoming the standard of care, the latest and greatest. I do have a question, and I'm not sure if there's going to be anything from the audience, but if a drug's already been approved for another use, does it have to start back at phase 1? How does it get approved for a different type of a use?
Yes, the answer to that question, Terri, is it does. It does have to go back into phase 1 testing because the dose may not be the same in a different type of patient or a different type of cancer. So all of the safety data has to be regenerated from the beginning. We can probably, even the preclinical data, frankly, is generated for new tumor types.
Okay, and then as far as that data that's analyzed, it's our information about what's going on in our cancer. It's not our personal information.
So patient level data is submitted, and I think that's the crux of the question. Forgive me if I've misinterpreted it. Patient level data is submitted as well as aggregate data to regulatory authorities, and numerous analyses are done to look at the data and cut it in different ways to ensure that we're properly understanding the side effects and the efficacy parameters. So it is your patient level data or your loved one's patient level data that is being examined, as well as group aggregation of data.
Okay. All right. So part of it becomes a math solution then. Okay. And then what about, let's see, I'm just going to read the question from the chat. I've noticed no one has discussed using AI, which is artificial intelligence, in drug development to reduce developmental timeline or identify new drugs or repurpose approved drugs. Is this being done or in seen that you know of?
I would say that there are efforts being made to address these issues. I think AI, we still have some time for that to be done, but it is being looked at. Real world data is another place that there's tremendous promise for. For example, patients are being treated with drugs in the real world setting, and sometimes this data can be used to give us some information about the historical control arm. And that can limit the amount of data that has to be generated in exposing patients to drugs that are already available, because we have numerous plethoras of data that already exist. So you're right, we haven't mentioned it, but it is going on in the background. And real world data especially is a major contributor. It's something that regulatory authorities look at, something we're expected to generate, frankly. The other thing we haven't mentioned that is really important, I think, to mention is diversity in clinical trials. And so the FDA has now mandated that we provide a diversity plan. So how are we going to have representation from different ethnicities, races, genders, et cetera, as part of these clinical trials? What's our plan? And we're obligated to think about this. I think it's extremely important that we think about this because we know that the patients who actually receive the drugs once they're approved are not necessarily the same that were enrolled in that clinical trial. So we do need to start generating data on diversity as well.
Okay, great. And I think we had a question from the audience.
We saw a big change in drug development timelines with the COVID vaccine and a push there. With the Cancer Moonshot initiative, do you see the drug development timeline being compressed? And then kind of a secondary question to that, how much of the timeline is medically driven and how much of it is just governmental or bureaucracy driven?
Yeah, I think, the moonshot, I think it had a second set of wings after COVID. Moonshot has been around for a little bit of time, but COVID has really highlighted the ability to bring drugs to market faster. Now, whether or not this will improve the opportunity for oncology drugs, I think is still a bit unknown. I will tell you that there is a sense of urgency in development of these drugs. I think we all feel it, including the government. So I think, to answer your question about whether this is bureaucracy and process versus the medical need, frankly, I do think it is important to take our time in the appropriate ways to develop these drugs. We're all working, I think, as fast as we can, but we do really need to ensure that we're not subjecting patients to side effects or untoward impact just because we were on a kick for speed. And this is going to obviously be different depending on the level of unmet need. So where there is opportunities where nothing is available, the timeline is accelerated. Where there are multiple things that are available, I think you'll see it being a little bit longer. And I think this speaks to the variability in timelines in general.
Okay. As far as communication between like a pharmaceutical company and the hospital locations, do you have any suggestions how to expedite or improve that communication?
Terri, I may not fully understand the question. So, I don't know if this question comes from.
I think it's a matter of like, we'll see, like, say if we go to your website and we see there's a clinical trial or something is going to be enrolling and we're trying to get into it or have access to it, find out if we qualify for it. And it's not always easy for us as a patient to get the information that we need, even from like the PI, the principal investigator, or from the hospital setting. Do you have any suggestions as far as how we can like open up those lines of communication?
Yes, it's a great question. I think it is actually an area of improvement that we need to be working on as a collective universe. I think, your patient advocacy groups, KRAS Kickers, et cetera, can definitely help in navigating the process, but the process is extremely complex. The one thing that I can continue to say is, some of these websites can give you guidance and direction on where these clinical trials are being offered, but that doesn't necessarily mean that there's a spot available, et cetera. So there is some investigation that will have to take place. There isn't an easy solution now to know whether there's a slot available, for example, for a phase 1 study, even though that phase 1 study is running at a particular institution. So there is still some work to be done there, I think, in improving that dialogue. The one thing, though, I would tell you is be your advocate, as you all are. Contact your physician, get the available information. If it's not necessarily available at the institution that you're at, perhaps it's locally available somewhere nearby. And so I wish I had a better answer to that question, Terri, I really do, but I think it's still a pretty complex process to navigate and there's still work to be done there.
I think there's a lot of communication channels that need to open up. We have another question from the audience.
Yes, I wanted to ask you a couple of quick questions. One, on this breakthrough or fast track, can those expedited paths be combined, or is it just one or the other? And then the other question is, they're talking about for unmet needs, but what pharmaceutical company would spend millions of dollars on something that wasn't, an urgent unmet need? It just seems like almost all of them would qualify.
Yeah, it's a great question. So the first is yes. The first answer is you can have more than one of those pathways implemented in your drug development process. You can start with fast track, you can then be granted breakthrough, you can then be granted accelerated approval, and then a priority review. So in fact, you can have all of them. The second answer to your question is a great one. Why would someone spend their time and energy trying to develop drugs that don't necessarily have high unmet need? And the answer is, I think by and large, all of the drugs that are being developed by sponsors are generally committed to improving what's already available for standard of care therapies. Oncology drug development happens to be a situation in which the majority of drugs that are being developed because the solutions we have today are not necessarily optimal. You know, I think you could argue that there may be some other diseases that don't necessarily always fit the qualification of unmet need, but in oncology, this is generally the case.
Okay, and I see another one. Let's see, can you talk about why there's a possibility of, okay, let me rephrase that again. Excluding patients that have certain prior treatments, such as like immunotherapy, and then it kind of causes patients to like hold out to make different choices. I'm kind of trying to summarize the question that you see here in the chat. Can you speak to a little bit of how those items are decided?
Some of it is, generally the items of who to include and exclude in a clinical trial are based on perceived safety. So there are oftentimes compiled toxicity that can occur when somebody's already received a prior therapy and now they're receiving your therapy of interest or our therapy of interest. And the goal here, I think in general in medicine, but also in clinical trial development, is to first do no harm. So the idea here is to try to ensure that those patients that are enrolling on the clinical trial aren't being harmed by an investigational therapy because of some prior therapies, or maybe their renal function, or maybe their white blood cell count, et cetera, that puts them at higher risk.
Okay, and I think I can see we're kind of like running short on time. So I'm going to kind of like throw this out there too. It says do no harm approach has to be in the first place. This is from the chat. And enrolling patients into clinical trials makes a difference. I know that we need to include the washout periods to make sure that we know whether the treatments are being effective or not. Is there any way we can speed that up as far as the patients getting involved into a clinical trial rather than causing a delay? Because that sense of urgency when it's our life we're betting with is really intense.
I know it's the washout period is actually not so much for efficacy as it is for safety generally, because we don't want the prior side effects of the previous drug augmenting the side effects of the therapy that you would then be testing in a hypothetical sense. But I feel the struggle. I think it's a very important consideration. It's one that we grapple with every day. Sometimes patients don't have time to wait. They don't have time to wait the 30 days that we insist that they wait upon for enrollment in a clinical trial. And this is something we're actively talking about, especially if there is a drug where the side effects don't linger on that long and the washout period can be reduced. And so I will tell you that there is active effort being done to refine and revise these processes. But the sense of urgency that I hear with that question is real. I hear it, I understand it, and we're working toward it.
Please do hear our voices, because every single person in this room and every single person online, either themselves or someone else that they love and care about, whether it's just a friend or a family member, it's not just a theoretical sense of urgency. This is our lives that we're playing with. And so anything that you can do corporately, or that we as an organization or as advocates can do to help speed up this process, we need to know what that is, and whether that boils down from the communication, establishing the clinical trial, or what have you. It really is. It's a big gamble, and we need to do it. And we want to be able to get to the drugs faster, sooner, better. and make sure that they work. And so thank you so much for the presentation today. It really helped to kind of like give us an awareness and set a tone for our next upcoming session around clinical trials. But you're not going to get away either. As far as like, and everybody in the room is laughing. And we're going to ask you to engage with our audience here on site and just kind of do a countdown. And all together, I'm going to do 1, 2, 3, and then the audience and you, we're all going to say kick cancer's KRAS. Are you game?
I'm totally game. And again, I want to thank you for the opportunity.
Okay, cool. Okay, 1, 2, 3. Kick cancer's KRAS. Okay, thank you. All right, well, we're kind of getting everything reset here, and we've got our next speakers that are going to be coming up and joining us up on stage here, right? I'm also going to draw a couple of names for folks that are online. Come on up. All right, I'm going to draw a couple of names because even though y'all can't be here in person, it doesn't mean that we don't want to include you. And so, although we don't have like a fancy bucket here today, it is a go ahead. It is a fancy little champagne bucket because you guys are all like nothing but special and bubbly. Okay, and so we have somebody who's actually here, so we're going to pull that one out. Hang on a second here. Technical difficulties. Okay, I pulled up 2. Okay, well, Pete, you're already here, so you already have a bag, my friend. Okay. Oh wow, Eswarian Devarjan. Oh my gosh. All right, so like I need to go learn how to speak. Okay, last name is Devarjan. And so we're going to be sending you a swag bag. And since I'm up here messing stuff around, go ahead and get you guys all comfortable up here. We've got Laura Collins is going to grab a swag bag. And I'm feeling pretty good about myself up here. I'm having a good time. Robert Mills. So like you guys, even though you aren't here in person, we're going to send you some stuff as though you were in hopes that next time we won't have a hurricane or COVID. And we're all going to feel well enough to actually show up in person and you can actually hang out with us because the energy and the hope that we feel in this room is powerful. And it's not powerful because of any one person. It's a powerhouse because it's a community of us coming together. And in that togetherness, there is that strength. So, okay, with that, I'm going to do an introduction and hopefully I won't brutalize these names too terribly bad. Okay, but I probably will because like, you know, I ate lunch and once I eat lunch, that's it. It's over for about an hour. Okay. I should make you guys introduce yourselves is what I ought to do. Is Eric on screen too? He's on?
I'm here.
Okay, we can't see him. Can we pull Eric up? Okay. Well, Eric is getting brought back up online. Okay, so I'm going to start here. Ladies first. Brenda, Brenda, I've never actually asked you how to say your last name. Jesteadt. Brenda Jesteadt was diagnosed with lung cancer. She is a G12V like Victory patient in 2021. Prior to her diagnosis, besides being an impressive person, she just worked as a pediatrician for 35 years in the suburbs of Pittsburgh. Beautiful Pittsburgh, and I'm hopeful to get up there. She was an avid runner, cyclist, skier, and scuba diver. And last night we were hearing about some of the amazing activities that she was doing. And she's really getting back to an awful lot of them, but she's really trying to get back to more. So welcome, Brenda. We're happy that you're here. Okay, and then we also have, Mohamed Salim or Salem?
Salem, like Winston-Salem.
Oh, Salem. Okay, sorry, like Winston-Salem. Okay, great. I was like, all right, I've been butchering everything all day. I thought, you know, I was going to mess that one up too. Okay, I'm just tickled to death that he's here today. He is a GI medical oncologist at the local cancer center here at Levine Cancer Institute in Charlotte. And we recently saw some of your KRAS information that was just recently published and getting a lot of attention, shall we say. So so I was excited that we have somebody special from in town, and he's going to be able to join us and speak to our topic. Our topic coming up is, don't worry, Eric, I haven't forgotten you. We're going to be speaking about clinical trials, how that looks from a patient perspective, from 2 patients, and the other patient is Eric Madsen. Eric is a computer cloud architect currently residing in Wolfboro, New Hampshire. He was diagnosed with stage 4 colon cancer in 2019. He is a KRAS G12C colon cancer patient, and after responding some standard of care chemo, he went through a KRAS HIPEC surgical procedure that was almost curative. Those are words in a patient's life that have a whole new meaning. So welcome, you guys, and I'm thankful for you to be here. I'm going to step down, and then I'm just going to let you, and I'll talk to you from there. Okay, am I in the right spot? Okay, I'm going to pull up a chair here. Hey, Brenda. How's it going?
Happy birthday, by the way.
Thank you. Thank you. Oh, thanks. Okay, thank you guys for being here today. You want to tell us, how did you get involved in clinical trials?
Well, I think one of the best things I did was get a second opinion before I needed it, before it was imperative and I was scared. So I went to Texas and had gotten a second opinion and knew that when things went downhill, there was somebody there with a trial for me. And interestingly, in January, when that happened, the same day, my community oncologists flipped a 30-page paper of a clinical trial that they had just gotten in Pittsburgh and said, this trial's good, I think you should do it, which was the only education he gave me on that, by the way. And in my head, I'm going, yeah, I'm going to Texas. But I went home and read it and researched it. And I thought, well, this is a pretty good trial. Talked to my consultant in Texas, who agreed. He said, for the ease of doing it in your hometown, I think you should do it. And if it doesn't work out for you, it wouldn't preclude you from doing the trial that I had for you down here. So that was how I made that decision. And it went quickly because of that. My last chemo was February 2nd, and my first trial drug was March 10th.
Okay, so you're talking all this year.
Oh, yes.
Okay, and so
I'm 15 months from diagnosis, yeah.
Okay, and so I probably should back up a little bit. How did you even find out that you were KRAS anything?
When I went to Texas, I had never heard the word before. I had been told I didn't have a targetable mutation, and I was pretty sick at that time and really didn't investigate it further. And when I went to Texas, my consultant was like, oh, you're lucky you have KRAS. And I'm like, what, lucky? And, actually sat down and drew out the pathway for me, explained where he could attack the pathway at that point, and gave me a plan B and a plan C and hope for the first time. Since I was diagnosed, I share that when I was diagnosed, my oncologist walked into my hospital room where I was alone because of COVID and said, you have stage 4 lung cancer. This is bad. You have maybe 2 years, get your affairs in order. So getting the hope, learning about KRAS and the possible treatments that were out there was one of the most encouraging things that ever happened.
So they just started off basically telling you, good luck, we're sorry you got lung cancer.
Pretty much. Yeah. Well, he said, unless we can find, this was before my mutation stuff came back, my biomarker testing, but, unless you have a targetable mutation, and that will give you, more hope.
Yeah. And you're G12V. It's not like you're kind of like, one of the quintessential targets.
Right.
So, okay. All right. And then Eric, if you pull Eric up on the screen. Hey, Eric, nice to see you.
Hi.
Wow. Look at you. Like you're like, really, you work in the cloud. We
Yeah,
have no idea.
I'm way up here.
So that's what the cloud looks like. Cool. Well, thanks for joining us today. I wish you could have made it here in person. I know it just didn't quite work out, but I'm glad you're here. You want to kind of like walk us through like a 90-second version of your diagnosis?
Sure. I was, so as you said earlier, I was diagnosed with stage 4 colon cancer, G12C, back in 2019. and went through some standard of care therapy. Folfox was not effective. Folfiri and bevacizumab was, which then made me a surgical candidate. So I decided to have CRS HIPEC surgery that was, and then, like you said, almost curative. It did clear out all the disease from my abdomen, but unfortunately, several months later, several small nodules started to grow on my lungs, and it later turned out to be the same colon cancer. So it did metastasize. And unfortunately, because of the locations and the number of these little tiny nodules, I really wasn't a surgical candidate to have those removed. But since I was lucky enough, and I guess I say lucky tongue in cheek, to be KRAS G12C mutated versus other mutations that didn't have any targeted treatments, at the time, both Amgen and Mirati were conducting clinical trials with their, at that time, very, very new KRAS G12C inhibitors. So my wife and I decided that that would be the next treatment option to try next.
Okay, so was it, did your doctor present it to you? Was it presented as an option or what?
You know, that's a great question. My oncologist at the time, you know, you said earlier, you really have to be your own advocate. That is just absolutely true. And my oncologist at the time, he's a wonderful oncologist. He really was provided great care, but he was really only focused on standard of care. He's just like more chemo. He didn't want to do clinical trials, didn't really know much about clinical trials going on. It was not his world. So it's important to find an oncologist that is more in tune with what your ultimate goals are.
What was your ultimate goal?
To be cured.
Okay. Brenda, what was your ultimate goal?
Live long and prosper. To live long and prosper.
Okay, great. All right, doctor. All right, we're gonna put you on the spot. Okay, and so it's like, all right, so tell us. I mean, first of all, it can't be your favorite part of your day walking in and telling somebody like, hey, you need an oncologist, right? But how do you enter into the conversation about a clinical trial?
Yeah, no, so let me say something first. I'm really grateful to you and your team for the invitation. And it's actually a pleasure and honor to be here with you guys today. And you here in the room and online, thank you for taking like a precious time from your weekend and spending that time here today. But this is what we need. We need more people, more voices. I heard the prior speaker was saying about the policy, and this is actually how can it change things. And trust me, doing this for a long time, patient voices is heard and usually have much powerful impact than all of us in the cancer center. So thank you for doing this, and of course, thank you for doing this in the hurricane, middle of the hurricane. So. Yeah.
We don't let anything, once you're a cancer patient, you don't let anything stop you. You like, COVID, cancer, we're like, bring it. We want a hurricane. Like next year, we're hoping to have like a nice, easy year. So.
Yeah. No, I don't want to digress too much, but I think it's important. I always say to my patient, I do believe that hardship brings the best of things in us. So when life is good and everything is fine, like really it's hard for you to know what's going on. But when you go through hardship, you know how strong your relationship, how strong your marriage is, how strong you as a person is. And being here in the hurricane and just insisting to do this, just say how much will all of you and you, Terri, has. So as I say that, thank you for being here and taking the time. And thanks for the invitation. It's really an honor. But back to your question, I think you were asking about clinical trials. So let me just give you a bigger little bit picture of how I see things, and just this is my personal perspective. I'm a GI oncologist, so I do only colorectal cancer and pancreatic cancer. I don't know how to treat lung cancer now, which is sad, but I've been focusing in GI malignancy for very much most of my career. But I think the concept is true for all cancer. Back in the early days, I think when I was in medical school, I'm not going to tell you when, but when I was in medical school, we only had one drug
Okay, well, okay, let me interrupt you for just a second.
sorry.
Oh, no, you're doing great. You're doing great. But in order to do that, like, you're kind of like, my doctor didn't present clinical trials to me. Okay, Brenda was at like a community oncologist, which is where most patients really go, like 80% the statistics show. Okay, and I don't even know as a patient even ask that question, right? I just walking in and it's like, fix me, I got cancer, right? So, okay, wow. I'm feeling the lights are going. But at the same time, so how do I, as a patient, know to ask that question? And how do I go through and pick a clinical trial for me?
Yeah, no, that's a very good question and fair too. But the first one is easy or easier because there is more awareness now, like in TV and radio and those platforms and everywhere about clinical trial. And this not just a rule like physician do, like perhaps even you and other advocates do that better, because when it comes from you, it even has a stronger voice. So I would encourage you, like anyone who knows anyone has cancer or got diagnosed, just tell them, talk to your doctor about clinical trial. Go to your doctor, ask them, okay, do I have clinical trial option, or do you have something for me? And if not, where can I go to check? The second piece is technology now is much better than before. So Twitter, Google, and search. It's actually much more easier to be able to find at least something sources. But I agree with you. I think it's very hard for any patient or most patient, even the oncologist, to figure out where is the best clinical trial and where it is. And I think that has to be a teamwork. At least sometimes you hear, okay, that's particular center, like they really focus on immunotherapy, for example, and maybe we should check that. So the doctor or the team taking care of you really have to guide you a little bit in that direction until you get to the right place. Say, okay, is this where maybe it makes sense for you? So it's a team effort, I think.
I totally agree. It's a team effort. Brenda, do you have anything to? You were part of the team. You jumped right in, but I think as part of it, as how did you know to jump in about?
Well, one of the first things I said when I was diagnosed was I'd like a clinical trial. And, my doctor just looked at me and shrugged his shoulders and said, we have chemo. And that was the extent of it. And I think that was the reason I went to Texas. I was doing well at that point on the initial, it was 3 months after diagnosis. And I just knew there was something more out there that I wasn't going to get in my hometown. It turns out that a trial did come to my hometown. That was chance or divine intervention. I don't know, but at that point, there was nothing for me there, and nor did they encourage me or offer anything. And when I said I was leaving, there was no hard feelings, they were like, well, good luck. But that was at that point, the most I got.
Okay. How about you, Eric?
Yeah, I think, the finding a clinical trial, like I mentioned earlier, having an oncologist who really is in that world is very helpful. Belonging to KRAS Kickers, very helpful. Support groups on Facebook, very helpful. But most of all, I think really, again, being your own advocate, going out on Google searches and clinicaltrials.gov and searching. First of all, knowing what your cancer is, knowing your KRAS G12C, G12D, whatever your mutation is, that's important because you need to find and match up the available trials with your disease to get the best chance of finding a trial that is compatible with what could be most beneficial for you. And I have found numerous trials online that even really there's a growing number that don't even have to do with KRAS specifically at all. A lot of new therapies that were talked about earlier by the doctors today that could be fantastic for almost anyone, either with cancer or with any KRAS mutation. So there's a lot of different sources to find it, but you really have to kind of sit down and spend some time and do some research, and you'll be surprised what you come up with. There's a lot out there.
There is a lot out there, and kind of finding it and narrowing it down. I think for me, it would be like, okay, so how do I compare and contrast? Right. And so some of the considerations would be things like Brenda had mentioned, location, right? There's location. There's like the treatment, there might be the timeline. I know some people have not been able to participate in a clinical trial because they were going to have to be in the hospital for like 6 or 8 weeks. They had young kids. They might be like a single working mom or something. What other considerations should we give to make her decision as far as like, even let's just assume it's the exact same thing, that we just want them each equally as much. What considerations should we give when we're looking at a clinical trial?
So it gets very complicated because, as Eric said, sometimes information is overwhelming, and you really have to have a team that supports you. Just make sure at least you trim this list down and make sure what is the best option for you that makes sense. And what clinical trials that make sense for anyone at some point might be like a month or 2 months or 6 months from now, actually not the right one, maybe a different option. So we really need a lot of guidance from our oncologists and team caring for us. But I do agree with you 100%. You really have to be the advocate for yourself. You have to ask us a question and say, okay, like what clinical trial option I have, where can I go? And frequently, which I see it often, the spouse, either the wife or the husband or the boyfriend or girlfriend, plays that role. And really it's fascinating how much input and help. And frankly, sometimes my patients, their loved one bring me a list of clinical trials, say, okay, look at this, which one makes sense for me? And some of them, I really didn't know they exist. So it's also educational for us. To your question, one of the challenge, and I think this really needs to change, and especially from the policy aspect, clinical trial most of the time must be done for good reason at the center that conducting the trial. And sometimes that means travel, could be like to Texas or like Arizona or somewhere else in the country that come harder. And also there is a lot of time taking, resources get spent and all that stuff. So sometimes logistically it become harder to do that. And I think having access to those clinical trial locally where you live has a huge impact, because that allow people to go and participate on it instead of like taking a month or 2 months off from everything to just go stay in Texas, for example, for 2 months.
It sounds like what you're saying is what Brenda did was a really good idea, is because she went and got in front of doing the clinical trial. She knew that she was going to need something. And so she did the analysis and went to another state to get the information around it. Is that kind of what you're saying?
Yeah, actually, I agree. I think having second opinion at any time and even like maybe initially, maybe later on, is a very good idea, because that's, I always say like if you have two people looking at the same thing, they usually come with a better idea. So second opinion is good. I don't encourage 10th opinion, because this things get confusing and like really hard and time consuming. But second, third opinion, I think it's good, especially if you're looking for clinical trial option. I have a question, if I may.
Yeah, sure, please.
So Brenda, why did you ask for clinical trial?
Well, I mean, honestly, when somebody tells you you're going to die, you need to get another opinion. And the standard of care offered me nothing really long term. And I wanted to live, and I knew a clinical trial was the only way to do that.
Yeah.
I mean, after the standard, which didn't work, the only thing they had for me was docetaxel. And they still offer me that. So I was very happy to go somewhere else and find other alternatives.
Yeah, sadly, our number one reason as a patient to get involved in clinical trial is not really to make a big research difference. It is really to live. And, and that's like, I mean, that's the whole reason we started with it at all, you know.
I'm sorry, I have to say, like, many of my patients also are very generous and they also wanted to help the science and the legacy. But I tell them, very important, that's a secondary. You really will have to focus on you now and you doing better and at least get this under control. So I think contribution to the science, very important aspect, because that help everyone else. But bottom line, we need to do what needs to be done for that person right now.
Yeah, we're all for that, 100%. That's why we want to kick cancer's KRAS. I mean, like simply said, right? We can kick all KRAS, all cancer. We don't even want to kick all the KRAS because we know that some of it we need. So, okay, does anybody have any questions in the audience as far as around a clinical trial? How would you pick a clinical trial? What would you need to know about a clinical trial? Yeah.
During the day-to-day, I checked on my patient portal to make sure of some facts, and there were clinical trials on my test results page from the pathology of my tumor. I never knew that before, so I thought that was pretty cool.
That is pretty cool. And I think a lot of us aren't always getting our NGS testing done. We're not getting our mutation results. We don't know what we are. And once we find out what we are, we're not always sure exactly what that means, let alone what to do with it. And I mean, the statistics in lung cancer, they kind of lead the way a little bit as far as biomarker testing is concerned. And we're still considerably lower than 50% of the patients are being tested. And of the 50% that are being tested here, like less than half of them are finding out what they are, so we can't even get the information. Does anybody, you look like you had something you want to say. Tell us how you found.
Oh my gosh, I have so much to say. My name is Colleen. This is my sister Kalisha. We're from the Georgia area. We just moved here. We just moved here from Virginia, and our mom was diagnosed with stage 4 lung cancer. Never smoker, no family history of cancer. We don't even know where this came from. One day she just woke up not feeling well, found out it was an ulcer. And they did everything. They did x-rays and CT scans and everything like that, and come to find out it was endometrial, and then it was his lung. And so us being of a very big family, about a big faith-based family, we were just like, Okay, where do we go from here? And we're still like that right now, because I never heard of a community doctor. But basically kind of just told us, this is what you do, and this the same day start chemo. No information. I've worked in oncology for 10 years, and talking about clinical trials, I thought it was like that was the last result. And a lot of patients think that that's the last results, like there's nothing else to do except to kind of, give your services to a clinical trial to help later on in the future. But now I'm sitting here and I'm just like, what is going on? Like all of this information, and even now my mom's in the hospital. We weren't going to come, but the tornado and hurricane and everything else going on in the world, but we made it through here because we're like, no, we need information that we're not getting. And right now her hemoglobin is low and we can't get it up. And it's like every day she's getting a blood transfusion. And they did a thoracentesis and then punctured her lung. That's on top of the lung cancer. And we have to wait for that puncture to heal and so forth, but she has a chest tube. And right now we're kind of just like in the same state, like it's not closing. In the meantime, they're not focusing on the cancer, but in the meantime, the hemoglobin is dropping. But when you walk into that, the hospital room, my mom's up smiling and just her faith is so strong. But I mean, right now we just want answers. We just want answers.
I know that we can't fix your problem, but I'm glad you guys are here and we're going to help you get through this. We can't make it better. We can't fix, but we can make it better together. And a lot of it is going through just knowing that we're connected together around clinical trials, and we're going to help figure out whatever we can do. But I'm glad you guys are here. Thank you for being here. But that does kind of like lend to a patient. When is the optimal time to look at a clinical trial or to start a clinical trial? I mean, you just heard. This is, and I wish I could tell you this is the exception. This is standard.
First of all, thank you for being here again and for taking this time. I'm so sorry your mom is in the hospital, but just our prayer for her and you to as well. And this just highlight also something I strongly believe in. I think cancer diagnosis is not one person diagnosis, it's actually the entire family diagnosis. And we should actually make it a community diagnosis because it touches every single one. Like it doesn't matter if you are the husband or the wife or the son or the daughter, it affects every single one in the family and impact them in one way or the other. And also I think cancer care, it's like a group or like a village care. It's not the oncologist or the nurse or one or two person, it's just a group. And perhaps that's the role you're playing here, educating people and bringing awareness and attention to that important topic. But you mentioned something very important, which I think is very commonly people believe in that clinical trial is the last option. I don't think that's true. There is, as a prior speaker, she mentioned there are different kind of clinical trial, and it depends on what kind. Maybe that's actually the last option, but also quite often that's actually the first option. So sometimes, like for example, in colorectal cancer, we had what we call frontline, meaning like patient didn't receive any treatment before. That was the first option using immunotherapy for certain tumor called microsatellite high. But the point is, like it could be the first option, could be the second option, could be the third option or the last option. So it always should be on the list. And a good point I always tell anyone I talk to is when you hear the word cancer, the next thing to it should be is any clinical trial possibility for me or option for me. And then when the cancer starts to build resistance to the treatment that you already on receiving, that will be also a second good time to ask about clinical trial, because most of clinical trial will mandate or require for good reasons that whatever treatment you are receiving now is no longer working, because it doesn't make sense to stop something working to something else. So it's like an ongoing conversation, but I just want to highlight it's really a group effort. And it's never enough to have like resources and talk to people as much as possible. Of course, I mean, cancer is a little bit sensitive and private, but it's always good to seek second opinion, community leader, like advocacy. That always helps.
That's part of what we aim to do is we try and bring this together. I've got another question here. Yeah, Hope. Yeah.
Thank you. So I actually, my connection to KRAS Kickers is my mom has stage 4 lung cancer. She gets her treatment at Wake Forest Baptist. We've had a great experience there, but I work at Atrium Health and I work with health literacy. And one of the things I was wondering is, so my mom, luckily, her oncologist, he told us to do the biomarker testing from the beginning. He got her in a clinical trial. She was on the Amgen trial. She's still doing well in Lumacras. But what I'm wondering is, how do we change the practice of oncologists? Because with health literacy, patients don't know what they don't know. They don't know what to ask because, like you said, we were blindsided. My mom fell. We thought she broke some ribs. All of a sudden, she's got stage 4 lung cancer. We didn't know what to ask. We've never dealt with this. So how do we make it so that the standard of care is every oncologist says, hey, you need biomarker testing. You need to look into clinical trials. Let me help you. Why isn't that a standard practice? Oh, I just wonder.
Sure. No, I think that's a very good question and excellent point. I have to say we actually came a long way over the last few years, especially the last like 3-4 years. Not too long ago, like 17 years ago, we actually didn't do much of testing, and clinical trial also was not like in the center of what we do. And so definitely there is a huge improvement. It's not good enough. Is not what really we would like to be to achieve. But I think we are in the right path. I just wanted to highlight something. Not too long ago, like 3 years ago or 4 years ago, KRAS was something all of us thought there's nothing we can do about it. That's like a common mutation. This is by far the most common mutation happening in cancer, and drive a lot of cancer. And we thought there is nothing we can do about it. And now today we start saying, okay, no, this is not true anymore. There is like, this can be done, that can be done. And I don't think it's gonna stop. I think this is just like gonna continue to go on. But to your point, more voices, more awareness, more platform like Facebook, and like everything has good and bad. Like social media can play a pretty powerful role in this, like Twitter, group like this and others, talking to people. I shouldn't say that because it might be not like a good thing, but I also believe things happen for a reason. And it's amazing that some, at least patient I saw personally, they have cancer, how much change they did for the community and the world. And we are in US leading the cancer care in the world. So I think like the voices here are very, very important and needed change.
Well, and you know, as the KRAS Kickers are a global community, and we're, you know, tumor agnostic. So there's patients around the world, and a lot of times they can't even, one of the benefits we see like in the Facebook group is we'll see people from Europe or India or Australia or whatever, and they're like seeing what we're doing in the States. And first people were like, I had naysayers. They're like, oh, they're going to find that as disappointing. Actually, it's bringing them up to a little bit more awareness as far as what's coming next, and encouraging folks to be able to look at like those clinical trials more first line. So it's actually having kind of like a reverse effect, which is improving everybody's care overall. Now, we did have a question come in line. Okay, as far as like side effects when you're on a clinical trial, there seems to be always consistently liver levels seem to be a particular problem across cancer types. How do you know when you should get taken off the clinical trial, or or what? You know, like that's the beginning, the middle, the end, right? First finding out that you've got a mutation. Now I start the mutation. Now I go something. Now I'm looking for clinical trial. Do you have to come off the clinical trial when your liver numbers are bad or as a result of like, say, immunotherapy or something?
Yeah. So clinical trial now, which is great thing, also is very, very highly and tightly regulated. So like anyone participating in clinical trials, there is a very, very like tight criteria, what should be done, how often it should be seen, what you should get, like things like that. So side effect is potential possibility and real possibility from any drugs we use. And usually clinical trial will mandate like to check, like let's say the liver function every this often. And there is a spelled out criteria that like if that number goes from here to here, there is those steps you need to take. And like it's really very regulated. So you are right, the liver usually get impacted for a lot of reasons, but there is safety and criteria built in the protocol that guide whoever treating that. I don't think you have to come off the clinical trial, depending on the specifics, the criteria and the actual drugs and things like that. But there is also like criteria said, okay, if this happened, patient need to stop the clinical trial and move on to something else.
Okay. And Eric, we haven't forgotten about you yet, but you keep disappearing off screen. So it makes me like undecided I'm on here. So I'm going to pull you back in here in a second. But I do have another question is like, as patients that have like, say a G12R or some other like less common mutation, should they hang around and wait for a specific targeted therapy specific to that mutation? Or should they like consider looking at something else?
You mean no treatment?
As far as like a clinical trial, I mean, should I like, okay, so let's just say I'm G12R, I'm G12D or whatever I might be, right? Yeah. Should I just stand back and wait? Or should I be looking at something else now?
Okay, so it really depends on the actual scenario, and whoever asks the question, I'm happy to chat with them later on offline, because it sounds like there is more details into it. But like if like anything, if you have diabetes, high blood pressure, including cancer, you don't want to leave that untreated. Like people or patients should get some kind of treatment. And if the clinical trial, whatever it is not available or is not an option, so I think we still talk about some kind of treatment should be given to try to suppress and control the cancer. So I wouldn't wait with no treatment whatsoever for the next best thing. But while you are actually receiving the treatment, I think it's a good idea to look for other options too. And I also think it's like a chess game. It is not like one step now. You're really looking for the long term, like 2, 3, 4 steps ahead. And when you are doing that, think for the next step and for the next step and the plan ahead, which help everyone.
Definitely. Definitely. Brenda, were you going to say something? You're just being in agreement.
We're just all looking for the Holy Grail. I mean, you just, we want it.
Yeah, retweet that. Okay. I got to keep an eye on the time here too. Eric, what is your takeaway from what it is that we're saying out here? It truly feels like you're off in a cloud over here. I mean, and you've got the whole moon thing going on behind us.
Well, you were talking about, I just wanted to make a couple of comments here. One of the lady in the audience mentioned something about the liquid biopsy and the information was actually matched up the mutations with clinical trials. And that's great. And that's wonderful. And I have seen that on my own reports. But having seen that, it didn't list everything that was available. So it definitely not a complete list. It's really only targeting the exact mutations that are listed. And there's so many other trials that are, a lot more agnostic as far as what they're looking for. You don't have to be specific KRAS mutation. You don't even have to be KRAS mutated. Different types of things. I'm looking into one right now that is just a completely different approach. And so, really, it boils down to just searching and investigating and just keep looking. And there's a tremendous amount of information out there. I know it can be overwhelming, But get help from your oncologist. My oncologist was the one who just out of the blue came up with this clinical trial that's near Washington, D.C. at the National Institute of Health that could be a great option for me. And, it's something that I never considered before. So, there's places, and I also have to give a strong shout out to my wife. She has done 100% of the investigative and work to try and find all this information. And I wouldn't be here today if it wasn't for her. So, it's important really to not only try to do as much research you can yourself, but just collect as much information as you can. Any family members that can help is great.
It is a community within our own families as well. And Dusty had a question too.
Yes, actually, this is for you, Terri. Can you tell us a little bit about the KRAS Clinical Trial Navigation Service?
Okay, yeah, I'm happy to do that. We wouldn't really call it a service, but part of what we aim to do in the KRAS Kickers is KRAS isn't just a biomarker to us. It is about the KRAS knowledge, the research, advocacy, and all for survivorship. And one of the ways that we do that is not only we're bringing up information into the Facebook group of which most of y'all are in here so that we can engage in different conversations and have those sorts of topics. We also enjoy bringing in the pharmaceutical companies in order to kind of discuss what they've got going on in that realm. We have specific programs that are designed for early clinical and preclinical. And so you guys are going to start seeing a little bit more of those coming online really soon, as far as being able to come in so we can actually pull in the questions. But we also on our website, have a list of the KRAS clinical trials. Now they're KRAS specific. And just because you have a KRAS mutation does not necessarily mean you belong in a KRAS specific clinical trial. You may belong in a different sort of clinical trial, like that may involve surgery, immunotherapy, or whatever. But it's been very difficult for us as patients to be able to know and navigate what it is that we need and what it is that is best for us. And then there's always the option to reach out by email. I mean, I make myself really available, like almost too available sometimes, that if you ping me in Facebook, if you hit me up with an email or almost any way, shape, or form, you just hit me up. I'll do what I can to either connect you to something I may know about, answer a question about it, or point you in a direction that I can get you, whether that's with one of the partner organizations that we work alongside of, because we do not work alone in a silo here. We are a community. And by community, I mean, we are a cancer community, we are a family, and we have to work on and connect with each other and share that knowledge. And so we don't operate in a silo. We build on alliances with the other organizations, whether that's the lung cancer organizations, colorectal organizations, pancreatic, working to expand those connections so that I can have brought in the KRAS focused information and then help connect back out, and then work together. But it was a great question. Thanks for reminding me to highlight it. But I don't want to push us too far back, but I do want to give you an opportunity to say something, Doctor.
Can I have a question to Eric, actually?
Sure.
Because Eric, if I may, I'm seeing you on the screen, but I wanted to get your input. How did you navigate? I know where you live, but from where you living to going to Washington D.C. and getting your treatment? This usually is not an easy process. So can you explain how it went?
Yeah, so yes, it's been a journey. My wife Janet and I started out. We were living in Jersey City, New Jersey. We owned a condo there. I got the diagnosis, complete life change, and after getting some treatment and being close to New York City was wonderful because there are obviously a big city and lots of options there. But really, after having my surgery, the CRS high-tech surgery, and then having these nodules appear on my lungs and no longer being a surgical candidate, there really isn't, and I wasn't responding well to the chemotherapy after that. Clinical trial was really the best option, and the one that my wife found, she found an oncologist and a great clinical trial. It was the Mirati Crystal 10 that was being conducted in Boston, and so I hopped on a train and went straight up to Boston, and this of course was in the height of COVID, and so I was fortunate enough to be able to travel relatively easily and not horribly expensive by train to Boston. And I got on the clinical trial. And because it was something that was going on every couple of weeks, getting treatments, it was expensive. And I had to get some temporary housing up there. We moved up there for about 6 months, and eventually had to sell our condo. And we moved up to the Boston area, and now we're up in Wolfboro, New Hampshire, which is about 2 hours north of Boston. We wanted to stay reasonably close to Boston because the Massachusetts General Hospital was one of the leading research hospitals, and we wanted to stay within a couple of hours drive to that. And all of the clinical trials that we were looking at after the Mirati trial, unfortunately, it kept me stable for about 8 months, but eventually failed. We were looking into other trials there as well. So now that there's other potential travel going on, I just went to Houston to MD Anderson to look at a trial there. Unfortunately, I got there and all the testing and then they said, well, sorry, we have no slots. so that was kind of a setback, but they did have some other trials that were possibilities there. So there's really a variety of options that I have, but yeah, traveling is probably the biggest challenge that I have had with participating on trials. And the Washington DC one, it's reachable by train, and we may spend some time down there. This particular trial, however, it looks like it could be maybe, as most trials are, you have to participate quite a bit in the beginning, but maybe in the first month or so. But this one could be as infrequent as every 3 to 6 months. So, and that's very doable as far as traveling down there from here.
Yeah, it makes a big difference as far as where you are located compared to where the trial is located. And that has a financial implication as well. And that's a really big topic right there. That is just, and anybody who knows about this, or any of the pharmaceutical companies, it goes a long ways when you cover our parking, our lunch, our travel, our expenses when it comes to becoming involved in this clinical trial, because it makes a big difference. Cancer is expensive.
And many clinical trials, by the way, I should mention this, many of the clinical trials, including the one I'm looking into, will cover much, if not all, of your expenses to participate. So look for that. It's a very relevant thing to ask, and there are financial programs. For example, Amgen has their nurse navigator program, and I think they call it Amgen 360. I forget what the name of it is. They have financial assistance programs. They have question and answer, very, very helpful. And I think several of the drug companies have a similar program like that. So definitely look for it.
Yeah, that's a great point. And so, Doctor, I was going to let you kind of like wrap us up.
Yeah, thank you, Terri. I know we are short of time, but Eric, thank you for sharing this story. And this exactly highlights the point. I really think patients should not go to clinical trial. I think clinical trial should come to patient, because not every patient will be able to travel from financial, childcare, health, everything else. So the way we can make this happen, that clinical trial come to every patient where they live, is by changing policy and by more voices from everyone, all of us together. I think Terri, you're doing a great job, you and your team in this, and really appreciate the work you do.
This is live. Okay. I'm not going to let you guys get away because like we get to do this live. And so what we've done with all of our speakers, I don't know if you've heard the last couple of months, is we as a team, as a community, to really show our spirit is in this group here and our participants all said together to kick cancer's KRAS. And so if you're game, okay, they're pretty warmed up. I don't know how warmed up you are, but okay, I gotta stand up because otherwise my knees are gonna give out. Okay, fortunately, I'm like short enough I can actually still get on the camera, I think. Okay, you ready? 1, 2, 3. Kick
Cancer's KRAS.
All right, y'all. Thank you. Thank you so much, Eric.
You're welcome. Thank you for having me. And this was wonderful to be able to participate remotely.
All right, y'all. Okay, I'm gonna pull a name or something here, and then all right, we're gonna pull a name and see what we've got. And if we pull another person that's here in person, I'm gonna scream because I didn't think we put them in here. Kimberly Tagliarino. Boy, you know, I used to think those substitute teachers, they just really would mess up names. What idiots they are. When you pull a name, you have no idea what it is you're looking at. It's a lot harder than you think. Tagliarino. We're gonna go with that. And let's see what I got another one here. Okay, Gladys Honan. Honan? Like that. All right. Are we don't go into a break, or we've got I think we should have Dr. Greenfield up next, right? I can't see the clock. Yes. Is he online? He's good to go. Oh my goodness.
Is.
All right, cool. Okay, cool. I'm just a mess up here on stage. So, you know, keep rolling in there. Okay, well, I gotta tell you, Dr. Greenfield, Dr. Russ Greenfield is an integrative oncologist. I had no idea what an integrative oncologist was or what it was going to mean. And then I come to find out that it is about how to integrate oncology and still have a life while you're going through it. He is the lead physician and integrative oncologist in a cancer center, Novant. Let's see, I'm just like trying to scan through here. Doctor, and how can you, like, they didn't spell it out so I can see it. Is the lead physician, integrative oncology, Weissager Cancer Center, Novant Health, and senior physician consultant at the Office of Patient-Centered Care and Cultural Transformation for the Veteran Health Administration. He completed his residency training, emergency at UCLA, and he was in Charlotte, and he's no longer my integrative oncologist. So like, there's my disclosure slide, because he moved. But he does make a fantastic cup of green tea. And welcome, Dr. Greenfield.
Thank you very much, Terri. And it is nothing less than an honor to be before all of you in this way. And so I want to say really how grateful I am to be here. What I hope to do in our time together is to share with you some of the ideas that honestly I've heard from my patients that have been beneficial for them. There are all manner of options with regards to some of the ways we can get information about what might be helpful to us, whether it be from a conventional perspective, from a complementary, even alternative medicine perspective. And from the get-go, I think it's important to acknowledge how overwhelming some of this can be. I mean, Eric was speaking about his wife and all the work with regards to research and things like that. That's one major component of all of this. But so often the equation that faces us in the midst of this, it looks like an either or. Either we're going to do conventional medicine, or we're going to do this complementary alternative medicine stuff. And the two sides, if you will, just don't talk to one another. And that leaves us very stressed and often overwhelmed, because we get a notebook from our conventional physicians and all these appointments. The internet says something different. This book says something. Our friends are giving us things. The television show says something else, and it's wildly overwhelming. And opportunities like this with meetings with KRAS, to me, this is an opportunity for all of us to pause together and just take a moment and exhale and see, okay, what are some reasonable, rational ways that we might be able to better take care of ourselves, both during treatment and after, in ways that don't add to the overwhelm, but actually are life affirming and give us the opportunity to feel like we have some sense of control in the midst of times that often feel like they're completely out of control. So in this moment, it is an opportunity, if you like, certainly take notes if you want to, etc. But I'm just going to go through some things that the people that I've had the chance to sit across from have shared with me with what has been beneficial about what we've spoken about. I usually tell folks that when we have the opportunity to meet together, there's anywhere from 3 to 25 different things that any one of us might benefit from were we to participate in some complementary alternative modalities. But if I offer any one of the people I'm sitting with 10, 12, 15 different things to do, it adds to the overwhelm. It makes for more stress. And frankly, folks aren't going to be able to do this. I have a number of people that I meet with who say, Russ, I want to do all of this because I really want to optimize my health and well-being. And while I understand that, I strongly recommend against it, because usually when folks do that, within a week or 10 days, they're pretty toasty and they're like, I can't do any of this. And so they leave the entire program to the side. What I would ask you to do again is take this moment to pause and take in some of the things that we're going to talk about and just think about which aspects of this might actually be beneficial for you. Remembering that each program that we talk about for folks coming through the door is individualized for them, for their preferences, for their family, for their unique circumstances, and then trying to make it work within their lifestyle together with the great conventional medical care that they are getting. So some of the topics that come up, we're going to talk about now include, for example, diet. And it's seemingly everybody wants to know, how should I eat during treatment? What's a good anti-cancer diet after treatment, et cetera? And again, there are all manner of experts out there saying all different kinds of things around this. Remember, first and foremost, from my perspective, food should be a source of joy and not a source of fear. And all too often, especially when we go to the internet or some books that are out there, food becomes a source of fear. As an example, we often hear sugar feeds cancer. And that makes us, really nervous and scared. And a lot of us like sweets, and we're like, oh my gosh, sugar feeds cancer. I have to cut all cancer out of my diet. All sugar out of my diet. And to be honest with you, you actually can't do that, because most foods ultimately get turned into sugar in the body in the form of glucose, glucose being the main foodstuff, the main energy source for organs like our brain, our heart, our muscles, et cetera. And so you need sugar to live a healthy life. It's simply that we eat too much of it, and often that we eat it in highly processed forms. So when talking about diet overall, we want it to be a source of joy for you, a source of sustenance, a source of health for you. With that in mind, the occasional splurge on something that may not be quite so healthy is not only something that we think is okay, we recommend it. And the reason is because there are some foods and some activities that might be good for our physical body, and that there are others that help us to be with our friends, with our loved ones, that actually sustain our spirits too. So the occasional splurge, if tonight's the night for, I don't know, pizza and ice cream, have a really good time, and then come back and say, all right, that wasn't all that healthy, but man, we had a good time. And I was engaged in life with friends and I had fun, and I'm going to eat healthier over the next week or so, and then let's do that again. So we want this not to be so much prescriptive as an opportunity, an offering. Generally speaking, what we recommend is that we try to lean towards a pattern of eating that is what we term anti-inflammatory. Inflammation under ordinary circumstances is actually a really, really important part of a properly functioning immune system. However, when inflammation sets up shop where it doesn't need to be, or if it persists longer than is necessary, it may do damage to otherwise healthy cells. So we want to keep inflammation in that healthy range. So what are some of the easiest ways to actually damp down very inflammatory states? Diet and stress management are some of the best too. And we'll talk about both of those briefly. So what does an anti-inflammatory pattern of eating look like? The closest thing to it probably is a Mediterranean style of eating. So that's actually more fish than red meat. It's more vegetables than even fruit. It is whole grains. It's healthy fats like extra virgin olive oil, these types of things. It's not saying you have to go vegetarian, vegan, although a plant-based diet does look like a healthy way to go for many, many people. So you want to work with your nutrition team. You want to work with your family because this is a family engagement, especially around food, and see what might work for you in this regard. There are a couple of really great books out there that I recommend for folks, especially for people who are in the midst of treatment. One book to keep in mind is written by a woman named Holly Clegg, C-L-E-G-G. I have no arrangement with these folks. I'm just telling you stuff that I tell the folks I get a chance to meet with. And that book is called Eating Well Through Cancer. And part of what I really like about that book is they spend a lot of time on what meals might you eat, or what soups might you drink, or what beverages might you enjoy if you're having this side effect or this side effect from treatment, those types of things. So again, taking control where we can in the midst of something that feels a little bit out of control. Another good book that you might take a look at is by Rebecca Katz, and that's The Cancer Fighting Kitchen. Now, that book is also excellent, but it really is a cookbook. And so some folks I meet with say, you know, there's just too many ingredients for some people, but they're healthy and the meals are really delicious. She also has a website called Healing Kitchens that provides some similar recipes that are far fewer in number with regards to the number of ingredients that, again, a lot of our patients really like. Now, you will hear all manner of things out there. You'll hear about fasting. You'll hear about all different kinds of ketogenic diet. You'll hear about all different kinds of things. Again, we want this to be relatively easy for you, and we want it to be safe. And so for a moment there, I want to speak with you about fasting. So there are all different types of fasting programs. There's time-restricted eating, there's modified fasting diets, there's alternate day intermittent fasting, there's all these different kinds of things. And it's okay to speak with your nutrition team and your oncology team or your integrative medicine team around fasting and whether it might be good for you. But there are two precautions at a minimum that you want to take into account here. If you are losing weight in a way that is not healthy for you, if you're not able to maintain a healthy weight and you are losing weight, fasting is not a good way to go. And there is the potential that if we fast extensively, we can become malnourished, which makes our opportunity to respond to treatment much less so. So there are some intriguing aspects to fasting, but we are very, very conservative with our recommendations around this because we want to make certain that people are safe and healthy while they're receiving their treatment and afterwards. A lot of different resources around this type of thing. It's intriguing, it's interesting, but the information is not yet definitive. And so I ask you to be cautious around that. Remember, we want you to enjoy food and feed yourself foods that sustain you not only physically, but spiritually and socially as well. When speaking of food, we always talk about exercise. Not everybody can exercise in the midst of all that is going on for them, but we ask that they engage in some type of movement, if at all possible. Many folks who are just relatively a few weeks out from their surgeries may be in discomfort or may have difficulty moving in certain ways. There are certain forms of physical therapy that are even perhaps more beneficial than straight on physical therapy. And one to keep in mind that you may ask about is called myofascial release. Myofascial release focuses on the soft tissues in the body with a belief that the soft tissues are all interconnected. And that again, we focus on balance in this regard because in integrative medicine, we actually define health as balance. So myofascial release, first and foremost, that you should know about is it's really gentle, and yet it's very, very effective. So if that initial phase of physical therapy didn't get you where you want to go, you might look into myofascial release physical therapy. You might look into acupuncture. Chinese medicine is a complete system unto itself. Acupuncture being just one small component of that. But what's nice about acupuncture is that, amongst many other things, it's individualized to you. It actually is taking a look at your energies, your chi, and seeing if any of that might be blocked, excessive in any way. When you go to meet with, we call them a TCM practitioner, traditional Chinese medicine practitioner. They will get your story, but really the first thing they're going to do is they're going to look at you. What's your color like? Because that gives them information about what's going on inside the body. They check your pulse at your wrist, but not one place like we do in conventional medicine. 3 places at your right wrist and 2 different depths. 3 places at your left wrist and 2 different depths. 12 pulses, each one assigned to something that is going on inside the body. They will ask you to extend your tongue, and they will take a look and see, is it thick, is it thin, is it trembling, is it scalp? Again, more information about the body, and then they will offer you an individualized treatment, because if your chi, if your energy is balanced, all of your systems will function better, and you will respond even better, perhaps, to the treatment with fewer side effects. So to be honest with you, one of the most consistent recommendations that we offer to our patients is to look into acupuncture. Here in the States, acupuncture is rarely covered by insurance, but a more affordable way to explore acupuncture is literally in a small group. So you can have 4 or 5 people, sometimes even more, in a small group in these COVID days, socially distanced. You don't have to get disrobed. The practitioner usually treats only the front of the body, and they usually have the room a little bit cool. There's music playing, you get to meet other people, so there's that community aspect of care. And it's just as effective as one-on-one acupuncture, and usually about 1/3 of the price. Another thing that can be beneficial for you with regards to helping you move after a procedure, or if you have a port in place and being cautious about that, is working with an oncology certified massage therapist. Now, a lot of us look at massage as an indulgence, but I'm not talking about it in that way. This is a therapeutic intervention that can be very beneficial. If for no other reason, when the door closes, when you're lying on that massage therapy table, you get to shut out a lot of the noise. And this is just about you receiving a different kind of healing, because human touch, when it is appropriately applied, is very healing. And again, you get that opportunity in the midst of all the craziness of appointments, etc., to pause and simply receive a different kind of healing. You do want to work with an oncology certified massage therapist, because a lot of massage therapists, as good as they are, may not have training about what areas to stay away from with a person with this type of cancer or this type of cancer. What do we do with a port in place? What do we do when they're post-op, etc.? So look for somebody who's oncology certified. Usually those folks are on the internet with that designation. And then we also, in our space, we offer yoga therapy, but it is yoga therapy. So I'm not advocating that regardless of where you are in the journey, that you go out and join a yoga class, because you'll be treated just like everybody else. The idea about this approach is really about being centered, and almost anybody can do yoga, because it can be just breathing exercises. It can be very gentle twists. It can be very gentle ways to increase our range of motion of our arms or our legs. It's really gentle, but a lot of it is about flexibility, not just of body, but of mind and spirit as well. It can be very gentle, but it should be individualized so that you are not placing yourself at risk or hurting yourself in any way, shape, or form. So looking for a yoga therapist can also be beneficial in helping you to move. If you are able to move on a free basis, walking is one of the best things that you can do, because it gets us outdoors oftentimes. We can do it with a friend or a small group community. You don't have to necessarily run in order to get the benefits. In fact, you can start off relatively slowly if you like. 5 minutes is still 5 minutes more exercise than perhaps we were getting before. And then you can increase that. You might even go a little faster one day. You might even throw in some intervals where you're walking a little bit quicker for about 20 seconds and then walk slowly for a minute. A little quicker for 20 seconds and a little slower. Why do the intervals? It actually kind of tricks our body into thinking that we're actually exercising for longer than we really are. We talked a little bit about diet. We talked a little bit about exercise. Another thing that comes up very commonly is vitamins, supplements, and herbs. Now, there are many, many different agents out there, and there are only a few that we actually get behind when talking with folks about what they might do during treatment and after. During treatment is a different consideration than following treatment. During treatment, there are some concerns, for example, that antioxidants in high dosages like vitamin C, vitamin E, even turmeric is an antioxidant. There is a concern that antioxidants may interfere with certain forms of chemotherapy, and even more concerns that it may interfere with radiation therapy. So yes, we want you to get your antioxidants from diet, from vegetables and fruits and things of that nature, but it may not be the right time during treatment to actually take antioxidant supplements. One of the other things that was brought up, we were talking about clinical trials just a little while ago. You want to be honest and upfront with your clinicians about what it is you're thinking about taking and doing, because if you are taking a vitamin or supplement or something of that nature, that may throw in another variable into the midst of this clinical trial that they can't control for, and that might really interfere with what you're trying to do with the clinical trial, and also what the clinicians are trying to do with learning about what can be beneficial or not. So we want to be very, very careful about using any vitamin or supplement or herb during a clinical trial. Again, making certain that you are sitting across from somebody you can be honest and upfront with about what it is you're doing. Let's take a little turn here for just a second. Every once in a while, I will meet with a person who says they have a really good oncologist, but they just don't have a good relationship with them, but what can they do? This is a difficult concept, but the reality is that you are in charge. When we are patients and sitting across from the doctor, even in the midst of treatment, if that doctor is not meeting us on our value system, or somehow doesn't feel like we're connected, remember that folks are just folks. And sometimes that just happens. It's not a judgment, it's not ill will, anything of that nature. You have the opportunity, in fact, you have the right, and you really should take a look and see, is there somebody else that you could work with? Almost all the oncologists that I work with have no problem with that whatsoever. They just want you better. And so if you're going to be better, if you connect better with this person over here, or you click better with this colleague over here, they will not have a problem with that. What's important is that you are comfortable and that you can move forward safely. Some of the vitamins and supplements that come up a lot, just so that you know about, that you might look up: vitamin D3, turmeric we mentioned just a little while ago. It is a potent antioxidant. It also thins the blood a little bit like aspirin. And so during chemotherapy, if your platelet counts are dropping, it's something we want to be very cautious about. You can eat curry dishes, but you may not want to take the supplement. There are medicinal mushrooms out there like maitake, reishi, something called turkey tail, because it looks like a turkey's tail out in the wild. These are very interesting and might be good for you. However, if you are receiving immunotherapy, these medicinal mushrooms seem to work specifically on certain portions of the immune system, and we don't know if they play well with different forms of immunotherapy. So again, you want to be a little bit cautious about these things. A resource you might like to know about that you can look up online, it is not free. It costs about $40, $45 a year, but it's really good. It's called Consumer Lab. So it's CONSUMERLAB, as in boy, lab. So Consumer Lab, CONSUMERLAB, Consumer Lab. And what Consumer Lab does is it takes products off the shelves and sees if what's on the label actually matches what's inside the pill or the liquid. It's still largely a buyer beware market when it comes to vitamins, supplements, and herbs out there. So having this kind of guidance towards quality product, along with very good write-ups about the various supplements and herbs, can be invaluable to you. We talked about reducing inflammation and using diet, but stress and stress management is another way to make that happen. One of the best ways to do that is what you're doing today, which is coming together in community. In a day and age where we can connect with people all across the globe with the push of a finger, people have never been quite so isolated before, especially with regards to the COVID era. Coming together in some way, shape, or form, having that human connection, being able to share our stories, even if we're not speaking, sometimes makes all the difference in the world and lets us know that we're not alone on this journey. There are many different ways to help manage stress. You have to find out what works for you, but we want them to be healthy choices. Exercise is one of the single best ways to manage stress, to manage depression, to manage anxiety. Better than almost any prescription that we can offer for anyone. So movement works. being in community works. I'm actually a really big fan of good support groups like this one. I underscore good because there are some support groups that just manage to bring us down rather than bringing us up. And so you got to find the good ones out there. You want to be engaged in other kinds of activities too that are more life affirming. Remember, we don't want your 24/7 to be your health to the extent that that is possible. We want you engaged in life and those things that remind you of life and that inspire and invigorate, that give you a sense of meaning and purpose beyond when this is all behind you. And that exploration is critically important too. There's journaling, there's meditation, there's all different things that can be done in this regard. But one of the things that I like about meditation, specific forms like mindfulness, is that many of the people I meet with who have done very, very well, about a month before their next scan or next set of blood tests, whatever, start getting a little antsy. And so we talk about mindfulness as a practice. Now, you may have heard about mindfulness. This is a practice that is borrowed from Buddhism, but is not a religious practice at all. It is about being in this moment. The experts speak about this and say, the past is gone, there's nothing we can do about it. 90+% of what we worry about in the future is not going to happen, but human nature is such that we worry about it anyway. So the only part of all of that we can actually control is this present moment, and this next one, and this next one. The idea being, whether it's a really great moment or a really bad moment, to experience it, to process it, to be fully aware and alert with it, and then to let it go. To not place undue judgment or meaning to anything that happened in that moment. It just is. And so for folks who have completed their treatment or are going for, or in the midst of their treatment, or going for their next scan, whatever, in this moment, odds are pretty good that you're either in the midst of appropriate and helpful treatment, or you're doing pretty well after all of this. And there's every reason to believe that the next studies are going to be okay. And the same thing in this next moment, and the same thing in this next moment. Some great apps available on your phone or computer out there, like Insight Timer is one of my favorites, Headspace, Calm. While sometimes they charge for some of the guided meditations that they offer, there are many that are free as well. And you know, I'm not the kind of person who does a 40-minute meditation, but I will sit for 1 minute when I've got a crazy day going on. I might start my day with 5 minutes or end my day with 5 minutes. It all depends. And you be gentle with yourself in this regard with how you want to explore these things. All of these things we're talking about, they're reasonable options, but they're not mandatory, which leads me to the last thing that I want to offer for you. Now, I have been through internship, residency, medical school, 2 fellowships, and I never got this, what I'm about to share with you through there. I learned this from the people I've had the honor to sit across from. The centerpiece of healing, the centerpiece of healing is kindness. But in this regard, it's kindness towards ourselves. We usually struggle with this because we equate that with being selfish. You're not going to be selfish, but you might act in some ways that are more self-sustaining, that are more self-supportive. The same goes for the significant others involved in the care. For your significant other, if they're invited out to get a cup of coffee or something like that, they might feel like, well, I'm not leaving. No. But every once in a while, it's really important that they do that. So when they come back, they've got more juice in reserve so they can actually be better for you. But for you who are going through or who have gone through treatment, be gentle with yourself. Be kind with yourself. Offer yourself additional cushion. Some of the folks that I've met with say an extraordinary thing, and I'll leave you with this. They say, Russ, in a weird way that I wouldn't wish on anybody, having cancer was a gift. So I look at them quizzically, and they share with me that it has permitted them to say no to some things that they wouldn't have said no to before, and to say yes to some things that they wouldn't have permitted themselves to agree to in the past. So may you say yes to life-affirming things. May you say yes to the things that feed your spirit, your mind, your body, your family, your community, and your environment, and use them together with the good conventional medical care that you are getting. All these things that we were speaking about, any one of them might be a benefit for you. Please do not act upon more than 2 at one time, because otherwise it adds to that overwhelm that we were speaking about. I hope that this was of some service to you. It is simply an honor to be before you. I wish you all well, and I just greatly appreciate this opportunity. Thank you.
Thank you so much, Dr. Russ. We really appreciate your being here. And I think that you're exactly right. Learning to be kind to ourselves. We can't change what was yesterday. And it's just really a lot of it. And it's really brutal. And I get so much out of having a sense of community. And I know that this is really where my heart is and what makes a big difference. I have a quick question if you have a sec.
Sure do.
And we might have a couple others, like, meandering around. And this is going to be, I can only have you speak to it in general terms, but many of us, whether we're on a clinical trial or on any sort of treatment or whatever, it seems like this mysterious liver function is a nonstop battle for us. I mean, is there anything from an integrative point of view that we can be doing?
Yeah, so a lot of folks who go to the internet will read about things like milk thistle or schizandra berries or dandelion tea. These are all things that are used by some folks to try and maintain optimal liver function and to protect the liver. And remember that something that has an action in the body can also have an interaction in the body. And so because of that, we just ask you, depending on the treatment that you're receiving, talk with your oncologist, talk with the pharmacist, if you can, about making certain that these kinds of things might be safe for you. Aside, because remember, all these different things we're talking about, the only thing that my colleagues get upset about when talking about this kind of stuff is when you're talking about pills, when you're talking about vitamins, supplements, and herbs. All the rest of the stuff, yeah, we're all in. And so you just want to remember, safety is the bottom line. We want you to be safe. We don't want to be working against the therapy. Some of these agents actually act on the way that the liver metabolizes certain drugs. And so if you're taking something and the herb or supplement actually increases drug metabolism, you may not be getting as much benefit from your treatment. So yes, those things are out there. You might look into them, but you want to keep everybody on the same page. Again, I share with you that if you come to your doctor and you say, hey, can we talk about milk thistle? And they're like, all this stuff, acupuncture, milk thistle, all this kind of, it's just who he just listened to me. That's when you ask your friends, do you know another doctor? Because you want to be able to work with somebody who is going to be open to anything and everything that might be a benefit to you. There are also a lot of intriguing studies suggesting that Chinese medicine, specifically acupuncture, may help in this setting as well. And so those types of things can be really beneficial. You'll read about all manner of alternative therapies out there. And this is another area that could be spoken about for quite some time. And so whether we are talking about hyperbaric therapy or we're talking about saunas or different kinds of things, some of them might be beneficial, but some of them might not be and might even be dangerous. And so the idea is, how do you discern between what is safe and something I should do, what looks like something that's promising but might be early, what is promising but may have some risk, and what is a waste of time or might at worst be dangerous. And so you want to work with somebody who can help you navigate through those types of things. Most areas around the globe now have some folks who can speak to integrative therapies and all. If not, there are some great resources out there, even on the internet, whether it be some of the great university settings that have websites and things of that nature. You can find out this information in reasonably fast terms so that you don't have to wait so long in order to decide whether you're going to use this. But I do encourage you, even though it's nerve-wracking, to speak with your medical team about what you're thinking about using, simply because we want to keep you safe. I usually tell the folks that I meet with, Terri, that if I say, hey, maybe you should take vitamin D and this medicinal mushroom as an example. There's also an herb called astragalus that we sometimes use. We sometimes talk about melatonin. There's all different things out there. If the oncologist comes back and says, I know where Russ is coming from, but I just can't guarantee that those agents are going to play well with the medicines that you're getting right now, they're right. We don't have the data about all that kind of stuff. And so if I say, hey, you might consider using this, and the oncologist says, I know what Russ is saying, but the problem there is that places us as patients right in the middle. And that's more stress. So the vitamins and supplements are nice ideas, but they're not mandatory. So if ever I run across the oncologist who says, I'd just rather you wait until you're done with treatment to use these, I say, listen to the oncologist, because the vitamins and supplements are nice ideas. They're not mandatory. When you're done with treatment, then we can talk about which ones to use.
It probably pertains as well to anybody who's selling you something, particularly on the website. Just look more than once and talk to your doctor about it, because there are crazy interactions from some of the strangest things. I mean, grapefruit's supposed to be incredibly healthy for you, but it's constantly an interaction on everything under the sun. So if anybody has any questions, I'll bring over the microphone to you. Otherwise, we're asking all of our speakers to join in a little kind of like a cheer, if you will, to let us know that you've got part of our community. And so we're doing it with the team, with the folks that are here live, in addition to you. And I was hoping you would participate. Are you game?
I'm ready.
Okay, cool. So we're going to do like a count off, 1, 2, 3, and then we're going to say all together, kick cancer's KRAS. Okay, y'all are ready. Like, this is really good. And it I should probably make everybody's like stand up on our feet, but I'm going to do that because I can't sit still. Okay, you ready? And 1, 2, 3. Kick cancer's KRAS. All right. Thank you so much for joining us today.
Thank you all. Everybody be well.
Take care. All right, you guys. Well, it's like we're coming up to the end here, and I couldn't think of a better way to kind of like wrap that up than having an integrative oncologist, because the reality of it is that our goal here isn't to get treatment. Our goal here is to live. And it's living with the treatment, living with the cancer, and going through our lives. And like I said earlier, we can't make it go away, but we can make it better by connecting with each other and supporting and sharing each other. And, the KRAS Kickers, I don't know who knows my story and who cares to hear it ever again, but, it's been 5.5 years. And I was the same person I was before I was diagnosed as I am today. But my way is like what Dr. Russ was saying, my way of being able to change the thing I can is being able to have this war cry, name this devil that I'm fighting, this KRAS thing, and being able to share this in the community and having the community spirit, the people, the heart. I mean, and many hands make for a little work, right? And so connecting with everybody here does that, right? I mean, it's the voices in this room, the opportunities that we have, the clinical trials that, we're involved in, or just sharing the experiences or sharing that hope from it is really what makes a big difference. So I'm going to actually, if anybody has any questions or anything, I'm going to open the floor. They gave me 15 minutes at the end of the whole session, and they didn't know what I was going to do. But at the same time, I think that there's just so much more to being able to just engage, whether it's amongst each other or across channels. So if anybody has something they want to say, otherwise I'm going to just I'll even do something that's never happens, and that shuts up something down early, right? Yeah. And as everybody's getting a little wiggly in their chairs here. So if anybody has anything they'd like to say or connect or share. Yeah. Brenda, can we get a wireless mic? Do I have one? Oh, hang on.
So one of the things that the Kickers do is give hope. And I didn't have a chance to give you my hope when I was up on stage, but I wanted to let you know that I have been out of the clinical trial that I was in since June 17th. And my 3-month scan showed no evidence of active disease. So miracles happen. And I wanted to give you that hope today.
3 months.
3 months.
Okay, and all right, now you were diagnosed, and remember the doctor came in and told Brenda, get your affairs in order.
And that there was no targeted therapy yet. I just got off of targeted therapy. So it pays to look into things. There are highs and lows in trials, guys. I mean, I had a cliff in mine. But good things happen. You know, you've got to have hope. I don't know what the future holds, but today I'm good. And what more can you ask for?
Well, thank you for being here. Thank you for sharing your hope. You know, as like the t-shirts say, okay, hope shared really truly is hope multiplied, right? And by sharing it amongst each other, if we keep sharing it and maybe we just keep planting it, it'll keep growing and sharing more. You look like you have something that you want to say. Yeah.
I was just going to say.
Yeah.
Hi, I was going to say the same thing. Belonging to KRAS Kickers has given me hope. I was diagnosed in 2015 with stage 1, and then this past December with stage 4. And I heard stage 4 and thought my life was over, and it was ready to go pick out the plot, and I had to get. They didn't tell me to get my affairs in order, but I'm like in my head, I know my father died of stage 4, and I took care of him. But I happened upon KRAS Kickers, and I just remember breathing a sigh. We were down in Fort Myers on Zoom, the two of us watching. And I just was like, oh, thank God. Thank you for getting me to this group. And I encourage you to join, get involved. And, you know, it definitely gives you hope. It's not the end of the world, which is where I went immediately. That was the end of my life. I was saying goodbye to my girls and my life was over, which it is not.
No, it's a far away from over. And I mean, and that's the whole thing. And whether you're newly diagnosed or oldly, like been diagnosed for a while, right? We get it, you know, and we're not a replacement. We're an extension of family. And I know that like right now your mom's in the hospital. I'm talking to you guys, and right now your mom's in the hospital, but you know, we got it. And we're here for you in any way that we can help out. Okay.
This is from the online, and they're talking about supplements and the whole idea of how do you bring up the topic to oncologists when you're taking supplements?
Question. And doctors dropped off, but I can tell you that everybody in this room has seen a doctor for anything that really felt serious. You're kind of like hesitant to tell them what all you're doing or not doing. I mean, it's bad enough to go to the dentist and they're like, you know, you haven't been flossing every day, right? Okay. I mean, who really wants to admit that you're taking some sort of wonky mushroom or whatever? Okay. But the reality of it is that your doctor can't be looking out for certain things unless they know what it is. And as much as I don't want to get on the scale or make, you know, own up to whether I'm flossing or not flossing, you really need to tell them so that they can do a full evaluation. And how do you bring it up? You just be like, you know, hey, I heard about this. Hey, I'm taking this. You know, what is your take on it? And part of it is just reading what the doctor has to say about it. Because if they're just completely against the idea of taking anything, and maybe I would really encourage you to get that second opinion. I really would encourage you to reach out past that. And at the same time, if they're like, oh yeah, well, whatever, and dismiss it, are they really being fully aware? You know, there's different drugs that, you know, whether, you know, immunotherapy or chemotherapy or whatever. And just from some of the most basic of things from my own personal experience, which is not as extensive as some of y'all here. There's different medications that, you know, if you're on immunotherapy, we would have to change or whatever. And then, you know, part of what they do, I mean, we all know from any sort of treatment, it's like, really, how much blood do they really need from us? But they do have to keep an eye on those things and those liver functions. I mean, that is kind of like cleans out our system. And so they really do need to, it does matter. So they need to know what it is they're looking for. And if it's as simple as you stop taking whatever the thing is that you're taking, hey, that's that much better. So, yeah.
Well, it just so happens that I go to the Weisiger Cancer Institute, and the integrative therapy or integrative medicine group is great. And I made an appointment with one of the pharmacists and I took my grocery bag full of stuff and we went through every single thing I took. Instead of taking away anything, they added one, but I wasn't taking any supplements that were detrimental to my treatment. So,
And didn't that give you a little bit more sense of confidence?
oh yeah, definitely. And I've used several of the integrative medicine services. I haven't got to acupuncture yet. That's next on my list.
I highly recommend it. And that whole massage thing, anytime they're massaging, I mean, like really totally makes a difference. And it is different. Did you guys have something you want to say? Go ahead, Colleen.
I don't know about everyone else, but with me, I feel as though any doctor that comes into the room, especially with like the oncologist or community oncologist, that bedside manners is very important to me. If I feel as though the doctor's not looking at me or looking my mom in the face, or, if I've had one oncologist when I took my mom back to Virginia, that my mom was in the hospital and he pulled up a seat right in front of my mom and looked at her in her eyes and said, "What do you want and how can I help you? And let's go from A to Z." And then I have not found that in Georgia, unfortunately, at this time. And it's like the oncologist that we found is kind of like, if you say to my mom says, "No, I got a headache." Well, it might be nuts, it might go into your brain. And I'm sitting there and my mom's sitting there and I'm looking, and it takes everything out of me to kind of like lash back. But me personally, I think with my family too, yes, we want education, but when I see a doctor go into a room or whatever, I want you not to look at my mom as a patient. I want you to look at her as your mother, your sister, a family member, but what would you tell your family and how would you treat them? So I just want to know if that's something that everyone here agrees.
Oh, 100%, 100%, because I don't want to be seen as just a number. I want to be seen and I want to be known and prioritized. Okay. I am 100% of the patient population that needs to be treated. And so, yes, you need to have that sense of confidence and that sense of trust and that sense of like eye to eye, we're on the same side. Because if you don't have that sense you're on the same side, you've already lost the battle, right? And so that's why we encourage, get a second opinion. Okay. And if you have any question or whatever about it, reach back out, right? And keep looking. Yeah. Again, now if you're seeing 30 and 40 different oncologists, then maybe maybe the doctor's not the problem. Okay. But at the same time, right, you need to have that sense of rapport. And sometimes I would love that doctor to go out to coffee with, but I wouldn't necessarily want them to be my doctor. So yeah, Ellen?
I left 2 doctors because their treatment, they've spent more time in their computer than paying attention to what I was trying to tell them. And one, I had a fight to get rid of it at Sloan, actually, because I was having this terrible pain. And he says, "There, you have a pleural effusion, but let's not upset the apple cart. Let's wait till it gets worse." And I went back to the trial oncologist and I said, "I told you it'd be useless to go there." They wanted me to have it drained, and this guy wouldn't do anything until it got worse. He found me an oncologist and I had a fight to get him with patient reps and everything. Went to the new oncologist and ended up draining a new pulmonologist. He ended up draining a wine bottle worth of fluid. And he thought he was only going to get, maybe 300 cc's, and he just.
Wow, that's crazy.
And another one was at my primary care. All I would do would be type in a computer and ask me questions. And, did you have a mammogram? Did you have a colonoscopy? I kept saying, no, because my oncologist said, they'll find it. You got enough battles right now. Don't worry about the other stuff. And he looked at me and he said, "Well, you better do something and be a little bit more proactive about getting cancer." With that, I stood up and walked out. And I turned and said to him, "I've been battling lung cancer for 3 years now. Didn't you read my chart?"
Wow.
But also you've been doing.
Yeah.
Wow. Yeah.
Now you're doing the acupuncture.
What,
We're gonna hear you, we're gonna hear you, Mark.
yeah, I'm doing acupuncture, I'm doing exercise, doing all this other stuff, and my oncologist I have now approves of it. I went for a second and a third. I don't call looking for a trial. I go call for looking for a second opinion and a third opinion to find out what other doctors in different geographical locations have other treatments that they would think would be appropriate for me at this point. And I've been presented with a couple of different trials and a couple of different treatments.
And the integrative medicine you're doing
Yeah.
as well.
Yeah, I'm doing the integrated medicine.
Really, whether we like it or not, doing two different types of medicine is integrating it. We just need to really make sure that we're doing it right.
Eastern, Western, holistic.
Oh,
I'm mixing
totally.
them all up.
Oh, yeah. And it makes a difference. You know, it really does make a difference. And, you know, being the here, being the now. Okay. Does anybody else have anything? I see everybody at the back table back there. Do you guys have anything you want to say back there? James, Emily, Chris? Oh, yeah. Yeah, James, yeah, give James the microphone.
Emily's talked me into a lot of this. I say talk me into it. She's more of a student of this stuff, and I was more skeptical of this stuff. But I had acupuncture, and it reversed, stopped, and then reversed the neuropathy that I was having in my feet, which makes it neuropathy in your feet makes it hard to run if you're a runner. So it was a good thing to get that stopped. And then all of my supplements are like right in my charts now. So every time I go in there, they want to go over the list, and I just go, nothing's changed, because it's a long list. And but the deal with the doc is just he said a few times, he's like, I'm not sure it's going to help, but it isn't going to hurt. And for me, that's good enough. And now it's on the record. He knows what it is and is aware. So, but as far as bringing it up, I would just say, hey, I want to make sure this is, I get that it might not help, but I want to do all I can do. I just want to make sure it ain't going to hurt anything. So.
Oh, those are really good call outs. Okay. Well, I'm going to wrap this up, but as a community, okay, and I can view this is a community, right? Y'all are extension of our family, and we need to keep connected. And how we do that is through the website and through, the Facebook group and whatever. But more importantly, I mean, connect with each other, right? And connect here. If you want to get more involved, let me know, okay? Because I want you to be the advocate that you need to be. And maybe that's a social media diva. Maybe that's somebody who does administrative stuff. Maybe that's just somebody who moderates a Facebook group, whatever that looks like for you. Okay. I want you to be involved because I want you to be able to, many hands make for little work, and many voices make for changes. And so here we are together. And tonight we're going to take some pictures. We're going to eat some fine food, and we're going to work together, and we're going to kick cancer's KRAS, and we're going to do more. We're going to keep on doing it. And thank you guys for being here, because really, you guys really are what makes my day good. So with that, thank you guys. And thank you everybody online for attending and being here, because you guys are really what makes this powerful. And I'm not just including people in the room as to who can participate. I want you to participate and join us in helping to make things better. Thank you. Bye.